Hello everyone. Morning everyone. I need to test my mic. Okay, testing. Okay, I hope everyone can hear my voice. Okay, so for today class, so good morning everyone. So I think my mic is okay. So for today class is about, I think today and tomorrow is about the division class lah. For those STPM 2023 that already have taken the Ulangan 2 retake for semester 2 next week. I think biology is next Wednesday, right? So so for today, Friday and tomorrow Saturday, I will focus all the topic for make a final revision lah. So for today tentative, I think for those who join our Telegram already know, for today topic I will teach make a revision for first 4 chapter 7 gaseous exchange, chapter 8 transport, chapter 9 control and regulation, and chapter 10 reproduction, development and growth. Okay, so for now it's already 8 o'clock. So for the first hour we will finish up the chapter 7. So hopefully we can finish up lah. So for today is about the revision class. So normally I will more faster lah compared to the lesson that we conduct before. Okay, because this is the revision class, right? So I think everyone should know how to what you will learn already lah. I think okay. Okay, so for gaseous exchange for this chapter, I think everyone know we have 3 topics, right? Gaseous exchange in human breathing mechanism, and also gaseous exchange in plant. Okay, so firstly we will learn about the human respiratory system. Okay, so I think everyone know that our human respiratory system is our lung, right? A lung play the role in the gaseous exchange. So we need to know the lung structure. So normally the location of the lung is located in the thoracic duct, enclosed by the ribs and diaphragm. So this is our lung location. So normally it is located in the thoracic duct. Thoracic duct is the places that dekat kami, I think everyone know lah, thoracic duct is at back, and that is enclosed and protected by the ribs and diaphragm. Okay, so I think this is our lung structure, everyone should know. So in this first outcome, we need to learn about the human respiratory structure. So we need to learn about the respiratory tract. So not only learn about the lung, we also need to learn about the respiratory tract. Okay, so if you see this diagram, there our respiratory tract divided into upper respiratory tract and also lower respiratory tract. Okay, so upper respiratory tract consists of nasal cavity. Nasal cavity is our nose, pharynx and larynx, and then later that we will go to the trachea, and then go to the bronchus, and also our lungs. Okay, so normally our air, when we are breathing, the air will flow into the nostrils with our nose, or will flow into the nasal cavity, and then the air will be filtered by hairs. So not all the air will be come into our body. Some of the air, like the toxic gases or the unreliable gases like nitrogen gas will be trapped at the hair, and then will be warmed, humidified, and sampled as the air flow through the nasal cavity. And then the nasal cavity will lead to the pharynx that link by the auditory tubes. Okay, so next we go to the pharynx structure. So after the air is already passed through the nasal cavity, it will go to the pharynx structure. So you see here, this diagram, pharynx structure is here. This is our pharynx structure. So normally pharynx is located near the esophagus, is the intersection point where the air and food pass through. So if you remember, if you see this diagram, actually you see that the pharynx structure actually is the intersection point between the air flow through and also food go through. So your food later will also pass through the pharynx also. But however, pharynx is normally open to let the air pass through, while esophagus will only open for the foods only. So you see here, after the pharynx structure, they will go to the larynx and also esophagus. So for larynx only air will pass through, and esophagus only the food will be passed through. So it is act as the intersection point that whether the food will go through at here or food will go through the esophagus until into our stomach, and also the air will go through the larynx structure to go to our lung. Okay, so after the air is already passed through the pharynx structure, the air will go to the larynx. Larynx actually is our voice box. Penghasilan bunyi. When we can talk, why we can talk, all the sounds that we produce is due to this structure, larynx structure. Okay, so larynx actually, when the air will after the air already pass through the pharynx structure, it will go to the larynx of the glottis. So it will pass through the glottis at here. So glottis actually is a line between the vocal cord that are the mucosal fold supported by the elastic ligament. Okay, and then we also have the epiglottis. Epiglottis actually is a flap of tissue that prevent the food pass through the larynx. So epiglottis actually is at here at the upper part. Its function is to prevent the food enter to the larynx. And then glottis actually is our voice producer. It will produce the voice, glottis. So normally when the air is passed through the glottis, the vocal cord will vibrate to produce the sound. So you see this diagram, this is our vocal cord. Vocal cord is the sound production in the larynx. So meaning that this is our glottis. When the air is passed through the glottis, this vocal cord will be vibrate. It will vibrate and then it will produce the sound. So if the people that already does not have this vocal cord, or the vocal cord is misfunction, or the vocal cord is already destroyed, breakdown, like when you are sick, when you are demam, all that, meaning that this vocal cord is already have the inflammation occur. Okay, and then when the food is swollen, the larynx will move upward and tips on the epiglottis that locate over the glottis, and then this will cause the trachea to open. Okay, so after the air is passed through the larynx, later it will go to the trachea lah. So trachea is already the lower respiratory tract. Okay, so normally trachea is also known as the wide pipe. It is like a pipe, and then it is opening pipe, and then the air will pass through it, and then trachea later will brush into the two bronchus leading to the lungs, and then the bronchus will brush repeat unit into thin tube called bronchiole. So this is our bronchiole lah. There are many thin unit that already extend to it. This one is we call as bronchiole, and then each bronchiole is enclosed by the multitude air sac called alveolus. Okay, so now next we go to the alveolus. Alveolus is the most important part lah. They play the role in gaseous exchange. So normally alveolus, this is our structure of alveolus. So normally alveolus actually is covered by many blood capillary. You see here there are many blood capillary are enclosed to the alveolus. So normally it's in the large surface area. There are about 100 meters square at our lung. So large surface area can make the gaseous exchange more efficiently. So yeah, because we need to make a gaseous exchange. If we want to efficiency our gaseous exchange, we need to have a large surface area so that our oxygen gas and carbon dioxide gas can be passed through the alveolus more easily. And then the alveolus is very thin cell that only one cell thick of squamous epithelial cell. So this one, why it want to very thin? Because it need to increase rate of diffusion at the lung. So if the cell is very thin, meaning that the diffusion rate is very low, so the gaseous exchange rate will also very low. So this one is not that we don't want lah. Because gas is one of the most important thing to make the human to survive. Okay. So this is the more detailed structure of our lungs that you need to know. So actually, if you see here, this our lung structure. So our lungs also consists of the fluid called surfactant, which make our lungs become moist. And then the surfactant is made up of protein and phospholipid, which coat our lungs and reduce the surface tension. And then each our lungs also covered by network of blood capillary, where it carry the oxygen away to keep the partial pressure low. So this one is actually want to maintain there are the differences of partial pressure between the blood capillary and also the lungs. So if the rate is equilibrium, then no gaseous exchange will be occur lah. So it need to maintain that the oxygen will be carried away, because oxygen when we breathe, oxygen come out, and that oxygen will go to the blood capillary, and then this oxygen will be carried away to the tissue by using the blood. Later we will see it. Okay, so I think that's all for the human respiratory structure. Oh, music. Okay, sorry. Sorry. I forget to close the music. The capillary and also the outlet. Okay, so I already closed the music. Sorry, I forget to eat. I forget to close it. Okay. By using the blood. Later we will see it. Okay, now we go to the hemoglobin structure. So I think this hemoglobin structure, I think maybe you already learned in Sem 1, but I think you already forget because Sem 1 is very far away from you. Okay, never mind. So hemoglobin, I think everyone should be know that hemoglobin is the tetramer structure, which is our one type of the example of quaternary protein structure and also conjugated protein. Okay, that you learn in semester 1. And then we know that hemoglobin contains 4 polypeptide chains, which are 2 alpha chains and also 2 beta chains. Okay, and then hemoglobin will contain the respiratory pigment, which is we call as hem group, that can be found in the erythrocyte. Okay, erythrocyte. So normally hemoglobin is the one of the main key component, key protein component that carry the oxygen and also carbon dioxide play the role in the gaseous exchange. Okay. So I think so this is our hem group. So we know that each hemoglobin each change will contain in the hemoglobin will contain 1 hem group. So altogether there are 4 polypeptide chains, so meaning that 1 hemoglobin will contain 4 hem group here. Okay, right here. So each hem group, so this is the hem group chemical structure. So normally each chain is incorporated with a non-protein prosthetic group called hem. So that is why the hemoglobin is also one of the example of conjugated protein. Okay, and then hem group will give the muscle and blood with the red color. So that is why that when you see our blood and our muscle, why it is red color, because of due to the presence of the hem group. And then each hem group is the polyphene ring structure that encloses the ferrous atom. So this ferrous atom is very important, that carry the carbon dioxide and also oxygen in the blood lah. Okay, and each hem group can associate with 1 oxygen through oxidation, and then meaning that each hem group can carry 1 oxygen molecule. So altogether, since our hemoglobin contains 4 polypeptide chains, so meaning that one hemoglobin will can bind 4 oxygen molecules at once. Okay, so I think this is the simple general structure that you should know. Okay, after that we will go to the transport of carbon oxygen in the blood. Okay, so transport of oxygen in blood is more easier compared to carbon dioxide because it only consists of one pathway only. So normally this you will see this structure. So this is the alveolus, and then this is our blood capillary. So in the alveolus blood capillary, when the oxygen is already reached the alveolus, the partial pressure oxygen for in the blood capillary is very low. Is very low for the blood capillary. Now is very low. Partial pressure of carbon dioxide is very high for the blood capillary. So that due to the differences of partial pressure between these, so the oxygen will be passed through the alveolus and then diffuse into the blood capillary, and then this oxygen later it will diffuse into the erythrocyte. Firstly, it will diffuse into the blood capillary first, any part, not necessarily into the erythrocyte first, maybe it into the blood capillary first, any space in the blood capillary. After that, this oxygen later will diffuse back into the erythrocyte, red blood cell. Okay, and then this oxygen will bind to the hemoglobin, will bind with the hemoglobin to form oxyhemoglobin. Okay, so once it already formed oxyhemoglobin, this oxygen will be transported in the form of oxyhemoglobin, and then this red blood cell will be transported away from the alveolus, go into the active tissue site. So meaning that now the erythrocyte is already reached the tissue capillary that already contain many active tissue. Okay, so this active tissue is require oxygen, so that now at here the partial pressure of oxygen now is oxygen in the blood capillary is very high, but in the interstitial space of the active tissue side, the partial pressure of oxygen is very low. So due to the difference of the partial pressure, so that the oxygen in form of oxyhemoglobin will be dissociated into the hemoglobin and also oxygen, and then the oxygen will diffuse out from the red blood cell into the blood capillary, and then this oxygen will be diffused out from the blood capillary into the interstitial space, and then the oxygen will be used up by the cells that require oxygen, so that it will continue to for the metabolic activities. So we know that oxygen is one of the key component. Why? Because oxygen acts as the final electron acceptor for the cells, right, that you learn in semester 1. Okay, so actually this is the overall for oxygen. So the most important things is oxygen is carried out is transported from the alveolus to the blood capillary in the erythrocyte in the form of the oxyhemoglobin. So that's all for transport oxygen. So it is very easy, very straightforward only. It directly bind to hemoglobin, form oxyhemoglobin, and then directly go to tissue capillary, dissociate into oxygen again, and then this oxygen will be used up by the active tissue for metabolic activity. Okay, so I think this one is explanation. Okay. Okay, so now I will go to the okay, for this one is about the, I just want to mention about the, later you will learn about the Bohr effect, right? Bohr effect and also oxygen dissociation curve. So this one I just want to mention about, if you know the oxygen dissociation curve for the hemoglobin is about sigmoid curve, right? And then once the sigmoid curve, right, okay, meaning that when the oxygen gas is bound to the one of the hem group, actually it will make the hemoglobin more easy to bind the another oxygen molecule. So the further oxygen molecule will more easily bind to the hemoglobin. So that why it is happen, one of the reason is that the hemoglobin will change their shape, alter their shape to make the hemoglobin structure is more easier to bind with the oxygen molecule. So one of the reason lah. Okay, and then transport of carbon dioxide in blood. Okay, carbon dioxide is more complicated compared to oxygen lah. Okay, because carbon dioxide we contain of 3 pathway. The carbon dioxide can be transported in 3 pathway: blood plasma, hemoglobin, and also in terms of bicarbonate ion. Okay, so the blood plasma is the least pathway, the least amount of carbon dioxide will be passed through it. Hemoglobin is the second least, and then the bicarbonate ion is the most. Normally all the carbon dioxide will be transported using in terms of bicarbonate ion. So this is the summary of the transport of carbon dioxide in blood. So normally this one is about the active tissue side. That the cells is already undergoes the metabolic activity, already release the carbon dioxide. So once the cell release the carbon dioxide will diffuse from the cells into the blood capillary. So one of the pathway that the carbon dioxide will not diffuse into the erythrocyte, it will directly diffuse into the blood plasma, and then transported. And then but the remaining carbon dioxide will be diffused into the erythrocyte. Okay, so in the erythrocyte, one of the pathway is carbon dioxide is directly bound to the hemoglobin, bound to the hemoglobin to form the carbanomino hemoglobin. Okay, another one is that we call as bicarbonate ion, meaning that the carbon dioxide will form the bicarbonate ion and then will be transported in the form of bicarbonate ion. Okay, so we will go to the one by one. So the one of the pathway is hemoglobin pathway. So hemoglobin pathway is more easy. It also very straightforward, like the oxygen, meaning that the oxygen will associate with the amino group of hemoglobin to form the carbamino compound called the carbamino hemoglobin. So it directly like the oxygen. So the oxygen bind with hemoglobin form oxyhemoglobin, right? So directly carbon dioxide bind with directly with hemoglobin will form carbamino hemoglobin. So normally the carbon dioxide will be transported in terms of carbamino hemoglobin and then toward the pulmonary capillary, toward the lung lah, and then they will dissociate, and then after that they already reach the lung, they will dissociate the carbon dioxide. The carbamino hemoglobin will be dissociated to release carbon dioxide. Okay, so this bind lower the affinity of oxygen to hemoglobin will force the hemoglobin to release oxygen, become deoxyhemoglobin, and then carbon dioxide will diffuse out through the alveolus to the lungs and then to the atmosphere. Okay, so it's like the reversible pathway to oxygen lah. So it's very straightforward. And another one is plasma pathway. Plasma pathway also very straightforward. It directly, the carbon dioxide will not enter into the erythrocyte. It directly dissolve in the plasma, and then directly the carbon dioxide will be transported into the pulmonary capillaries, and then will go to the gaseous exchange there. Okay, the more complicated one is about the bicarbonate ion. So if you see here, this is the summary diagram. So normally the carbon dioxide already produced from the tissue, and then it will already enter the erythrocyte. Okay, firstly, the carbon dioxide will react with the water molecule, and then to form the one of the intermediate component is about carbonic acid. And then it will using the enzyme we call as carbonic anhydrase. Okay, so once it already formed carbonic acid H2CO3, and then this carbonic acid is the intermediate, it is unstable compound, so it will directly dissociate into the hydrogen ion and also bicarbonate ion. So that is why that it will say that the carbon dioxide is transported through bicarbonate ion, and then this dissociation will be using the same enzyme to carbonic anhydrase. Okay. So if you see here, hydrogen ion and bicarbonate ion, okay, and then this bicarbonate ion will be diffused out from the red blood cell into the blood plasma. This bicarbonate ion will not remain in the red blood cell. It will directly, it will diffuse out from the red blood cell into the blood plasma, and then due to the bicarbonate ion is negatively charged ion, so when it is diffusing out, meaning that the charge in the erythrocyte is imbalanced now. So the chloride ion from the blood plasma will diffuse into the red blood cell too. So it will diffuse into the red blood cell. So this one process is recalled as chloride shift process. So this process main purpose is to balance the electrical charge that already imbalanced in the erythrocyte. Okay, so bicarbonate ion will will be diffused out from erythrocyte, and then it will be transported in terms of bicarbonate ion from the blood capillary, from the tissue capillary into the pulmonary capillary, which the lung. Okay, once it is already reached the lung, okay, the bicarbonate ion will diffuse back into the erythrocyte. Okay, so it will diffuse back into the erythrocyte. At the same time, the chloride ion will be diffused out from the erythrocyte, and then this bicarbonate ion later it will react with hydrogen ion again, to form carbonic acid, and then after that it will form again the carbon dioxide and water molecule, so that this carbon dioxide later will diffuse out from the erythrocyte, and then will go to gaseous exchange, will be passed through the blood capillary and the alveolus wall, and then this carbon dioxide will be expelled out from the alveolus. So this is the gaseous exchange will occur So at the same time, oxygen molecule will be passed through in. Okay, so this is about the bicarbonate ion. So actually it's very simple too. Okay, so this is the explanation. I think I don't need to make more. Okay, so that's all for the transport of carbon dioxide in blood. Okay, next I will go to the oxygen dissociation curve. Okay, so I think oxygen dissociation curve, in our syllabus you will learn about 3 types of oxygen dissociation curve: maternal hemoglobin, which is our normal adult hemoglobin; fetal hemoglobin, our baby hemoglobin; and also myoglobin. Okay, so if you see this kind of graph, this y-axis and the x-axis, if they maybe in exam they want you to draw the oxygen dissociation curve. Not only the curve is you need to draw a curve, and also you need to label about x-axis and also y-axis. So normally the x-axis is about the partial pressure of oxygen in mmHg, and another one, y-axis is the oxygen saturation percentage, meaning that this is the point that oxygen molecule already is fully saturated into the hemoglobin or myoglobin structure, meaning that in 100%, meaning that all the hemoglobin is already fully saturated, all the oxygen molecule is already fully bind to the hemoglobin. Okay, so if you see here these three curve, you will see that myoglobin is in the left hand side one. The left hand side, this red color is the myoglobin curve structure. This the blue color is the fetal hemoglobin structure, and also the green color is the normal adult like us for the maternal hemoglobin structure. Okay, so why it look like this, we will see one by one. Okay, so the most important is the oxygen dissociation curve for hemoglobin. Okay, so this is for the normal one, like us, normal adult hemoglobin. So normally it is what it tell you is, it actually is tell you about the relationship between the partial pressure of oxygen and also the percentage saturation point of the hemoglobin. Meaning that when you see here, where the partial pressure of oxygen is increasing, your oxygen saturation point of the hemoglobin is also increasing. Okay, okay, okay. But if you see here, as the oxygen increase, there is a progressive increase in the percentage of hemoglobin that is combined with oxygen, and then it will produce a curve we call as sigmoid curve. So it is progressive increase. It will not increase like the myoglobin. You see the myoglobin is increased very sharp one, like roller coaster one. But for the hemoglobin, for the maternal hemoglobin, it is actually steadily increased. It will not increase like myoglobin one. Okay, so this shape. So normally this kind of shape, sigmoid curve, will indicate that the first binding of oxygen molecule will facilitate the binding of the second, third, and fourth oxygen. So you see here that when the partial pressure of oxygen is very low, actually the saturation point is also very low. But when the partial pressure is starting to increase, you will find that the saturation point will also very easily to be increased compared to the lower part. Your lower part is not easy to increase one. But when it very become more higher, you will find that the saturation point, saturation percentage is very easily to be reached now. Okay, so this sodium binding behavior is referred to as cooperative. So meaning that the binding reaction in the individual site in each hemoglobin are not independent to one another. So you see this diagram again. So normally when the partial pressure oxygen in 100 mmHg normally is already reached the saturated point lah. Meaning that all of the oxygen is already bound to the hemoglobin already. So you see here. But in the 40 mmHg, normally this one is recalled as resting tissue lah. Resting tissue, meaning that the cell is in the resting state. Like now lah, we are in resting state dekat rehat punya. So normally only 70% of oxygen is saturated. Only saturation point is about 70% only. But when we are doing the exercise, doing exercise, meaning that the saturation point is very low, because the oxygen unloading percentage is very high. Because the oxygen is our cell is require high amount of oxygen for metabolic activity, meaning that oxyhemoglobin will release higher amount of oxygen into the cells compared to the 40 mmHg that the cell is in the resting state. The oxygen is unload very stable. No, because at here the cells does not require large amount of oxygen lah. But when it is in the exercising, our cell is required to have high amount of oxygen, because it need to using for the metabolic activity. Okay, so this is you need to know lah, whether which point, maybe in structural question they ask you which point is about the saturation point in the resting state, which point is about in the exercising state. You need to know lah. Okay, so this is about the hemoglobin structure. So the hemoglobin structure is very simple. That you need to know is it is in sigmoid curve. And then as oxygen concentration increase, it is progressive increase, not increase very sharp one. Okay, but for the myoglobin, okay, so before we learn about the oxygen dissociation curve of myoglobin, we need to learn about the myoglobin structure first. So myoglobin structure is different from the hemoglobin because myoglobin is only the tertiary structure and it is a globular protein. Why? Because myoglobin is only contain single one polypeptide change only. Compared to hemoglobin contain 4 polypeptide change, but for the myoglobin it only contain 1 polypeptide change only. And the same as the hemoglobin, the polypeptide chain contain the hem group. But the difference is one hemoglobin contain 4 hem group, but for the myoglobin it only contain 1 hem group only. So has only 8 helical region, and then since only 1 hem group, so one myoglobin only can bind 1 oxygen molecule only. And then Now, and then the myoglobin, unlike the hemoglobin is present in all part of the cell one, but myoglobin is only present in muscle tissue. Muscle tissue will have the large amount of myoglobin compared to other parts. Okay. So because of only single hem group in one myoglobin structure, that is why that you will see that the curve for the oxygen dissociation curve of the myoglobin is in the hyperbolic pathway, hyperbolic curve one, the hyperbolic shape. So why? Because it is very easy to reach the saturation point, because it only contains one hem group. So meaning that when the oxygen already bind to the only one oxygen molecule bind to the myoglobin, and then it directly will reach the saturation point. Compared to hemoglobin curve, this curve, meaning that the oxygen molecule has the high affinity toward myoglobin compared to hemoglobin. Meaning that if there are two choices for oxygen molecule, if you want to bind hemoglobin or hemoglobin, it will bind to the hemoglobin first, rather than bind to hemoglobin. Because the hard saturation of the binding site is very low. You will see here. If you compare to oxygen dissociation curve, the half of the binding site is at here. Normally it requires maybe 30% partial pressure of 30 mmHg. But for myoglobin, you will see here, normally just maybe 0 point something only, or only 5 to 6, 7 like that only. So it is very low, meaning that oxygen molecule will be easily to bind toward the myoglobin compared to hemoglobin. So this is one of the differences between myoglobin and hemoglobin. Okay, so when maybe in the exam want you to come up with the differences, so you need to know what are their difference. So though in the hemoglobin, it is sigmoid curve, myoglobin is hyperbolic curve, and then hemoglobin is low affinity to oxygen compared to myoglobin, and then the hemoglobin, the half saturation of the binding site is very high, but the myoglobin is low, and then at the same partial pressure of oxygen, the saturation level for hemoglobin is high, but for myoglobin is low, and then the hemoglobin is positively cooperatively. But the myoglobin does not have this property because myoglobin only contains one polypeptide chain, so it does not have this kind of property. But hemoglobin will have this kind of property. Okay, another one is about the oxygen dissociation curve of fetal hemoglobin. Okay, for fetal hemoglobin is about the baby one, the hemoglobin that found in the baby or in the fetus. So meaning that although it is also the hemoglobin, but there are the differences between the adult like us and also their mother maternal, and also the baby. What are their difference? Is that the fetal hemoglobin is made up of 2 alpha and 2 gamma change. This is the key difference. For hemoglobin is 2 alpha and 2 beta, but for the fetal hemoglobin it's 2 alpha and 2 gamma. So meaning that it will bind less strongly toward BPG. Okay, and then the curve shape, although it has the same as sigmoid curve, same as the maternal oxygen dissociation curve of maternal hemoglobin, but the oxygen binding affinity of the fetal hemoglobin is higher than maternal hemoglobin. So if you see here, the fetal part is shift to the right hand side. It is located to the left hand side from the maternal hemoglobin one. So meaning that the oxygen affinity is more higher for the fetal hemoglobin compared to maternal hemoglobin. So oxygen binding affinity of fetal hemoglobin is higher than so meaning that it can allow the efficient transfer of oxygen from mother to the fetus. Because if you know that during pregnancy, I think everyone know, because you are in Ulangan in chapter 4, later we will learn, during pregnancy, our placenta play the role for the gaseous exchange, right? For blood gaseous exchange, also play the role of gaseous exchange between the mother and also fetus, right? So So if the hemoglobin affinity toward oxygen for both are same, meaning that it is in the equilibrium state. Equilibrium state meaning no gaseous exchange will be occur now. And if no gaseous exchange occur, what happen? Baby akan mati lah, memang akan mati lah, because the fetus cannot receive the oxygen molecule from the mother. So we want to overcome this problem, meaning that the fetus hemoglobin need to have the higher affinity of oxygen molecule compared to the maternal one, because fetus is more fetus is require high amount of oxygen from the mother part. So it is very critical for fetal hemoglobin to pull oxygen off the maternal hemoglobin. Okay. At a particular partial pressure of oxygen, maternal hemoglobin will favor unloading while the fetal hemoglobin will favor loading. Okay. So normally you will see here, oxygen will flow from maternal oxyhemoglobin to fetal deoxyhemoglobin. So they are the point. When they have reached the point, meaning that oxygen will flow from the mother side to the fetus side. Okay, so that's all for the oxygen dissociation for fetal hemoglobin. Okay, next we go to the Ball effect. So normally Ball effect is only focused about the oxygen dissociation curve lah. Okay, so meaning what is the Ball effect? So the definition of Ball effect is a regulation of the oxygen binding by hydrogen ion or change in pH and carbon dioxide. Okay, meaning that it is one type of regulation of oxygen binding, the binding affinity of oxygen is affected by the change in pH and also carbon dioxide concentration. So when our hydrogen ion, you will know that, you will still remember that the bicarbonate ion here, you will still remember that in terms of bicarbonate ion in transport of carbon dioxide, so actually it will release hydrogen ion too. So when the hydrogen ion is released in large amount, meaning that partial pressure of carbon dioxide is increased very high. So the pH value will also will be very low. So when this will happen, meaning that Ball effect will be occur. Okay, so why Ball effect is very important to us? Because it regulates the oxygen binding affinity. So if you see this kind of curve, okay, so if you see here, this red color one actually is the normal state. When in the normal state, like in resting state, our oxygen dissociation curve is in the left hand side, right? However, when Ball effect is occur, normally when the partial pressure of carbon dioxide is increasing, Ball effect will be happen, meaning that the curve will shift to the right. You will see here the curve will shift to the right. So what is their meaning? Why it will shift to the right? When it shift to the right, what is it mean? Okay, what is it mean? So okay, so this is the explanation. So when the partial pressure of carbon dioxide is increasing, so meaning that when the carbon dioxide is very in high concentration, the hydrogen ion will also be increased, since more carbonic acid is produced and also hydrogen ion is produced, and then for carbon dioxide transportation purpose resulting low pH value. Okay, so when the partial pressure carbon dioxide increase is mainly due to the high metabolic activity in the tissue. Why suddenly the carbon dioxide partial pressure is very high? Because now the cell is require high metabolic activity. So normally in exercise, when you are doing exercise, meaning the metabolic activity is very high, so the carbon dioxide will be released in higher amount compared to the resting state. So meaning that the now tissue is require high oxygen. Kerana dia hasilkan banyak carbon dioxide, memang at the same time it will need high amount of oxygen. So the hemoglobin will has so meaning that at this point the hemoglobin will has a low affinity for oxygen under this condition, and it will release the oxygen when it is needed at the cell tissue. So meaning that at the point now, the oxygen affinity toward hemoglobin is very low because the cell are require high amount of oxygen now. Meaning that hemoglobin dia tak suka oxygen lah. Dia tak suka oxygen. Dia memang kena release out. So if you see here, that is why that the curve need to shift to the right. Meaning that the affinity of oxygen toward hemoglobin is very low. So that once the oxyhemoglobin reach the cell tissue, it will directly release the oxygen toward the tissue. Dia tak akan maintain lah. Dia tak akan maintain dekat sana lah. Dia memang akan release out in very fast response. So normally you will see here in the summary, when in the lung, the higher pH than actively metabolizing tissue, the hemoglobin will bind toward oxygen, and then hemoglobin will release hydrogen ion. Meaning that at the lung, the pH value is more higher. Higher, and then the hemoglobin will bind toward hemoglobin will bind toward oxygen molecule very fast. But when it reach the actively metabolizing muscle, the pH value now is very low. Due to production of hydrogen ion, and then the oxyhemoglobin will starting to release oxygen gas in very fast manner, and then the hemoglobin also will bind to the hydrogen ion. So this is the summary of the Bowl effect. So the Bowl effect in summary is that the effect will be occur when the partial pressure of carbon dioxide is very high. Why? Because the oxygen affinity toward the hemoglobin now is very low. The cell nak oxygen lebih banyak, one require higher amount of oxygen, so the oxyhemoglobin will release the oxygen in very fast manner. Okay, so this is what we call as Bowl effect. Okay, next we go to the breathing mechanism. Okay, so breathing mechanism, breathing mechanism, I think this one everyone should be know about the inspiration and versus aspiration. So I think I don't need to cakap banyak-banyak what is meaning of inspiration, what is meaning of aspiration lah. So inspiration is normally is that the point that we receive the air, and then aspiration is the point that we release our air. Okay, so normally this is what happen in the inspiration. So our rib cage will push upward and outward, and then the external intercostal muscle is contract, but internal is relaxed, and then our volume thoracic cavity will be increased. Why? Because air is we need to receive the air, right? So yeah, you need to increase your thoracic cavity to increase the maximum loading for the air lah. And then the pressure in the lung is lower, and then radial muscle of diaphragm is contract, and then diaphragm will become flat and move downward, and also air from the atmosphere is easily inhaled. But for aspiration, yeah, your rib cage will push downward and inward, external intercostal muscle is relaxed and internal is contract, and then your volume thoracic cavity is decreased, and then the pressure in the lung is more higher, and then radial muscle is relaxed, and then the diaphragm will become dome shaped and move upward, and then the air inside the lung is easily to be exhaled. Okay, so I think this one is very easy, because I think this one you already learned in STPM, right? So you should know. Okay, so the most important part is about the mechanism. Because although the breathing is involuntary action, everyone will be breathe at the every time, even you are sleeping, you also will breathe. But breathing mechanism is that we need to learn about which point when will happen that when we need to increase our rate of inspiration. why we need to increase our rate of inspiration. Like if you know that if you're doing exercise, your breathing rate is very high, right? If you're doing exercise, you are you need to receive more oxygen and also release more carbon dioxide, right? So your inspiration rate is very high. So why is like that? So because in breathing mechanism, our brain will contain the receptor. So what contain the receptor and also respiratory central and also our muscle. So meaning that receptor is the key point that it receives the signal from the anyway. When it receives signal, dia terima signal, dia akan send this signal, dia macam CPU lah, computer punya CPU. When it receives the signal that we need to increase our breathing rate, it will send the signal to respiratory central. So this respiratory central will modify this signal and then will release to the respiratory muscle. So all this ini either it need to the rib cage is pushed upward, your external intercostal muscle is contract or relax, or internal is relax or contract, or your thoracic cavity or your diaphragm is what like that lah. Okay, so normally in receptor we have two type of receptor: mechanoreceptor and also chemoreceptor. Okay, so mechanoreceptor, another name is we call as stress receptor lah. Okay, the stress receptor that you see in Oxford book, maybe in Oxford book they say is stress receptor, but actually both are same. Okay, so normally mechanoreceptor different is chemoreceptor is located in the brain or in the heart, but the mechanoreceptor is located in the bronchial tree. It located in the bronchial tree to maintain the expansion in the lung. Okay, response to excessive stretching of the lung during large inspiration. So meaning that it actually is help to protect our lung. Because in our inspiration, if in our inspiration process, breathing mechanism, we need to increase the rate of inspiration, but actually our lung also have its maximum intensity lah. The lung also cannot stretching very long time in the large inspiration one. So mechanoreceptor is to protect our lung. So when the inspiration rate is very high, your lung is stretching very long time or very intensity is very strong, so that this mechanoreceptor will be activated, will be stimulated, so that when it is stimulated, it will send the impulse to respiratory central, so that it need to inhibit the inspiration activity. Inspiration activity. So it is responsible for the adjustment in the pattern of breathing to minimize the energy cost of breathing. And then it also prevents the lung overstretching when the ventilatory demand is very high during heavy exercise. So effect of stimulation is lowering the respiratory frequency by increasing expiratory time. So actually it is a protect mechanism. It need to maintain that, to ensure that our lung, the ventilatory rate is in the normal state. Although you are doing exercise, but our lung also have our lung cannot be stretching overstretched at the very long time. So mechanoreceptor, it need to regulate, control the inspiration rate lah. Okay. So the main receptor is chemoreceptor. So in chemoreceptor, we have two types of receptor: peripheral chemoreceptor and also central chemoreceptor. Okay, so what are the difference? The location are different. Peripheral chemoreceptor is located in the carotid and aortic bodies, but the central chemoreceptor is located in the medulla oblongata. So if you see here this diagram, see this diagram, at the medulla oblongata is the central chemoreceptor. Central chemoreceptor. But for the peripheral one, your chemoreceptor is at the carotid and aortic body, at the heart, near the heart. The location is near the heart. What is not on the brain one. Okay, so the function also both function is to detect change of pH, but the location is different. For peripheral, since it is near to the heart, so that it is detect the change of pH in blood. But for the central, it is detect the change of pH in the cerebrospinal fluid. It is a fluid that can only be found in the brain. Dalam otak kami punya. Ini dalam darah kami punya. So both are the same function, but the detection is location is different lah. Peripheral is detected in the blood, and central is detected in the cerebrospinal fluid. So this is about the chemoreceptor, and then respiratory central. So I already mentioned that when the chemoreceptor is stimulated, it will send the impulse into the respiratory central. So we have two respiratory central: inspiratory central and also expiratory central. So normally inspiratory and expiratory central is located in the medulla oblongata. Both of them are located in the medulla oblongata one. So at here, it is actually near to the chemoreceptor one. Okay, so normally the key function is inspiratory central is key play the function in inspiration. Expiratory central is a key function in play the role in expiration process. Okay, so this is the, I just repeat, this is the effector. What are the effects? So when they are both of them is stimulated, they will send the impulse to the muscle, and then all this will happen. When it is inspiration, this will all be happen, and then expiration, this all will be happen. So this is about the effector lah. Okay, so normally when your partial, this is overall of the breathing mechanism. So normally when your partial pressure of carbon dioxide now is very high, if your partial pressure of your carbon dioxide is very high, meaning that your peripheral chemoreceptor and central chemoreceptor now is starting to stimulate. You see here. So when the partial pressure of your carbon dioxide is very high, peripheral receptor, chemoreceptor I already mentioned is located in the aortic and carotid body, right? So it will only detect the pH change of the in the blood, so it will directly. But in the central chemoreceptor, since it detect the pH change in the in the cerebrospinal fluid that we found in the brain, so actually the carbon dioxide will be diffused into the brain first, and then when there are the pH change, when your pH now is increasing, the central chemoreceptor will be stimulated, and then it will go to send the impulse into the inspiratory central, and then it will increase the ventilation rate or our breathing rate lah. Breathing rate. So when your breathing rate is now increasing, so that it will it will ensure that your partial pressure of carbon dioxide in the cell is now decrease. So it is actually the negative feedback mechanism. It need to control the partial pressure of carbon dioxide. So this is another profile. So you see here, when the partial pressure of carbon dioxide is very high, meaning that your hydrogen ion is very high. So it is very high, so that your chemoreceptor inside peripheral chemoreceptor is stimulated. So at the same time, when the high hydrogen ion, your central chemoreceptor will also be stimulated, and then they will send the impulse into the respiratory central. So respiratory central. So when it is in the inspiratory central, so inspiratory central is stimulated, it will send the impulse to the diaphragm and also intercostal muscle through the intercostal nerve and phrenic nerve respectively, and then this will increase the rate of ventilation. And then when the ventilation is increasing, I already mentioned that stress receptor will be stimulated. Why? Because stress receptor is to protect our lung. We cannot let the lung is overstretching in a long time. So when the stress receptor is stimulated, it will inhibit the inspiratory center inspiration rate, so it will inhibit the inspiratory central to send the impulse, but it will send the impulse to the expiratory central to increase the expiration rate. So this is the negative feedback mechanism and then the overall process is to make sure that our partial pressure of carbon dioxide is returned to the normal range. So when it is returned to the normal range, the mechanism will be stopped so but the mechanism will be continue when our partial pressure carbon dioxide is still very high. It cannot return to the normal pathway. Okay, so I think this one is still same. Okay, so this is about the explanation okay. So next is about the lung volume. Lung volume, actually this one is very easy topic so you just need to memorize all this only. Just remember all this only. You need to know that this graph. Lung volume is about the volume of the lung. So normally you see here this diagram is very very easy. So at the yellow color one is about the tidal volume. So tidal volume actually is the volume that in normal pathway, in normal one. When we are in resting state, like now, when we are in normal state, our volume is our inspiration and expiration, the lung only expand in terms of the inside the tidal volume only. So tidal volume is that volume of air inhale or exhale during normal breathing mechanism, breathing. But in the inspiratory reserve volume, you see here, inspiratory reserve volume meaning that is the place that your lung is overstretching, meaning that is the place that your lung need to receive high amount of air, amount of oxygen. So this is an additional of volume that can be forcibly inhaled after a tidal volume inhalation. So when you are doing exercise, meaning that tidal volume is not enough Your volume now is not enough. It requires higher amount of gaseous exchange, higher amount of oxygen inside your body, so that you need to inhale higher amount of air from the atmosphere. So inspiratory reserve volume is the maximum volume that you can inhale. And then expiratory volume is that the volume that you can expel your air from the lung. Air from the lung. And then you see that there are the vital capacity. So vital capacity is the place that the maximum lung volume can be inhaled and also exhaled. And then residual volume is that your air that always remain in the lung and cannot be exhaled even after a maximum forceful exhalation. And then total lung volume is the maximum volume of air that the lung can contain. So this one normally it will come out in exam. It will normally either come out in objective question or structural question. But in structural question normally it will come out 1 to calculate it will give you this kind of graph, and then you need to know what kind of inspiratory reserve volume and what kind where is the, you need to state the value or what is the value of inspiratory reserve volume, and what is the value of TLC, what is the value of vital capacity, like that So next I will go to the last part. Stomata. Stomata. So yeah, stomata. Then now we go to the gaseous exchange in plant. So in human and also breathing mechanism I already cover. So now we go to the plant part. So plant. So firstly we need to know that the stomata structure and function. So stomata actually is the key structure in the plant that play the role in gaseous exchange. So stomata actually is a flank structure at the space between the two gut cells. So actually stomata is at here. This at here. This one we call as stomata. So it is actually flank structure at the space between the two gut cells. And then the gut cell is control the diameter of stomata by changing the shape through the widening the gap of between the gut cell pair. And then gut cell normally is like a kidney shape, kidney or bean shape lah, bean shape. And then the speciality of the gut cell is that the inner cell wall of the gut cell is thicker than the outer cell wall. So this is a key component. And then why is like this? Because it play the role. This structure is play the role in the gaseous exchange in plant. Later we'll see it. Okay, and then gut cell will consist of microfiber which arrange radially in gut cell that play important role in opening and closing of stomata. Okay, so normally stomata is play the role in gaseous exchange. Okay, and also it contain the also function as regulation of water. This one you will learn in chapter 8, right? Transport. Later we will learn it. And then normally water vapor will be escaped through the stomata, and then this ensure that the water can be transported to by the transpiration. So this is about the transpiration pool mechanism. Later we will learn it. Okay, so the mechanism of opening and closing of stomata. Okay, so about the stomata opening. So this one is actually we will learn about the potassium ion accumulation hypothesis. Okay, so why the stomata can be open? So if you see this diagram, this is a summary. So when there are the sunlight, normally stomata is open during the hot sunny day. Why? Because the stomata can absorb the blue light. When it is absorb the blue light from the sunlight, okay, it will activate the bottom pump, hydrogen ion pump. Okay, meaning that the hydrogen ion will pump out from the gut cell to form the electrochemical gradient. So this is one type of core transport, meaning that hydrogen ion is actively transported, pump out from the gut cell. So now, because of the hydrogen ion is pumped out, the gut cell now is imbalance charge. So at the same time, if you want to balance the charge, potassium ion need to enter the gut cell through the voltage activated ion channel. And then at the same time, your chloride ion will also need to enter the gut cell too. So when there are the potassium ion and chloride ion is already entered into the gut cell, meaning that water potential inside the gut cell is very low. It's very low because the solute potential now is very high. Water potential now is very low. So that water molecule from the surrounding spaces, so the surrounding will be diffused into the gut cell. So when the water molecule is diffused into the gut cell, the stomata will become turgid, and then it will open the stomata. So this is the summary process, and then this is about the explanation. I already mentioned blue light from the sunlight will be activation of proton pump, and then this proton pump will use ATP energy to actively transport hydrogen ion out from the gut cell, and then since the electrochemical gradient is formed, so that this one will facilitated diffusion of the large number of potassium ion into the gut cell. Okay, chloride ion will also go into the gut cell to balance the positively charged potassium ion. So the potassium ion and chloride ion will accumulate in the vacuole, thus increasing the solute concentration, and then this water will enter the gut cell surrounding by osmosis, and then this will increase the turgidity of gut cell, change their shape, because the thickened inner cell wall do not expand as much as the outer cell wall. So that is why your inner cell wall need to be thicker compared to outer cell wall. Because only the outer cell wall is expand only. It will expand throughout the outer cell wall to cause the stomata opening. But inner cell wall is not expand at all. The expand of the inner cell wall is very low compared to outer cell wall. So that is why that your inner cell wall need to very thick compared to outer cell wall. Okay. So this is about the stomata opening. So the key point is about the Firstly is about the stomata absorb the blue light, activate proton pump, so that hydrogen ion is already pumped out. It already create the electrochemical gradient. This will result the potassium ion diffuse into the gut cell. At the same time, chloride ion will also diffuse into the gut cell, so that the accumulation of the gut cell potassium ion, chloride ion, increase the solute potential inside the gut cell, so that water potential decrease. The water molecule will be diffused into the gut cell, and then cause the gut cell become rigid, and then the gut cell, the outer cell wall of the gut cell will be expand, and then cause the stomata to be open. So this is overall of the mechanism of stomata opening lah. Okay, that's it about the stomata closing. So closing is more simple compared to opening, because closing is that in opening when the stomata is open during the day, right, during the sunny day, so when at night the stomata is closed, so meaning that the proton pump are inactive, because no light already, no blue light, so the stomata cell cannot absorb the blue light, so meaning that the proton pump are inactive, and then active transport cannot occur, so that the concentration of potassium ion now will decrease slowly, because in sunny day your potassium ion is accumulated here, right, in the gut cell, right. But for now, in the night, since no core transport occur, no active transport occur, so meaning that your potassium ion inside the gut cell was starting to diffuse out from the gut cell. So when the potassium ion is diffused out, similarly your chloride ion will also diffuse out, and then meaning that the solute potential in the gut cell now is decreased. When it is decreased, the water potential was starting to increase. So increase of water potential will causing water diffuse out from the gut cell by osmosis, and then the gut cell will lose their turgidity. So when the water molecule is already diffused out, meaning that the outer cell wall is not expand, so the gut cell is lose their turgidity, and finally the stomata will be closed. So that's all for the stomata mechanism or stomata opening and closing lah. Okay, so that's all for chapter 7. So I already finished up the chapter 7. So now it's already 9:15, more early compared to the schedule, because I assume that I need to finish out within 1 hour and 30 minutes. Okay, so now it's already 9:15, so we will take a break, and then we will continue at 10 o'clock as scheduled. So at 10 o'clock we will continue the chapter 8 transport. So now we take a break first, and then okay, so we see you at 10 o'clock. Thank you. Hello, hello. Hello, hello. Hi. Hello, hello, hello. Testing. I need to see whether my voice is. Hello, hello. Okay, so I think mine is okay. Okay, so for today, so for now, I will proceed to the chapter 8. Okay, so I already close BGM sound, right? Okay. Okay, so now already 10 o'clock. Now I will proceed to chapter 8. 8. Okay, so for chapter 8 is about transport topic. So I think everyone already know that this chapter will have two main topics, transport in mammalian and also transport in plant. Okay, so actually this chapter is quite long. Okay, so I hopefully I can finish up by 12:30. Okay. Okay, so firstly I need to explain about the structure of mammalian heart. So I think that's all. This one everyone should know already what are the heart. So I think this is our heart structure. So if in exam, if exam question, especially essay, they ask you to describe the structure of mammalian heart, you need to mention all of this lah. Not only draw it, you need to also explain all this lah. Okay. Okay, so this is another diagram. Okay. Okay, so the firstly I need to mention is about the size of the heart. So normally in mammalian heart, our size is size of the clenched fist. They are much clenched fist. So what are the size of the clenched fist is like this. It's like this one. So awak angkat tangan awak, memang awak angkat tangan awak memang macam ni punya. Your heart shape, if you compare to your heart shape, memang apa yang tangan awak macam apa yang awak tangan cleft, your heart shape is like that lah. Okay. Okay, another one is heart location. So normally you know that our heart location is located in the left hand side, right? So normally it is located in the chest cavity known as middle mediastinum, directly under the breastbone. Okay, so if you see here, normally heart is located is in the chest cavity as mediastinum and directly under the breastbone sternum. So this is actually the breastbone sternum. If you know that breastbone is our at the tengah-tengah punya lah, at the middle part one. Okay. Okay, so I need to mention about the heart coverage tissue. So we know that the main heart coverage tissue is cardiac muscle, right? So beside from the cardiac muscle, we also have the coverage tissue called pericardium. So you see here this diagram. Okay, so this is myocardium and endocardium is cardiac muscle, and then the outermost layer of the cardiac muscle is we known as the pericardium. Okay, so what is pericardium? Pericardium actually is the connective tissue that encloses our heart. So the outermost layer of the heart is we call as pericardium. Actually, it is a smooth layer of the endothelium covered in the inner surface of pericardium and the outer surface of the heart. Okay, and then between the two surfaces, so we have two types of pericardium, peripheral and also vestibular. So between these two pericardium, we have the pericardial cavity that contain fluid. This fluid is to reduce the friction of minimum as the heartbeat. Okay, next is about the cardiac muscle. So I think the structure of cardiac muscle don't need to mention much because you already learned in semester 1. So you know that we know that cardiac muscle is one of the spatial muscle for the muscle for the heart only. Only the heart contain cardiac muscle only. So there are 3 types of cardiac muscle: epicardium, myocardium, and also endocardium. Okay, and then about the heart chambers. So we know that the structure that separate the heart chambers is known as septum. Okay, so actually it's a heart wall that made from the connective tissue, and then it acts as the separation wall for the right chamber and also left chamber of the heart. So it actually separate the right atrium and right ventricle and also left atrium and left ventricle. So if you see here this, you refer to the diagram, actually is at the middle part of the heart, middle part of the chambers. So there are 2 types of septum: inter-atrial septum and also interventricular septum. So for atrial, I already mentioned atrial. So it is a septum that on the upside one, that the separation between the atria. Okay, and then the ventricular, so it is a separation between the right ventricle and also left ventricle. And then we know that we have the 4 chambers in the heart, right? Left atrium, right atrium, and then right ventricle and also left ventricle. So I already mentioned inter-atrial septum. So there are the septum at the middle part. So inter-atrial septum is the separation between left atrium and right atrium, and then intraventricular septum is separation between left ventricle and also right ventricle. And then we know that in between the left atrium and left ventricle we have bicuspid valve, and then between the right atrium and right ventricle we have the tricuspid valve. And then between the right ventricle and the right ventricle and between the right ventricle toward the pulmonary artery we have the semilunar valve at here. And then similar to the left ventricle toward the aorta, we have also the semilunar valve at here. Okay, so if you see this diagram, I think I don't need to mention much. Left atrium, and then this one is bicuspid valve, and then it go to the left ventricle, and then after that it will have the semilunar valve at here, and then go to the aorta. Similar to the right hand side, have right atrium, and then they have the tricuspid valve, and then they have the right ventricle, and then they have the semilunar valve, and then go toward the the pulmonary artery. Okay, so actually semilunar valve, they have the 2 types of semilunar valve: aortic valve and also pulmonary valve. So aortic valve is spatial for the aorta, the valve for between the left ventricle and the aorta, and then pulmonary valve is the spatial valve for the between the right ventricle and the pulmonary artery. Okay, so if you see this diagram, aortic, this is the pulmonary valve, and then another one is about the aortic valve. Aortic valve is at the behind but in the diagram that are not show, but actually is located in the behind of the pulmonary valve. And then this is about the valve structure. So we know that they have bicuspid and also tricuspid. So bicuspid is at the left hand side, and then tricuspid is at the right hand side. And then the function of the valve actually is to prevent the backflow of the blood in the heart. And then about the blood flow in the heart, I think this one everyone should be know. We know that at the upper side I already separate it. Upper side is about the deoxygenated blood, and then the lower side is about the oxygenated blood. So normally if you refer to the heart structure, meaning that when the deoxygenated blood from the tissue side, they will pass through the vena cava, and then the vena cava will enter the right atrium, and then the right atrium will pass through the tricuspid valve, enter the right ventricle, and then right ventricle will pump the blood to the lung through the semilunar valve, pulmonary valve, and then go through the pulmonary artery, and then go to the lung lah. And then at the same time, when the gaseous exchange is done, the oxygenated blood that from the lung will go to the heart through the pulmonary vein, and then they enter the pulmonary vein. Enter the pulmonary vein, they will enter the left atrium, and then they will pass through the bicuspid valve into the left ventricle, and then this left ventricle will pump the blood through the semilunar valve, or we call as aortic valve, and then the blood will be pumped to all part of the body through the aorta. Okay, so I think this one is very similar, very simple, just that the left hand side, left chambers that transport oxygenated blood, and then the right hand side, right chambers will transport the deoxygenated blood from the tissue part to the lung, and then the left hand side is to transport oxygenated blood from the lung, and then pump to the whole part of the body. Okay, so next I will proceed to the cardiac cycle. So this is one of the very important topic lah. Okay, so in cardiac cycle, what is cardiac cycle? So cardiac cycle actually is refer to the complete heartbeat rate that take about 0.8 seconds. So we know that one heartbeat. Okay, cardiac cycle is that if we want to complete one heartbeat, what will happen in the heart? Because in our heart, the cardiac muscle will not contract and relax at the same time. They will have the separation of the contraction and relaxation in the heart. Okay, so in cardiac cycle, the contraction of cardiac muscle, we cannot use contract and relax. We need to use systole and diastole. This is the scientific term for the cardiac cycle lah. So systole is the period of contraction in cycle, and then the diastole is the relaxation. So systole equal to contract and diastole equal to relax. So in examination, you cannot use the contract, you cannot use the relax in the cardiac cycle. If you use these two terms, then totally no marks is given to you lah. Okay, so in cardiac cycle, we have three main cycle, which is we call as ultrasystole, and then ultrasystole, ventricular systole, and also ultra and ventricular diastole. So got three main phase lah. So it will refer to this. So if you refer to this diagram, so you see this diagram, there are many phases, but in overall there are only three phases: ultrasystole, and then ventricular systole, and also ultra and ventricular diastole. So systole I already mentioned is contraction, right? So firstly is about ultra contraction, next is about ventricular contraction, and then finally is about the ultra and ventricular relaxation. Okay. So in the first step is about the ultrasystole. So in the cardiac cycle, what you need to learn is about that. You need to know that which part is contract, which part is relaxed. First thing. Second thing is about the volume of the chamber. Third thing is about the pressure of the chamber. You need to mention these three key terms inside your examination lah. So inside the ultrasystole, what will happen is the blood will be passed through for the both chamber lah. The blood will pass through from for the left atrium. The blood from the pulmonary vein will go into the left atrium, and then the and then the blood from the tissue will go through the vena cava into the into the left right atrium. So meaning that at here your ultra chamber is contracted, contracted, meaning that your pressure inside the atrium is very high. Is very high now. Okay, so when your pressure is very high, it will force the ultraventricular valve, which is our bicuspid valve and tricuspid valve, to open. So at this time, only these two valves are open only. The semilunar valve is closed. Semilunar valve now is closed. Only AV valve, ultraventricular valve for both chambers are opening. So meaning that the blood will flow through the left atrium to left ventricle, and also right atrium to right ventricle. Okay, so this is the process that the pressure of the chamber, ultra chamber are very high, and then the volume of ultra chamber is decreased, is low, and then the blood will flow through the ultra into the ventricle until the blood is fulfilled in the ventricle, into the ventricle. Okay, so this is the ultrasystole. Okay, next, after ultrasystole, we will go to the ventricular systole. So ventricular systole is happening when all the blood from the atrium is already empty. All the blood is already enter the left ventricle and also right ventricle. So when all the blood is already enter the ventricles, AV valve, the bicuspid valve and the tricuspid valve will be closed now. Okay, so when they are closed, the left ventricle and the right ventricle was starting, the cardiac muscle of left ventricle and right ventricle was starting to pump blood. So that is why that we call as ventricular systole, meaning that the pressure of the ventricles now is very high and the volume of ventricle now is very low. So when the pressure is very high, when it contracts, it will pump the blood. So you see here, it will pump the blood through the semilunar valve. So for the left ventricle, it will pump through the aortic valve, and then pump through the aorta to all part of the body. And then for the right ventricle, it will pump the blood through the pulmonary valve, and then to the pulmonary artery, and then to the lung, so that the semilunar valve now is opening. And then this opening of the valve will create the sound we call as loud sound. So this is the first heart sound that you will listen, it will be produced. So there are total two sounds that will be produced, right? Loud sound and dull sound. Loud sound is the first sound that we produce during ventricular systole. And then why it is produced? Why it is produced with the mitral sound slightly you can hear first sound with the mitral sound slightly before the tricuspid. The closing of the AV valve. The closing of the AV valve and the opening of semilunar valve will create the loud sound. And then next is about the final phase is about the ultra and ventricular diastole. Okay, so this one we will produce another sound we call as dull sound, second sound. So what will happening is that both ultra and ventricle now are relaxing. Relaxed. So meaning that as ventricle relax, the intraventricular pressure will fall. So meaning that both ultra and ventricle the pressure now is decreased. So the ventricle pressure is decreased, and then the volume of the ventricle now is increasing. And then the blood will flow back from arteries, filling the cups of the semilunar valve and forcing them to close. So now the semilunar valve now are closing. So the closing of the semilunar valve will produce the dull sound. So that all the blood, the aorta, the blood from in the aorta and also in the pulmonary artery will not backflow, and then they will go to for pulmonary artery, they will go to the lung. For the aorta will go to pump throughout the body lah. And then at the same time, since now it's already relaxed, so that the IV valve now will be opening, and then it is relaxing, so that the blood from the pulmonary vein and also from the vena cava will be starting to fill in fill into the atrial chamber and also ventricular chamber. So this is about the ultraventricular diastole. So at this time, the most important thing is the semilunar valve is closing, and then this closing of semilunar valve will produce the duct sound in the heart. So this is the second sound will be produced. Okay, next we go to the graph of cardiac cycle for the left ventricle. So if you see this diagram, so we will more focus on the right hand, upper part. The lower part is the ventricular volume and also electrocardiogram that we will not in the syllabus. We will more focus on the upper part. Okay, so if you see here, this graph actually is represent the graph of the cardiac cycle what will happening in the cardiac cycle. So we know that cardiac cycle contain three phases, like ultrasystole, ventricular systole, and also ultra and ventricular diastole. Okay, so if you see here, there are 4 lines you need to know. Is this red line is represent the aortic pressure. So actually this is the pressure graph for the heart. So the upper part, the red line is we call as aortic pressure. So this is the pressure that we measure at the aorta one, in the aorta. The pressure in the aorta one. So in the pressure of the aorta is very high. Normally is very high. And then we will see here the blue line. The blue line is about the ventricular pressure, the pressure that inside the ventricle. And then you will see the yellow line. Yellow line is the ultra pressure. So the pressure that exerted in the atrium. Okay. So if you see here, you will see this diagram. In the diagram, what will happening is you see here is we call as ultra and ventricular diastole, meaning that both chambers, ventricle and ultra chambers are relaxed. And then once it enter here, this point is we call as mitral valve close, meaning it is every valve is starting to close now. So when every valve is closed, what happened? It is meaning that ventricular systole is starting now. So here is about the ultra and ventricular diastole, and then starting here mitral valve close, meaning ventricular systole is starting happening now. When the every valve is closed, meaning ventricular systole is happening now. So the ventricular pressure is starting to increase very high. So when the ventricular pressure is increasing until it reach the aortic pressure, meaning that the semilunar valve will be open. Aortic valve will open now. So will be open so that the pressure, the ventricular will pump the blood from left ventricle throughout the aorta, so that the pressure now is very increased drastically, and then until it reach the maximum, it will starting to decrease. So when it is starting to decrease, meaning that it already reached, it will begin the ultra and ventricular diastole is now to begin. So meaning that the aortic valve, we call as semilunar valve, is starting to close now. So when it is closed, meaning that the ventricular systole is already stopped, and then it will decrease until here the every valve will open. So when the every valve is opening, ultrasystole and also ultra and ventricular diastole now is happening now. So it will be back to the ultra and ventricular diastole again, and then ultrasystole is begin, and then it will repeat again. So meaning that at this point is ultra and ventricular diastole, and then starting is here, you will see that there is a starting slightly increase here, meaning this point is a point that ultra systole is happen. But when the AV valve is closed, meaning that ventricular systole is happening now, and then it will directly increase until here, the ventricular systole will be stopped, and then until here, the AV valve will open again, and then it will repeat again. So the most important thing is you need to remember the point at here. What are the points happen at here? What are the points happen at here? So there are the 4 points you need to remember. So here, meaning every valve is closed. This one is seminoral valve is opening, and then down after the ventricular systole, seminoral valve is closed, and then it will proceed to the every valve open again. So it will repeat the process again and again. So you see here, and also this point is the point that the first sound of the heart is produced. So when the AV valve seminoral valve is opening, the first sound will be produced. The lap sound will be produced, and then the second sound is produced when our AV seminoral valve is closed. So it will produce the second sound. Okay. So this is very important. Normally it will come out in objective question, even though in structural question maybe it will come out too. So you need to remember it lah. Okay, next I go to the heartbeat initiation. So we know that heartbeat actually is involuntary action, right? Our heartbeat is continue to be jump for every time. Even though you are sleeping, your heartbeat is still continue, right? Only the people that already die, you will not have any heartbeat. However, so what make the heartbeat jump? Okay, so there are the many electrical impulse will be produced in the heart. It is automatically electrical impulse. Okay, so the most important one is the sinoatrial node lah. Sinoatrial node and also ultraventricular node. These two are the main electrical impulse will be produced in the heart to make our heart will be jump. Okay, so this one is more clear. So sinoatrial node, ultraventricular node, and also we have along the ultraventricular node we have bundle of His, and also have the Purkinje fiber at here. So this is actually we call as the neuron cell, nervous tissue that inside the heart that play the role to generate electrical impulse to the heart to be jump. Okay. So we know that sinoatrial node is act as the pacemaker. So when the SA node is stimulated, it will send the electrical impulse to the both ultra, left ultra and also right left ultra and also right ultra, and then it will make the ultra to contract. So that is why our cardiac cycle is starting from the ultra contraction, ultra systole, because sinoatrial node is act as the pacemaker. It will automatically send the electrical impulse to the ultra. Okay, so when it is sent to the electrical impulse to the ultra will start to contract. So ultra systole will happen lah. Okay, so later that it will also send the electrical impulse to the AV node. Okay, so what it want to send to the AV node? Why don't want it directly go to the impulse to the bundle of His or Purkinje fiber? Because AV node will delay the impulse first. Why you want to delay? Because it need to make sure that all the blood in the ultra can be already empty and flow into the ventricle. If no AV node, it will directly send the impulse into the ventricle part, and then it will directly contract. So that it cannot ensure that the blood in the ultra is all are the empty already fulfilled in the ventricle. So you need to AV node. So it will delay the impulse first. So once the ultra systole is completed, AV node was stimulated, and then it will send the impulse through the AV bundle and also AV bundle of His, and then it will go to the right and left hand side right hand side and left hand side of the bundle, and then will go toward the particular fiber, and then this electrical impulse, once it reaches the particular fiber, it will conduct the impulse to muscle fiber for both ventricle. So it will make the both ventricle to be contract like this. So every node is played a very important role because it play the role to delay the signal first. Once the ultra, all the blood in the ultra is already in fulfilled in the ventricle, then the every node will be stimulated and then send the impulse to the heart. Okay. So that's all for the initiation of heartbeat. So it's very easy lah, I think. Yeah. Okay. So this is the explanation. So I think I already explained many on completely lah. Okay. Next is about heartbeat regulation. So although the heartbeat is the involuntary action, we will have a heartbeat anytime. But when you are doing the vigor exercise, or you are in panic situation, your heartbeat will be increased, right? So what regulate our heartbeat? Okay. So the first one is about the nervous system. Okay, nervous system. So our nervous system play the role in the heartbeat regulation. So when there are the sensory receptor in the wall of the certain blood vessel or in the heart chamber that detect the blood pressure change in the blood, sudden when our blood pressure is increased or decreased, it was stimulated, and then the receptor will send our receptor will be stimulated, and then the receptor will send the message to the cardiac center in the medulla oblongata of the brain. So when the receptor is already stimulated, this cardiac center will convert two sets of autonomic nerve that pass to the sinoatrial node, and then it will go toward the parasympathetic and the sympathetic nervous system. So the impulse was sent toward the parasympathetic nerve or sympathetic. So as you guys know that parasympathetic, and sympathetic, right? So parasympathetic will release acetylcholine, and sympathetic will release noradrenaline, right? And then parasympathetic play the role to decrease the heartbeat rate. It is play the role in decrease the heartbeat rate, and sympathetic is to increase the heartbeat rate, right? So this one actually you already learned in chapter 3, right? Although I not teach yet, but you are in Ulangan, you should know lah what is the parasympathetic and sympathetic. Okay, so actually acetylcholine, so this one is about the nervous system. It is more complicated to the chapter 3, synapse. So we know that in parasympathetic system it will release acetylcholine, right? And then we know that acetylcholine is increase the heartbeat rate. So how acetylcholine increase the heartbeat rate? So when the acetylcholine is released from the synapse into the synaptic cleft, and then acetylcholine will acetylcholine will bind toward the acetylcholine receptor at the postsynaptic membrane at here, and then postsynaptic membrane, and then this receptor protein will activate the G protein, and then this G protein will bind the potassium ion channel, and then to make sure that potassium ion channel is opening. So when the potassium channel now is open, meaning that hyperpolarizing membrane action potential will occur more slowly. Meaning acetylcholine will decrease the rate of depolarization by increasing the membrane permeability to potassium ion. So now potassium ion is more permeable to the membrane, the cardiac muscle membrane, so that potassium ion will pump out or diffuse out from the muscle membrane to the synaptic cleft lah. So now if you still remember about the sodium ion and potassium ion that you learned in action potential, so when the potassium ion is pumped, diffuse out, it will slow down the rate of depolarization. So slow down the depolarization, so action potential will be occur lah. So slow down the depolarization rate. So depolarization rate so that the action potential will occur become more slowly. So it already, because depolarization rate is the phase that if you still remember about the action potential, depolarization rate is that the potassium ion channel is diffused out, right? So when the potassium ion channel is diffused out, meaning that it already want to generate a new signal of action potential. So for now, it already slow down the rate of depolarization, so meaning that it also slow down the action potential too. So the time it want to generate another time of action potential now is decreased, is limited. So this is the function of acetylcholine. So at the end is that the acetylcholine will decrease the heartbeat rate. Another one is about the noradrenaline. Okay, so what that happen is when the sympathetic neuron release the noradrenaline, same as the before that you learn in synapse, it will bind to the receptor, and then this receptor will activate the G protein. But this G protein later it will activate the enzyme which convert ATP to CAMP, and then CAMP will activate the protein kinase. Okay, so this protein kinase will phosphorylate calcium ion channel that they open more easily when the neuron is depolarized. Action potential will be occur more easily. So meaning that this protein kinase will phosphorylate calcium ion channel so that it will open the calcium channel. So that the calcium ion from the synaptic cleft will be diffused into the cardiac muscle, so that it will make the cell depolarize more easily. So when the depolarization rate is increasing, meaning that it will easily to generate the another type of action potential, so that the noradrenaline will speed the heart rate and increase the strength of contraction by stimulating calcium ion channel opening during depolarization. So yeah, so this is about the sympathetic system, how the noradrenaline increase our heartbeat rate. And another one, not only nervous system, our adrenal gland also play the role in the increase our heartbeat rate. So normally adrenal gland will be in secrete adrenaline and also noradrenaline, so it respond to the flight and fight response, and then it will increase our heart rate lah. Okay, so that's all for the regulation of heartbeat. Now I will proceed to the cardiovascular disease. Okay. Okay. So let me for a while. I need to go to washroom first. So maybe you guys can digest first what you learned before. So I need go to washroom first. So we see you at 10:40 lah. Okay. Hello. Okay. Okay, so we continue the class. Okay, so now I will proceed to the cardiovascular disease. Okay, so about the cardiovascular disease. Okay, so actually this is very hot topics. I think you refer to past year, I think come out many times. I think already 5 to 6 times already. So this is very very important topic. Okay, so in cardiovascular disease, so there are the many terms that you need to alert. One of the terms is about the arteriosclerosis, and another one is about the atherosclerosis. You need to alert these two, because these two terms are totally different. Totally different. Arteriosclerosis and also atherosclerosis. The most that come out in exam is about atherosclerosis but actually these two terms are totally different. So what is the meaning of arteriosclerosis? Actually it is a condition that occurs when the arteries wall become hard and stiff that can already interfere with blood flow and disrupt the normal working of your circulatory system. So meaning that it is actually the hardening and stiffening of the artery wall. Artery wall, not artery. You need to mention artery wall. So any process that we about the hardening process and the stiffening process, and then this process already interrupt the blood flow this one we call as arteriosclerosis. So it is the general terms only. So under arteriosclerosis there are three main classes: atherosclerosis, arteriolosclerosis, and also monkey birch media calcified sclerosis. So in our syllabus we will more focus on atherosclerosis. These two don't need to focus. These two is not in your syllabus. So although it's not in your syllabus, just absent it lah. Arteriosclerosis normally it already mentioned arterial, so normally it is about the it involves the arterials wall. Arterials wall only compared to the atherosclerosis. Atherosclerosis is involve all the artery wall, but this one is only include arterial, the smaller one, the smallest artery wall. Okay, and then there are two types also, hyaline and also hyperplastic, but you don't need to learn. Another one is monkey birch media, so this one is actually is calcification process, but you don't need to learn too. Okay. But this one you need to learn is our syllabus atherosclerosis. So what are the definition of atherosclerosis? It is actually one type of disease where the interior wall of the artery, the keyword interior wall of the artery, are damaged, inflamed, and narrow as a result of lipid deposit in their wall. The keyword is interior wall of artery. You cannot say wall of artery, you cannot say artery only. If you write artery or wall of artery, then no marks given. You need to mention interior wall of artery. And what happened is it inflamed, and then the lumen of the artery are narrow as a result. Why it is happening? Because of lipid deposit. It is due to the lipid deposit or lipid accumulation in the wall. So you see this diagram, you see that actually in normal people, normal human, your lumen is actually spacing like this one. This white color is lumen, but actually your lumen space is should be whole part of here. But for now is already, you see here what happened now is your lumen now is very narrow due to the formation of plaque and thrombus at here. The plaque formation and also thrombus formation already block your artery, block your lumen of the artery. So meaning that the blood only can pass through at here only. And then if it is more serious again, it can be totally blocked at all. So if it is totally blocked, then will have the many dangerous effect, serious effect to you . Okay, so about the development of atherosclerosis. So what are their development? Okay, so the development of atherosclerosis is I I complete to four parts: damage to endothelium, fatty streak formation, lipid plaque lesion, and also complicated plaque formation. So what are the first step? Okay, so first step you will see, I think, okay, first step is about the damage of endothelial cells only. Okay, damage of the endothelial cell. So when the people already eat more lipid molecule, normally is LDL lah. The person that consume high amount of LDL cholesterol or the many high amount of lipid food, it will damage the endothelial layer of the outer V. So endothelial layer, I think you will see this diagram. Endothelial actually is the inner cell wall, epithelial tissue for the outer V. So when our endothelial layer is damaged, now is starting to damage, meaning that your smooth muscle was starting to migrate from its original position, and then your macrophage was starting to activate. Why? Because if lipid is too much at here, it already damaged your endothelial layer. So when it is damaging, your white blood cell like macrophage will detect that there are something happen at here. Okay, so when there are something happen at here, your macrophage was starting to engulf the lipid molecule. However, the purpose of macrophage is to engulf pathogen, not lipid. But now your macrophage assume that your lipid molecule is one type of pathogen. So it's starting to engulf. But in real function, that macrophage cannot digest lipid. It only can digest pathogen because it only contain lysozyme only. It does not contain lipase. Can hydrolyze lipid? Cannot. So this one will causing that fatty streak formation, meaning that the macrophage and your smooth muscle tissue was starting to engulf lipid due to they think that the lipid is the pathogen, but when they starting to engulf, they found that they cannot hydrolyze it. So now the macrophage will now starting to form the foam cell. Foam cell is the macrophage that already accumulated with the lipid molecule only. Because it cannot digest the lipid molecule, so it was starting to form cell and accumulated at here. All the macrophage were accumulated at here. It will cannot move to anywhere one. It will accumulated at here. Okay, so what will happen is the third phase is about the lipid plaque. At lipid plaque. So at the end, at the longer time, when you in more longer time, your smooth muscle was starting to proliferate, and then your collagen and other ECM deposition and extracellular lipid. So all the smooth muscle tissue and your lipid molecule, and even though your foam cell and other collagen was starting to accumulate at here. And then your smooth muscle tissue was starting to proliferate at here. Starting to proliferate at here. So it need to respond to injury, because now is your blood artery is already injured. So your smooth muscle tissue will assume that you already settle. This gun cannot settle already, so that it will form the new layer of muscle tissue at here, and then at the part, all the lipid debris at here. So finally, you will form the plaque. Complicated plaque will be formed, and then if the plaque is rupture, meaning that this one is we call as plaque, and if this plaque is already rupture, already released out from the artery wall, it will form the thrombosis. Okay, so you will see this diagram lah. So you see here, this one is we call as plaque, and then when this plaque rupture, it will form the thrombus here. So it's very serious lah. So you see this diagram too. So this one is normal one, and then if the atherosclerosis happen, it will form like this, like this one. All these inside here, all are the lipid component, and also the smooth new layer of smooth muscle will be formed at here. So that is why that it will narrow down the lumen of the outer V. So you see here, this is the explanation. So normally is due to the lipoprotein, LDL, bad cholesterol. So the large glycosylates will starting to phagocyte them, and then large number of foam cell will transform. Foam cell will be produced to the development of fatty straight throughout the initiation of the progressive of the atherosclerosis, and then underlying smooth muscle fiber migrate to the inner lining of the artery and multiply them. So it will form a new layer of muscle tissue at here. So because the smooth muscle tissue will assume that this layer, assume that this is the normal lah. Dia akan assume ini macam normal lah. So dia akan form the new layer at here. So if you see here, the normal one, the endothelium tissue, the below part is smooth muscle tissue. But for now, if no smooth muscle tissue, endothelial layer akan pecah punya. So if we want to protect the endothelial layer, so the smooth muscle tissue need to form at under the endothelial layer. So it will macam jadi bengkak lah. Dia akan jadi bengkak dekat sini. Okay, so the build out fatty substance, smooth muscle tissue, macrophage will form the atherosclerotic plaque, and then this plaque increase, remodeling of the artery will take place, so that the time of the size of the lumen will be maintained, and then smooth muscle fiber will form a protective fiber scape which wall of the plaque from the inner lining of the artery and the blood. And then macrophage and other cells will eventually die within the plaque, recruiting more macrophage to the area. So they akan when the macrophage is die, they form the debris. So they will stimulate more macrophage inside to it. So it will become very serious. It will become more serious compared to the before one. And then when the plaque become thicker, it narrow the lumen of the artery and even captured by thrombosis to form thrombus. So this is about the development of atherosclerosis. So there are 4 main stages you need to know. The endothelial layer is damaged due to the lipid, and then fatty strip formation, lipid plaque lesion, and also complicated plaque. And then this is about the differences between atherosclerosis and arteriosclerosis. So arteriosclerosis actually is a normal name. So it involves the hardening and thickening of artery wall. Okay. And then another one is about hypertension. Okay, this one is also the so this one is about the hypertension, another type of cardiovascular disease. So this is a main cause of the cardiovascular disease. So before you get cardiovascular disease, you will get hypertension first. Hypertension is the main cause of cardiovascular disease. So for those who are the hypertension people will have the higher ability to get the cardiovascular disease. So as you go that hypertension is high blood pressure. So 120 over 80 mmHg is our normal blood pressure. So this one is about our systolic blood pressure, and then this one is about diastolic blood pressure. So this is the classification of the blood pressure. So normally that the patient have the higher than 140 for systolic part, and then higher than 90 for diastolic part will be considered as hypertension patient. Okay, so hypertension relate to how hypertension can be related to cardiovascular disease. So we know that hypertension is about high blood pressure. So blood pressure is a result of the blood flowing through the artery and the resistance of the artery wall. So when the blood is passed through the artery, and then they akan macam have the resistance, right? Or they akan sentuh artery wall, right? So it is a result, the combination between these two. So when there are the resistance, if in normal situation your artery wall is not have many resistance, dia tak ada halangan punya. But if there are many halangan, like when you are forming a blood at here, or they form blood at there at certain place, this one will cause that your blood, or will cause that your heart require more intention to pump the blood. So your blood pressure is required to very high, because dia ada halangan lah. Halangan kat sini, dekat sana, memang tekanan dia perlu meningkat. So if it take more energy to get the blood through the artery, then your heart has to work harder than if each beat. So for normal people, maybe the heartbeat is only 120 100 or like that, 100, but in hypertension maybe your heartbeat rate is up to 150 160 like that. So yeah, and then next is about the effect of the atherosclerosis, the serious effect, which is we call as myocardial infection. So heart attack another, the normal name is about the heart attack. So what are the meaning is, it is actually the damage or death of cardiac muscle tissue resulting from blockage of coronary arteries. So this coronary artery, so if you know that our heart, although it plays the role to pump the blood, but our heart is also one type of cell, right? One type of organ. It is also one type of organ. So if it is organ, it is made up of the cell, so it also require metabolic activity too. So there are also it also require oxygen, it also require nutrient, it also require carbon dioxide, oxygen to carry out metabolic activity, so that the heart can pump the blood. But so there are also the artery and the vein, we call as coronary artery and coronary vein, that transport the blood into the heart, especially for the cardiac muscle to pump the blood lah, to have a metabolic activity. Okay, so the myocardial infection is caused when your coronary artery in the heart that supply the blood into the heart is already blocked. So when it is already blocked, we see here is already blocked, meaning that your blood cannot pass through it. So when cannot pass through it, then your cardiac muscle will be damaged or die. If your cardiac muscle is die, then heart will be stop beating, and then the person will be die. So that is why the patient that got heart attack, if they not get the immediately treatment, they will die immediately, because heart is very important. We cannot say that we can the people cannot have if the heartbeat is stopped, the people cannot survive in maybe certain minute only. Okay, so this is about the explanation. So normally I just want to explain about the thrombus. So normally myocardial infection can be formed obstruction by blood, or maybe they already have blood at here, or maybe have the thrombus. So I already mentioned thrombus is a rupture of the blood. So this thrombus, when it is ruptured, meaning that it can flow throughout the arteries wall, arteries lumen. Dia boleh bergerak ke mana-mana tempat. So it is very dangerous, meaning that when this thrombus, maybe at some places the blood artery diameter is more longer, so the thrombus can be passed through. But when it reach the coronary artery, normally the coronary artery, the diameter of artery is very short. It's very short. So when the thrombus is at here, it will totally block the flow of coronary artery. Dia akan sangkut dekat sini lah. So when it is totally blocked, it will blockage and destroy the cardiac muscle, and this large enough portion of the heart is affected, your heart will be stopped beating. So at this time, normally the people will be pensan lah, akan pensan punya, will be pensan, and then at this time you need to do a CPR or other emergency procedure to secure him to let him the heartbeat is continue. If not, the patient will be die immediately. And then but however, this one is because of fully blockage. But if they are only have the partial blockage, it will cause the chest pain. what we call as the scientific name is we call as angina pectoris. Angina pectoris. So this pain is normally that people feel like they are the laboring under strain or macam sakit lah. So for this kind of patient, if you feel like this, you should alert. You may have the heart attack. Maybe you already have the heart attack at there. Okay, so what are their cause? Okay, so if you learn about all this, what are their cause that can cause the atherosclerosis? So the first cause is about the sodium salt intake. So the people that intake very high amount of salt, normally is sodium lah. So the ion high amount of salt, you will have the higher risk to get the cardiovascular disease. Okay, so normally in the nutrition dietitian, they only suggest that every people only consume 2,000 milligram of salt per day only, meaning 2 gram only. But for nowadays, only 2 gram? No, I think you go to outside eating the cook, they already add out, I think already 10 gram or 20 gram of salt lah into your food already. So so if the patient too much of sodium, it will accumulate in the body. The body will hold onto the water to dilute the sodium. So meaning that when you eat too much of sodium, too much of salt, it will accumulate in your blood vessel. So meaning that your body require many water to dilute the sodium. So when you decrease your both amount of fluid surrounding the cell, your volume of the blood will be also increased. So when you sepatutnya your blood volume, your blood vessel only can maintain certain part only, can maintain about the 100 cm3 of fluid. But for now, because you intake more sodium, maybe the blood volume is higher than 100 cm3 lah. So increased blood volume meaning that more work is required. Karena dah banyak kerja lah, banyak air kan, so memang awak kena banyak. You need your heart pressure and your heartbeat is required to increase to pump all this blood. So over time, the extra work and the pressure can stiffen the blood vessel, leading to high blood pressure, heart attack, and also stroke. Okay, next is about the lipid food. So normally the people who eat LDL cholesterol yang paling banyak, normally ayam goreng lah, fried food, french fries, or you go to eat junk food, all these, you will increase your LDL cholesterol intake, meaning that it will increase the rate of atherosclerosis lah. So I think I don't need to explain more, because development of atherosclerosis is the key factor is about the when you consume more LDL cholesterol. Another one, smoking. Smoking is also can cause the cardiovascular disease. So normally, smoke. If you go that cigarette contain nicotine, right? Nicotine can constrict our blood vessel. And then nicotine, when you smoking, you will release carbon monoxide, right? So I think carbon monoxide can damage the inner lining of blood vessel, and also carbon monoxide as an inhibitor in ETC, right, that you learn in semester 1. So normally, if carbon monoxide already block the ETC, meaning that your less oxygen is carried by the blood, and then less oxygen will available for use in the heart, muscle, and throughout the body. Okay, so this is a smoking. Another one, obesity. So the patient will become more obese, which the BMI is higher than more than 30. You need to alert lah. So normally, you were associated with elevated blood pressure, because obese meaning that your blood pressure is very high, your blood lipid also very high, your blood glucose also very high. So this one already change your weight. Consider we change in this risk factor for this disease. So normally for the obese, they will have the higher amount of adipose tissue, right? So adipose tissue, like a major endocrine organ, will producing numerous bioactive peptide called adipokine. So normally this adipocyte create the imbalance different physiological function which are ultimately linked with cardiovascular disease. And then obesity will also accelerate the atherosclerosis process development and then not only lead to increased fat, typically disease, but also insignificant lipid deposit within the other organ. Okay. Another one, last one is stress. Orang yang sangat stress atau panic is also have the high risk to get the cardiovascular disease, because stress hormone or the people is in the stress condition, it will release the hormone called cortisol, or it's a one type of stress hormone. This hormone will respond to hydrolyzed glycogen, because if you are in the stress, meaning that you need to have a higher amount of blood glucose. Your cell is required to have intake high amount of glucose. So higher level of cortisol in long term can increase blood cholesterol and your lipid component, blood sugar, and also blood pressure. So if all this is happening, you will increase rate of atherosclerosis. And then other hormone like adrenaline and noradrenaline also can contribute to stress. And the high response with this hormone will change the promote to build up the blood deposit in the article. So although this hormone is good, the primary function is good, but if the people is in the secreted these two kind of hormone in very high concentration every day, then atherosclerosis will be happening very frequently lah. Okay, another one is about the amygdala. Amygdala is also another type of hormone initiating the stress response. So it will increase the white blood cell production in bone marrow, which contribute to in the inflammation of the artery. So increase in bone marrow activity and artery inflammation will increase the cardiovascular disease like angina, chest pain, heart attack, and also stroke. And then about the prevention of cardiovascular disease, I think I don't need to explain much. Exercising, bersenam, activity, exercise is the best prevention that you do in prevent the cardiovascular disease. Okay, so I think this is the advantages of the exercising. Not only reduce the cardiovascular disease, it also protect your against atherosclerosis, decrease stress, improve sleeping, limit obesity, counter diabetes, delay aging, improve circulation, improve lipid profile. All this this is the advantages. And then this is about the intensity of exercising. What kind of exercise you need to do. So when you do low intensity exercise, every week 4 times, and then each time you do 90 minutes, you will have this effect 250 kilocalorie. High intensity, and then differences between running and walking. So you see here, if running, your mortality rate will become higher compared to walking. And then another way is about the healthy and balanced diet lah. You need to maintain not only exercising, you need to maintain the balanced and healthy and balanced diet according to food pyramid lah. Okay, so that's all for cardiovascular disease. Next, I go to the lymphatic system. Okay, lymphatic system. So we know that lymphatic system is one type of transport system that relate to the blood circulatory system, and also lymphatic system play the role in the immune response too. So immune response, and then also lymphatic system play the role in adsorption. So but in this chapter we only focus about the collect and return interstitial blood to the blood collect and return interstitial blood to the blood only. The relationship between the interstitial fluid and also blood formation, blood only. The immune response you will learn maybe tomorrow. I think tomorrow I will finalize it, chapter 12, right? And this one is not in our syllabus, but I think this one you already learn in SPM lah. Okay, so about the interstitial fluid formation. So you see this diagram. So this one, the red color one is our arteriole, and then the middle one, purple color, is blood capillary, and then this one is our venule, and then this one is our lymphatic vessel. Lymphatic vessel. So we know that interstitial fluid formation is due to the high hydrostatic pressure and the low osmotic pressure between the arteriole. So the high hydrostatic pressure will push out the component from the blood into the interstitial space, and then to form the interstitial fluid. So you see here the exchange substance between the wall and take place across the thin endothelial wall. And then small molecule normally is oxygen gas, the gases, and then some of the plasma protein will be plasma protein, and then nutrient, and also some of the leukocyte white blood cell will be pushed out, will be forced out from the arteriole into the interstitial space, So this is what we call as interstitial fluid lah. So normally interstitial fluid formation, we will not only focus on hydrostatic pressure, we also need to focus on osmotic pressure. So these are the two relationship. So hydrostatic pressure, I think I don't need to mention. Hydrostatic pressure is the water pressure that already exerted. Hydrostatic pressure is a pressure exerted on the wall of the blood vessel, and then osmotic pressure is the pressure produced by the difference in isolated across plasma membrane, solute concentration across plasma membrane. So actually the movement of the fluid is due to this both pressure. Not only hydrostatic pressure, but osmotic pressure also play the role. So normally the high hydrostatic pressure and the low osmotic pressure in the arterial part, it will push out all the component like protein, plasma protein, nutrient, gases, and also some leukocyte from the blood capillary into the interstitial space to form the interstitial fluid. But at the end of the venule end, the hydrostatic pressure is now decreased, and osmotic pressure is quite high, so that the adsorption will be occur, so that the component inside the interstitial fluid, like water molecule, carbon dioxide, will be absorbed by the blood capillary. Okay, so this one is another one. So it arterial end, high hydrostatic pressure, and also osmotic pressure. So in the arterial end, the hydrostatic pressure is higher than the osmotic pressure lah. But at the middle part, in the simple diffusion, the hydrostatic pressure is equilibrium to the osmotic pressure. But at the end of venule, the osmotic pressure is higher than hydrostatic pressure, so that the reabsorption is occur. Okay, so I think this one, filtration and reabsorption. This one is high hydrostatic pressure compared to osmotic pressure, and then this one osmotic pressure is very high, and then this one is hydrostatic pressure and osmotic pressure are equilibrium. And then about the lymphatic system that relate to the blood circulatory system. So normally that one is just mentioned about the interstitial fluid. So normally the interstitial fluid later will be diffused into the lymphatic vessel, and then to form the lymph. So normally lymph is watery fluid formed from the interstitial fluid that contain high amount of leukocyte and lipid. And then diffusion fluid will diffuse into the lymph capillary, where it become lymph. Okay, the lymph capillary will become to form longer lymphatic and will go through the lymph node. So as lymph circulate through the lymph node, phagocyte will filter out the bacteria and other harmful material. So this one is about immune response. Then this vessel will join the circulatory system back again through the subclavian vein, and also thoracic duct. So you see, refer to this diagram. So normally the lymph will return back to the blood circulatory system through the subclavian vein and also thoracic duct. Okay, and then another one is about the functionalness in lymphatic system. So normally, if our lymphatic vessel or lymphatic fissure is already unfunctional, or there are blockage at there, so similar to the artery, they will block the lymph vessel, and then it will cause the disease. So normally, if the movement of lymphatic system is disrupted, will result in the fluid accumulation, or we call as edema. So normally this one is caused by parasitic worm. When there are the parasitic worm inside your lymphatic vessel, it will block the movement of lymph, and then this will cause one type of disease we call as elephantiasis. So in elephantiasis, this is marked by extreme swelling in the limb. So awak boleh tengok macam dia ada macam why is say elephantiasis, because it look like elephant. Dia macam bengkak dekat sini lah. But it is not necessary in the leg only, maybe it will also in the hand. You need to see what part of the blockage happen lah. Okay, so next I will proceed to the transport implant. Okay, later Okay, now I already finished transport in mammalian. Now I will proceed to the transport in plant. Okay, when we talk about the transport in plant, we need to make a recap that we learned in semester 1, that is about the vascular tissue. Because vascular tissue is play the main role in the plant transport. So we know that xylem tissue is play the role in transport water and mineral ion, right? I think everyone should know the structure of the xylem, the location of the xylem. So we know that there are 4 types of xylem cell, and then xylem vessel and trachea is play the role in the transportation of water and mineral ion. Okay, so water transport in plant. Okay, this one I need to mention about the adsorption of water from root cell, by root cell. So we know that plant, how the plant take water molecule, they will take the water molecule by root. Okay, so this is the explanation how the root absorb the water. Now the root cell is most important part in plant, because most of the adsorption of water and mineral adhere. And then root cell consists of many root hair, which the plasma membrane is permeable to water to increase the surface area. So if you see here, there are many root hair at here. So this one is we call as root hair. So actually, if you refer back to the real situation, actually there are many root hair at there. So normally the hypertonic region of the root cell will absorb the soil solution, which consists of water and mineral. So normally our root cell is in the hypertonic condition. It is in the hypertonic condition, so that it can absorb, so that the water molecule can be diffused into the root cell. And then the soil solution then is drawn into the hydrophilic wall of the epidermal cell and pass through along the cell wall into the cortex. And then this will enhance the exposure of the cortex cell to the soil solution, and then this provide the greater surface membrane area for adsorption compared to surface or epidermis alone. The active transport will also occur so that mineral is able to move into the cell. So normally this one is just about the adsorption of water by root cell. So the water solution is drawn into the hydrophilic wall of the epidermal cell and pass through along the cell wall into the cortex. Into the cortex. So normally the water will be absorbed, and then it will pass through along the cortex, and then it will enter into the root xylem. Okay. And then this one I need to mention is about there are 3 pathways that the water, once the root already absorb water, there are three pathways that the water and mineral ion can be passed through from the root hair along the cortex into the root xylem vessel. So there are three pathways we call as apoplastic pathway, symplastic pathway, and also vacuolar pathway. Okay, so what are they? So this one is another. Okay, so there are three pathways, right? Symplastic pathway, apoplastic pathway, and also vacuolar pathway. Okay, so what is about symplastic pathway? So symplastic pathway is that the water will transport through the plasmodesmata linked to the cytoplasm from one to another, meaning that it's directly passed through. So you refer to this is symplastic pathway is directly go into the cytoplasm of the plant cell directly, and then when it want to occurs to another cell, it will pass through the plasmodesmata. So it will directly to the cytoplasm, occurs through the cytoplasm. And then another one is vacuolar pathway. So vacuolar pathway is the transport that it pass through the vacuole. Whether the cytoplasm, symplas is through the cytoplasm, but this one is through the vacuole. From the vacuole to another vacuole of the cell, through the cytoplasm and also cell wall. So you see here vacuole, and then it will enter the cytoplasm and then through the cell wall, and then it will enter back to the vacuole again. And then another one is about apoplast. So apoplast is more spatial. It only pass through the cell wall only. Dia jalan guna cell wall. It will not enter the cytoplasm and also vacuole one. It will only transport through the cell wall only. However, apoplast pathway is cannot directly reach into the xylem vessel. Only the symplas and the vacuolar pathway can be directly from the root hair directly go into the root xylem vessel one. But apoplast pathway cannot, because there are the endodermis layer, endodermis cell that consists of Casparian strip at there. So before the root xylem vessel, there are the endodermis layer at here that contain Casparian strip. Okay, so because this Casparian strip is insoluble into the water, impervious into the water, so that the water from the apoplast pathway, from the cell wall, it will be forced move from apoplast to the symplas pathway. It will force out to the cell wall. It will force out from the cell wall into the cytoplasm, and the cytoplasm when reach the endodermis cell. So when it reach here, the water cannot pass through the cell wall. It will force out the water into the cytoplasm. Why? Because of Casparian strip. Because Casparian strip is made up of suberin, which is one type of wax, which is impervious to water. So water cannot pass through it. So memang the water is forced out into the symplas pathway, and then continue pass through it into the xylem vessel. Okay, so normally this is what I just want to mention. Endodermis is the innermost layer of the cell in the cortex that act as the last checkpoint for the selective pressure of the water from cortex into the xylem vessel. And then Casparian strip make the endodermis prevent the accumulation of solute backflow into the soil and keep and toxic substance out of the cell. So this is the function. Okay, so this one is the key important terms about the water movement from root to leaf. So there are three mechanisms you need to learn: root pressure, cohesion tension theory, transpiration pool. Okay, about the root pressure. Okay, root pressure actually is the pushing pressure. Compared to transpiration pool is pulling pressure, right? So root pressure is a pushing pressure, meaning that that water will move into the root from the soil, is pushed out through xylem toward the top of the plant. So it is a pushing force. Normally it will happen during night only, because the transpiration rate now is low. And the transpiration rate Transpiration rate is high during the hot sunny day only, but during night, the transpiration rate is low, so that root pressure will be take over lah. Okay, root pressure will occur because mineral ions that are actively absorbed from the soil are pumped into the xylem, decreasing its water potential. Okay, so if you refer to this again, when the mineral ion, you know that water is diffused through osmosis, but the mineral ion, some of the mineral ion is pumped through active transport. So when the mineral ion is pumped into active transport, through the active transport, it will accumulate, mineral ion will be accumulated here. So when the accumulate here, the solute potential will be increased. So when the solute potential increase, water potential decrease. So it will maintain the hypertonic condition, so that the water molecule can diffuse into the root by osmosis process. So this accumulation has an osmotic effect, causing water move into the xylem cell from the surrounding root cell. Now then, in turn, water will move into the root by osmosis because of the difference in water potential between the soil and root cell. Yeah, and then this accumulation of water in root tissue produce positive pressure that force the water up through the root xylem into the shoot. And then however, root pressure is not strong. Is very weak pressure. So for the plant that are very tall plant, the root pressure is cannot push the water from the root into the leaf one, cannot. Now it only suitable for the shorter plant only. The main reason is about the strongness. The pressure is not very strong. Okay, and then the root pressure also will cause the guttation. So guttation is the phenomenon where the liquid is forced out through the spatial opening leaf called hydatop. So this one is normally, if you in the early morning you go to see the plant, you will see there are the water molecule here, right? Water molecule at the tip of the leaf. So this we will call as guttation, or the happening due to the root pressure. Okay. Another one is about the cohesion tension theory. So this is very important, that normally in exam already come out many times. Okay. So normally cohesion tension theory is using the water properties. We know that water, one type of water property is it can cause adhesion force and cohesion force, right? Hydrogen bonding. Cohesion force is that the hydrogen bonding between the water molecule and the cell wall, and then the cohesion force between the water molecule. Okay, so normally cohesion tension theory is provided by transpiration. So normally transpiration already provide the pulling force, right? Pulling force and the cohesion of water molecule transmit this pull along the entire length of the xylem from shoot to the root. Hence, xylem sac were under negative pressure or tension. Okay. So normally generation or transpiration pool, I use things the diagram are more simple. So normally in the transpiration pool or cohesion tension theory process, it is actually a pooling process. Whether in transpiration the water vapor will diffuse from the moist A space of the leaf to the drier A outside from the stomata. So in transpiration, the water inside here, since the water potential now inside the leaf is high, and then during the hot sunny day, the water potential outside the plant is very low. So that the water will diffuse out from the leaf into the leaf to the atmosphere through the stomata, in the form of water vapor. Okay, so when the water is already removed, so that the water molecule now is starting decrease at the A space, now is decrease now. decrease now. So you see here at the first, the water vapor lost by transpiration is replaced by the evaporation from the water film that called mesophyll cell. Mesophyll cell. Now you will see that actually there are many water molecules, between the mesophyll cell one. But when the water molecule is evaporated, the water will be replaced by evaporation from water film that caught by mesophyll cell. And then the evaporation of the water film cause the air-water interface to retreat farther into the cell wall and become more curved. This more curvature will increase the surface tension and rate of transpiration. So in normal situation, the water is evaporated, and then this water vapor loss of water vapor will be replaced by evaporation from the water film. So in the normal situation, there are water vapor at here. So when this water molecule is already evaporated throughout the stomata, the water film will be evaporate to replace the water vapor at the air space. Okay, so once the water is starting to evaporate more and more, the water film now will become curved. Will become more curved. The curve will be occur. So this curvature will increase the surface tension of the water molecule and the rate of transpiration process. Okay, so when the surface tension of the water is increasing, it will pull the water from the xylem vessel into the mesophyll cell to replace the water molecule. Replace a molecule. The increased surface tension shown in step 3 pull water from surrounding cell and air space, and then water from xylem is pulled into the surrounding cell and air space to replace the water that was lost. So now in the mesophyll cell, the water molecule at here now is already decreased, so that the surface tension of the water is increased. So the water molecule at the mesophyll cell now is required to replace karena water dah tak ada, tapi dia kena maintain water molecule at there, water. So meaning that it will take the water from other part lah. So normally it will pull the water, dia akan ambil water daripada mana-mana tempat lah, at any places to replace this water film. So finally your xylem water in the leaf xylem will pull the water molecule and then replace the water inside the water film lah, in the mesophyll cell. Okay, so this one is about the generation and transpiration pool. And then when the generation and transpiration pool is occur, it will generate when the transpiration pool is occur, it will generate the cohesion tension theory, meaning that the water molecule from the root will be pulled up based on the property of the water called cohesion force and adhesion force, that water molecule can form the strong hydrogen bonding. So if you see this diagram, right, the water vapor already transpire from the surface leaves of mesophyll cell and lead to the surface tension to pull the water of the leaf xylem. So when it starting to pull, it was starting to pull up the water. The leaf xylem was starting to pull up the water from the root starting pull up the water from the root into the xylem vessel. So the cohesion of water molecule caused by hydrogen bonding allow an unbroken water column. So that why the water molecule can be pulled up, because of the cohesion and adhesion force. Because cohesion molecule make the water molecule is unbroken, is unbreak. So that the water molecule can be line up one by one, line up together due to the hydrogen bonding, and then it will will be pulled up into the leaf vessel lah. In turn pull outer up root xylem, forming a continuous column of water from xylem vessel, root xylem to stem xylem, and then to leaf xylem. So movement of water upward in root produce a pool that cause soil water to diffuse. So at the same time, the soil water also will be pulled up. So it's a entire pooling process lah. Because of this transpiration process. If this transpiration process is not occur, then no pooling process at all. But due to the presence of this transpiration process, so the pooling process is pulling entire up from the root up from the leaf vessel. Okay, so this is about the explanation. So yeah, so in this one is you need to write in exam lah. Okay, exam. Okay, so I think I already explained all. The curvature increase surface tension, thus the negative water pressure induce curvature of self-tension. Okay, yeah. Okay, more negative water potential cause water, yeah, because more negative water potential cause water be pulled into mesophyll cell. The water from the xylem then is pulled into the surrounding cell expect to replace the water loss. So negative water potential in the leaf will provide the transpiration pool. Yeah, because now our water is already all are removed, so the water potential in the water molecule will be decrease. So this why will cause the pooling process, or will provide the transpiration pool lah. Okay. And then coercion aeration will facilitate the transport of water by blood flow, because coercion water is an attractive force between the water molecule, and then will make the xylem start to be pulled from the root along the xylem vessel without the separation of the water. This is a key point. Because coercion property, it forms the strong hydrogen bonding between the water molecules, so the water can be pulled up without the separation. Dia tak akan break down punya. And then aeration molecule also form because between the water and hydrophilic wall. And aeration help to mitigate the downward force of gravity, ensure that water can be pulled out accurately from root to the leaf. Okay, so the summary of the coercion and tension theory lah. So the first tip is about the water loss by transpiration. So when your water potential of your mesophyll cell is decreased, your hydrostatic pressure also decrease lah. And then this will create the surface tension from the leaf until root, so that it will create the pooling force, transpiration pool. So that the water potential in the root xylem decrease, water from soil diffuse into the root xylem with the aid of coercion aeration force. Okay, next is about the translocation. So firstly we about the transpiration. So now we go to the translocation. It is about the transport of nutrient using phloem tissue. So I think this is just a recap about the phloem tissue. Okay, so I think everyone know there are 4 types of phloem cells: sieve tube, component cell, fiber, and parenchyma cell, right? And then sieve tube and component cell is play the role in the transportation of the organic nutrient. Okay, so translocation actually is a transport of the product of photosynthesis from sugar source to sugar sink by phloem tissue. Yeah, so from sugar source is the leaf, normally is in the leaf meaning that is the site of the production of produced sugar, and then sugar sink is the place that we need sugar. Okay, so this is the translocation. So one of the translocation hypotheses that we need to know is about the pressure flow hypothesis. Okay, so you need to know what is about the sugar source and sugar sink, right? So sugar source is the place that the photosynthesis occur and the organic solute are synthesized, normally sucrose molecule, right? So photosynthesis is only occur in green leaf, right? Green leaf. And then sink area is that the organic solute is unloaded from the sieve tube to the outer line or be stored. So normally this part is at the shoot, root or fruit lah, any part of the plant except the leaf. Okay, so normally sucrose, when the sucrose is loading into the sink from the sucrose from the source. So how the sucrose is loaded into the sink first, into the sink from the source. Okay. Okay, so normally this one is about the loading of sucrose into the phloem tissue. So you need to learn about how the phloem phloem tissue absorb the sucrose from the source. So there are the process. So you see here, this is a part of the cell wall, mesophyll cell that undergo photosynthesis, and then when the photosynthesis, they will produce the sucrose molecule. Okay, so when the sucrose molecule is produced, it will be transported same like the water molecule. It will complete either apopass pathway or sympass pathway, or apopass pathway or sympass pathway. So normally it will pass through the sympass pathway, and then it will pass through the sympass pathway that is across the cytoplasm, and then directly go to the sieve tube element, or go to the apopass pathway, which go to the cell wall according to the cell wall. Okay. So normally the first loaded by sympathetic pathway, then some of the sucrose will exit sympass near and travel through apoplastic pathway when the sucrose is loaded near the sieve tube. So actually normally in plant, they will at the beginning they will go through the sympass pathway first, but when they are near the sieve tube element, some of the sucrose molecule will be transported through the apopass pathway. Some, not all, some. Okay. So while the other will be loaded through sympathetic pathway, the sucrose travel in apopass will be accumulated nearby sieve tube or directly through the companion cell. In most sugar movement into phloem require active transport. So E actually is require active transport process. It does not like the water. Water is simple diffusion process, right? Osmosis process. But in the phloem, it require transport process. Called transport process. So that you learn in semester 1. So I think I don't need to focus at as much as this. You should know already. Okay, so this is the key point about the pressure flow hypothesis. So just now we learned about the how the sucrose is transported into the sieve tube. Okay, so now we will learn about the how the sucrose is transported into the sieve tube and then unloaded into the sink style. So normally this is the process that loading of the sugar into the sieve tube, when there are the many sucrose molecule at here, this sucrose molecule accumulated at here, it will decrease the water potential at there. The water potential will be decreased, so that the xylem vessel, we know that xylem vessel and xylem vessel and sieve tube are located opposite to each other, so that the xylem vessel was starting to diffuse the water molecule from xylem vessel into the sieve tube element. Okay, so when the water potential when the water molecule is accumulated at here, the uptake will generate a positive pressure that force the sap flow along the tube. So when the water potential now is increasing, so it will directly go down, create a positive pressure, bring down the sucrose molecule from the source cell into the sink cell. So when the sucrose molecule is already reached at here, the sucrose molecule will be diffused into the sink cell. So when the sucrose is already diffused into the sink cell, the water potential will be diffused back into the xylem vessel. So it will become a repeat process. So you see here, this grade also same. So the water diffused from the xylem as a result of decreased water potential in the sieve tube, more negative water penetration in the sieve tube, and then the increase in pressure force the fluid through the sieve tube element, and then sucrose is actively and passively unloaded into the sink cell, and then once the sucrose is diffused into the sink cell, the water potential now is increasing, so the water potential will be diffused back into the xylem vessel, and then the process is repeat again. So this is what we call as pressure flow hypothesis. Okay, so this is about the explanation. This cause high hydrostatic pressure in the xylem vessel diffuse out the water, because at this point the water potential is very low, so that it requires water molecule from the xylem vessel diffuse into the sieve tube element. At the sink, sugar is unloaded by various mechanisms, both either active or passive, from the sieve tube elements. With the loss of sugar, water potential in the sieve tube element at the sink increase. So that the water will be diffused back into the xylem vessel again. So another name of pressure flow hypothesis is about the mass flow hypothesis, we created by another scientist. So this is the experimental experiment. Actually same as the concept that we use it here, but this one is just that they prove this kind of hypothesis based on the experiment. So normally A and B, you will see here, A is the flow of the solution. A is the sources, leaf lah, and then B is the sink. But actually it can be reversible. Some of the books say that this part is sources, some of the books says that this part is sources. But maybe MPM pun boleh tukar-tukar lah, dia boleh tukar-tukar punya. So you need to alert for that lah. So actually it's same. So normally you see here when E is the sources, meaning that the sucrose concentration is very high, so that the water from the xylem vessel will be diffused into the A, and then this water will provide the positive pressure, and then bring up the sucrose solution through the sieve tube element. So this one is we call as sieve tube element, and then into the sink place. Okay, once the sucrose enter the sink place, so the sucrose will be diffused out or actively transport out from the sieve tube element into the sink in sink cell, and then the water will be diffused out from the sieve tube element, and then recycle back into the xylem vessel. So you see here, this one is sources, and then this one is sieve tube element, this one is sink, and then this part, the container, is we call as xylem vessel. So this is also very important. Normally come out in objective question, they will come out want you to calculate, not calculate lah, they will give many statement, and then you need to know lah. Okay, except from the pressure flow hypothesis, we also have another 3 hypotheses we need to know: electro-osmosis, and then another one is electro-osmosis cytoplasmic swimming hypothesis, and another one is about the peristalsis wave. Okay, so what are they? So normally this one maybe your school teacher will not teach you. Okay. So about the electro-osmosis, so if you see here, actually this one is another type of translocation process. So actually it is a movement that involves electrical charge, because it already mentioned electro. So osmosis meaning it will involve electrical charge and also water molecule. So normally it involves movement of ion in an electrical field through the fixed porous which is electrically charged by carrying water and potassium ion. Okay, so if you see here this diagram, so when there are the solute inside in the sieve tube element, so same as here, when there are the solid sieve tube sucrose molecule at the sieve tube element, so they will not follow the pressure flow hypothesis. What they will follow is potassium ion, and the water molecule will be diffused back into the it will bring up the solid molecule through the pores, fixed pores. You will know that between the sieve tube element, you will still remember the structure of sieve tube. Between the sieve tube element, there are many pores, right? So it will use the electrical charge. So normally this one is positively charged. Positively charged, they will accumulate with higher amount of potassium ion, so that it will directly, so it will pass through these pores, and then the potassium ion pass through, and then hydrogen molecule also will pass through it. So it will based on the electrical charge. And then once the potassium ion is already at here, so because it need to maintain the negatively charged at the bottom part, so the potassium ion will be diffused back to the companion cell, and then diffuse back again to the upper part of the sieve tube element, and then this process is repeat again. So this is what we call as based on the electrical charge and also water molecule. So the water molecule also will come from the xylem vessel, just that it does not create the pressure, it will create the positively charged ion, so the potassium ion play the role lah. So potassium ion, so at the bottom part, the potassium ion is accumulated high because you will need to maintain positively charged, and then it will create the electrical charge, and then positively charge will come out, go to the negatively charged, and then at the same time the solid will be passed through these pores. Okay, so this is about the explanation. So normally sieve plate and the flume protein are normally in negatively charged, thus forming a porous surface with an electrical charge. So I already mentioned, right, at here is negatively charged, and then at here is positively charged. As mass flow occurs through the negatively charged sieve plate, an ion will repel, but cation will be able to pass through. So when mass flow occurs downward through the flow, the repulsed ion will accumulate above the sieve plate, so that the cell above the sieve plate can become negative. So the sieve plate now will be fixed a porous surface within an electrical field such as is needed for electroosmosis to occur. So when the critical potential difference across the sieve plate is reached, hydrogen ion will lowering the pH and making the cell above the sieve plate is positively charged. So as mass flow occur, negatively charged sieve plate and ion will be repelled, but cation will be able to pass through. And then when mass flow occur downward through the flume, the repulsed and ion will accumulate above the sieve plate, so that the cell above the sieve plate will become negatively charged. So at the beginning, the charge at here is negatively charged first. Okay, so the fixed sieve plate will now at the fixed porous surface within an electrical charge that is needed for electroosmosis to occur. So when a critical potential difference across the sieve plate is reached, so there will have a bottom pump here from the wall of upper cell into its cytoplasm by lowering its pH, making the cytoplasm above the sieve tube to be positively charged. So that is why it happening positively charged at here. And then the increased positivity charge generated by the hydrogen ion push other cation, mainly potassium ion, by electrically repulsion through the sieve plate from the upper to the lower cell, and therefore electroosmosis occur. So the potassium ion are secreted to the other side of the sieve plate. The presence of potassium ion on the side of the sieve plate induces negatively charge on the other side of the sieve, creating a potential difference across the sieve plate. So this such hydrated potassium ion will carry water molecule and dissolve solute like sucrose across the sieve plate. So you will still remember, the pressure of potassium ion on the other side of the sieve plate will induce negatively charge of the other side of the sieve plate. So they can create the potential difference at here lah, so that the potassium ion will be repelled, will be passed through the sieve plate, and then bring up the solute potential through the electroosmosis process. Okay, another one is about cytoplasmic swimming hypothesis. Okay, so actually this one is that the molecule move at the fast is that the molecule and the water differentiation relationship between the molecule and the water. So normally this one, cytoplasm of plant cell is also to move around the cell, we often call as streaming. So it has been proposed that solute might be carried from one end of a sieve tube element to other by swimming and then transfer across sieve tube by active transport. So normally both swimming and transfer it through the sieve tube would be energy dependent, especially high turnover of ATP in phloem cell. So different solute move through the sieve pore at different rate due to their different molecular characteristic and different level of impermeability of sieve plate to different solute. So normally this one is recalled as swimming. So you need to know about the swimming is that the plant cell is ordered to move around within the cell, meaning that the molecule at here is free to move as well. So molecule normally at the bottom part is moved at a very fast speed, and then when it reaches the sieve tube sieve plate, molecule will move at a very low speed, and then when the molecule will first out of the cycle, and then molecule will join next into the next cycle. So dia akan macam flow dia based on the active transport, macam keluar lepas tu penetrate like that. Okay. And then last one is about the perithallis valve. Okay, so perithallis valve actually is involved only transcellular strand contain critical protein which are present in some sieve tube. So sieve tube actually is filled with 5 cytoplasmic filament continuous from sieve tube to the next through the pore of the sieve plate. Okay, so it containing phloem subtube constrict and relax, alternatively pushing the phloem sub from one sieve tube to the next. So actually this one is using the contraction relaxation of the cytoplasmic filament. It will not involve in the phloem sieve tube element, but it will involve in the continuous sieve tube cytoplasmic filament. So that the continuous filament, so at the point when it contract they akan push the molecule and then at that point it will contract again, it will push the molecule, and then it will contract again, push the molecule. Dia macam ni lah, using the contraction and relaxation process one. And then this one is only occur in the cytoplasmic filament. It is not occur in the sieve tube element one. It only occur in the cytoplasmic filament. Okay, so that's all for chapter 8. Okay, so that's all for chapter 8. So hopefully you guys can understand, take the time to digest it, what you will learn in chapter 8. Okay, so now it is already 12 o'clock. Okay, so I will give a break, lunchtime, for now, until we will continue the chapter 3 at 2:30. So we will see you on 2:30 lah. Okay, so take a lunch and have a break, and then see you guys on 2:30. Thank you and bye. Hello. Okay. I think my sound is now is okay. So okay. Okay. So now is already 2:30. So we will start our continue our revision. Okay. So for now I will proceed to chapter 9, control and regulation. So for this chapter we have two type of topic, right? Nervous system and also hormone. So for nervous system we only learn about the human because only human contain the nervous system, and then for the hormone part we will separate into two, right? The mechanism of steroid and non-steroid hormone, and also the plant hormone. Okay. So for today, so for first, we will learn about the nervous system first. Okay. So for the nervous system, the key point that we need to learn is about the neuron tissue that you already learned in semester 1. So this is just a recap. So I think we all of us know that we have three types of neuron tissue, right? Afferent neuron, interneuron, and also efferent neuron, right? So this is one of the example of efferent neuron. Okay. So we know that the neuron is the functional unit for the nervous system, and then so we know that I repeat again. We know that we have three types of neuron, right? Afferent neuron, interneuron, and also efferent neuron. And then we know that the neuron is the basic unit for the unit cell for the nervous system. And then we know that inside the neuron we have the cell body, and then a highly biased attention called dendrite, and then dendrite receive the signal from the other neuron. So normally you will see the afferent neuron. So all of this is about the dendrite that they receive the signal, and then you will see the interneuron. The part that receives signal is we call as dendrite, and then the part that transfer the signal we will call as axon. So you will see the efferent neuron. That is why that the efferent neuron, the axon is more longer compared to dendrite. But for the afferent neuron, the dendrite will be more longer compared to the axon. And the dendrite will be reached to the anterior cell body. After the cell body, it will become the axon. So if you see here, for efferent neuron, the cell body is at the beginning of the site. So the dendrite is more shorter. So after the cell body is will become the axon that transfer the signal, the nerve impulse to the effector. Okay, so normally neuron also contain a single axon and extension that transmit signal to other cell. Okay, so normally the axon are more longer than dendrite, and then will have a cone shape based on axon called axon hillock, and then near to the end the axon will be divided into many branch, and each branch end will transmit information to another cell junction called synapse. Okay, and then in some of the neuron, they will contain the myelin sheet. So if you see this picture, you will see here there are the cover at the axon, right? At the axon structure. This one is we call as myelin sheet structure. So why it contain myelin sheet structure? Because it is made up of one cell. So if the axon that are covered by one cell, it will form the myelin sheet structure. So why it is very important? The member of myelin sheet is mostly lipid, and it is a poor conductor for electric current, resulting in a good insulator. So normally in this structure, it will provide the fast conduction of vertebral axon. and then there are the gap between the myelin sheath is called a knot of Van. So you see here, there are the gap between the myelin sheath we call as knot of Van. So this is the point that the electrical impulse will be occur only. So meaning that the electrical impulse will occur, and then it will jump toward the knot of Van, and then it will jump toward the knot of at the cell that have the myelin sheath structure. The nerve impulse will not be carried out. So the nerve impulse will be jump from one point to one point, jump from one point to one point until it reach the effector lah. So it will enable the fast response compared to the non-myelinated structure. Okay, so normally the extracellular fluid will only contact with the axon membrane at the knot. So thus the action potential will be generated at the knot only. And then in myelinated axon, the liberalizing current or during action potential at the one of the knot of the nerve along the interval to axon to the next code. So on the other word, action potential will appear jump from one node to one node as it travel along the axon. So this one later you will learn about it. I think you guys also know the transmission along the action potential transmission of nerve impulse along the axon. Okay, and then in nerve impulse in neuron, not only contain neuron cell, we also have the cell that we call as glia cell. So normally glia cell actually is act as a support to the neuron tissue that play the role in insulating the axon of the neuron and regulate the extracellular fluid from the surrounding neuron. Okay, so if you see this diagram, all this one is we call as neuron. Neuron, this is our neuron, and then there are many types of glia cell like ependymal cell, astrocyte, oligodendrocyte, microglia, and also segment cell. All of that is one type of one of the glia cell. So you can read yourself. Next we go to the human nervous system. I think I already closed the music, right? Okay, now we go to the human nervous system. Human nervous system. So we know that in human nervous system we have two main category, CNS and also PNS, central nervous system and also peripheral nervous system. So CNS normally is in the brain. The nervous system that we control in our brain, that located in the brain that control all anything we will consider as CNS. And then you all need that a brain, it continues with the dorsal tubular spinal cord that's serving as central control. This organ will integrate incoming information and determine appropriate response. And another system we call as peripheral nervous system. So it is actually made up of the sensory receptor and the nerve, which are the communication line. So peripheral nervous system is the communication line between the effector and also the central nervous system. So you see this diagram, the central nervous system is including the brain and also the spinal cord that located in the vertebra bone. And that peripheral nervous system is the communication line that join with the CNS, and that it will spread along all the part of the body lah. So in PNS, there are two types of PMS that you need to learn, which is somatic division and also autonomic division. So if you see autonomic division, so if you see the summary signal of pathway. So normally when our sensory receptor, any part of sensory receptor in our skin or any part is stimulated, then will send the impulse through the sensory neuron, and this signal will be sent to the CNS sent to the CNS. So when the CNS is receive the signal from the sensory receptor, it will release the the CNS was starting to integrate and modify the information that it received, and then it will send the signal to the motor neuron to transmit to the muscle, and then it will cause the muscle to be contract. So if you see this diagram, so normally when the receptor and our sensory neuron, afferent neuron is stimulated, information will be transmitted to the CNS. Okay, so once the CNS receive the information, it will starting to integrate the information, and then it will transfer the information to the efferent neuron. So either the efferent neuron inhibit the signal, or it will stimulate the signal. Either it inhibit or stimulate up to the signal that received from the sensory receptor. Okay, so if you see here, okay, so PNS is transmit the information to and from the CNS, play a large role in regulating an animal movement and its internal environment. So you see here, when the afferent neuron is stimulated, it will send the impulse to CNS, and then it will send impulse to efferent neuron, which is the peripheral nervous system. So sensory information reach the CNS along PNS neuron designated as afferent. So following information processing within the CNS instruction then travel to effector along PNS neuron designated as efferent. So all the information after the CNS is processing, and then it the nerve impulse is transferred to the efferent, is known as the efferent neuron. Okay, so the neuron of the motor system then can carry the signal to skeletal muscle for voluntary action, but some of that is involuntary action for the autonomic nervous system. So for if the signal is carried to the motor system, meaning that it is for the skeletal muscle, which of voluntary action, but some of them will go to the autonomic nervous system, which is the involuntary action. So under the autonomic system, we will separate into two: sympathetic nervous system and also parasympathetic nervous system, right? Okay. So next, we will go through the differences, comparison between sympathetic and parasympathetic nervous system. Okay, so this is the autonomic nervous system under sympathetic and parasympathetic. So if you see the two main difference is the similarity of these both nervous system are they contain preganglionic neuron and also postganglionic neuron, and then at the middle part it contains the ganglia structure. So you see here, preganglionic neuron is the myelin structure, but the postganglionic neuron is non-myelinic structure. And then you see the differences between sympathetic and parasympathetic nervous system is that preganglionic for sympathetic is more shorter compared to the postganglionic, but for parasympathetic, preganglionic is more longer than postganglionic. And another difference is the acetylcholine, the neurotransmitter release. So for the sympathetic, it will release noradrenaline, but parasympathetic will release acetylcholine. So the function of the noradrenaline and acetylcholine I already mentioned in last chapter, right? Chapter 8, I already mentioned it, which they can also regulate the heartbeat, right? But not only that, actually they are the parasympathetic and sympathetic will control many part of our organ, not only our heart. Okay, so for sympathetic, normally is to stimulate something, it will increase something happen. So normally you will see here, it will accelerate the heartbeat rate, right? And then it will dilate pupil of eye, inhibit salivary secretion, and also stimulate glucose release, stimulate adrenal medulla, and also promote the education. But for the parasympathetic, normally is inhibition process, will compete to inhibition process. So this is the differences between the parasympathetic and sympathetic that you need to know. Okay, next we go to the resting potential formation. Okay, so in neuron that transmit the nerve impulse, we will have two types of potential, right? Either it is in resting state or it is in action state. So when we want to maintain the resting potential, how it is be maintained? So it is a resting neuron that is not sending a signal. Okay, so we know that what is the definition of membrane potential? It is the differences in the concentration between specific ion inside the cell and those in the extracellular fluid. So this is the definition of the membrane potential lah. Membrane potential is the differences that when the specific ion inside the cell and outside the cell have the difference, it will create the membrane potential. So normally in the neuron, the two key ion is about the sodium ion and also potassium ion. So in the resting state, inside of the cell will be contain the potassium ion, and then the outside of the cell is accumulate with the sodium ion. So normally in the resting state, your potassium ion is greater inside than the cell. So more potassium ion is accumulated inside the cell, but the sodium ion is accumulated outside the cell. So this asymmetric distribution of ion across the plasma membrane of vase is brought about by the action of selective ion channel and ion path. So normally in resting potential, the concentration gradient are maintained by the sodium potassium pump. So that is why that in sodium potassium pump, sodium ion will be 3 sodium ion will be pumped out from the cell, and out from the cell, and the 2 potassium ion will pump into the cell. So it need to balance the charge. So it need to have a differences between charge between the outside of the cell and also inside the cell. Okay, so not only the sodium potassium pump, there are also the ion channel. So normally there are 2 types of ion channel, either a potassium channel or sodium channel. So normally this ion channel is to make the ion diffuse across the membrane in and out according to the facilitated diffusion down the concentration gradient. So normally, and as ion diffuse through the channel, the electrical charge is produced, and that ion can diffuse rapidly through the ion channel, causing a net movement charge near membrane potential. And then if there are 2 types of ion channel, right? So potassium is only special for potassium ion to pass through only, while sodium ion is only special for the sodium ion to pass through only. So if I mentioned before, in resting state, potassium ion is more accumulated at inside the cell, right? And then the sodium ion will more accumulated outside the cell. And then the potassium ion channel always open, causing potassium ion diffuse out from the cell. So due to the high concentration of potassium is high inside the cell, so the net outflow of potassium ion, causing the net outflow of potassium ion. So the outflow of potassium ion will lead to the negatively charged inside of the cell. So furthermore, only a few sodium ion or channel open due to sodium ion cannot readily across the membrane. At the same time, the voltage-gated sodium and potassium ion channel are closed. So the membrane is said to be polarized due to the inside of the is more negative. So as mentioned before, the potassium ion is high inside the cell. So that there are many potassium ion channel, so that the potassium ion will be diffused out from the cell, so that the inside the cell now is more negatively charged, and then the sodium ion channel is more little compared to potassium ion, so that it need to maintain the negatively charged inside the cell. And then at the same time, the voltage-gated sodium and potassium ion channel are closed at the resting potential. So this is a voltage-gated ion channel. So the difference is that it can be closed. The gate is like a gate, it can be closed or open. Like this one is totally open one. The ion can be passed through any time based on the concentration gradient. But for this one, when the ion channel is closed, the gate is closed, then no ion can be passed through. Okay, so this is about the resting potential. Now I will go to the action potential. Okay, so action potential is that there is the electrical signal or the nerve impulse that traveling rapidly down the axon into the synaptic terminal. So when there are the electrical impulse or nerve impulse is passed through, then action potential will be generated. Okay, so how the action potential is generated? So this one is very important graph lah. So we know that there are 5 main phase, right? So the first phase is about resting potential, and then once the electrical impulse is starting to stimulate, when there are the electrical impulse, depolarization was starting to induce, and then once the depolarization is exceed the threshold level, rising phase is starting. So action potential is generated. So once after the action potential, the repolarization is occur. Okay, after repolarization, after that undershoot will be occur. Okay, so we will see it one by one. So in resting state, I don't need to say more. So normally the gated sodium ion channel and the potassium ion channel are totally closed. Okay, close. And then after that we will go to the depolarization. So starting the depolarization, the electrical impulse is starting to be generated. When they receive the axon was starting to receive the electrical impulse, some of the sodium gated ion channel was starting to open. So you see here, this yellow color one is the sodium ion gated channel that already starting to open, but not all. So that the sodium ion was starting flow from the outside into the cell. So that if the stimulant is very strong, normally is exceed to the threshold level, it will trigger the action potential. Okay, so once they already trigger the action potential, reach the threshold level, the further depolarization will be occur, causing more sodium ion channel will be open, and then the sodium ion will start to influx into the cell, making the cell more positive. So now the cell inside the cell is more positive, and then the outside cell is negative already, and then the gated potassium channel is remain closed, and then after they already generate action potential, it was starting to repolarize. So repolarize meaning that the sodium gated ion channel now is inactivated and starting to close already. So now the sodium ion is already blocked. The sodium ion outside the cell is blocked and prevent the entry of the sodium ion into the cell So at the same time, your potassium gated ion channel starting to open now. So when it's starting to open, the potassium ion inside the cell was starting to release out. So that your cell will become more negatively charged. Because the potassium ion now is starting to release out already, and then the sodium ion is cannot enter into the cell. Okay, once all the potassium ion is released out, and the final phase is recalled as undershoot. Okay, so the membrane permeability toward the potassium ion is higher than the resting phase. So meaning that at this time, all your membrane of the axon will have the higher permeability to potassium ion. So meaning that all of the many potassium ion were starting to influx out from the cell to maintain the negatively charge inside of the cell. Thus, the membrane potential now is lower than the resting potential. So meaning that there are more potassium ion with influx out from the cell compared to resting potential, because resting potential is just to maintain the equilibrium only. But for the undershoot, many potassium ion will be influx out from the cell. So this reaction is recalled as hyperpolarization. So after that, your potassium gated ion channel will be closed, and then no more movement of potassium ion. So now it will reach to the resting state. So it will repeat the process again and again. It will repeat the process again and again. So once after the undershoot, it will go to the resting state, and then when it receive the signal again, it will repeat the process again and again. And then we need to next we learn about the nerve impulse characteristic. So nerve impulse have their own characteristic. The first characteristic is about the refractory period. Okay, refractory period, what is it? So normally, if you know that the sodium ion channel will remain inactivated during the falling phase and the early part of the undershoot. So meaning that during the repolarization phase and also undershoot, your sodium ion channel is inactivated. Or inactivated, and then if a second depolarizing stimulus occurs during this period, it will be unable to trigger the action potential. So you see this diagram. Again, when the action potential, when the first stimulus is already reach the threshold level, it will trigger action potential, right? So starting they reach the threshold level, refractory period is achieved. Is occur. So at this period, although if there are another stimulus is reached at the same time, so when the time is like at here, and then at the same time got another stimulus, so that this will prevent the action potential to be produced one more time. The action potential will be produced after they already finish the undershoot hyperpolarization process, and then when it is in resting state, got stimulus, then action potential can be triggered. And then if during the depolarization or repolarization process, action potential will not be triggered again and again. So although there are the stimulus come in, the action potential will not be triggered at this time. So this is what we call as refractory period. So refractory period, we have two types of refractory period, like absolute refractory period and also relative refractory period. So you see here, okay, so no absolute refractory period is that no additional stimulus can initiate for a further action potential. So it is period at here during the depolarization process and also repolarization process, and then during this period, if you add the additional stimulus, there are also no action potential will be triggered at this time. And then another one is about relative refractory period. So if an additional stimulus is to initiate another action potential during this time, it must be larger in order to reach threshold. So you will see here in relative refractory period is under the undershoot process already. So if there are the stimulus at this time is happening, so this stimulus you need to be very strong compared to the stimulus in the resting potential to trigger the action potential. Why? Because at this time your membrane potential is very highly negative compared to the resting state. So if there are the stimulus here, you still can trigger action potential, but then your stimulus need to very strong until it can reach the threshold. If not, then no action potential will be produced. Okay, so the refractory period is very important that it allow the action potential move in one direction only, and also it limit the frequency of action potential. And then another one about the nerve impulse is all or none law. All or none law. So meaning that it only either can generate the action potential or directly no action potential is occur. There are no like the middle one. The action potential cannot like that. Or when there are the stimulus, then maybe it like at the half of the action potential, no like that. So meaning that if the depolarization increase the membrane potential, the further depolarization will occur. If not, if the stimulus is not reach the threshold level, the greater potential is formed. So the greater potential will induce more electrical current that dissipate as it flow along the membrane. Thus, this indicates that the action potential either fully occur or do not occur at all. Either it fully occur or not occur. So they represent as all or none respond to stimulus. Hence, the action potential is not affected by the intensity of stimulus. The site of potential is constant through the intensity stimulus is very high. So meaning that although your stimulus is very high, but your action potential is only have one type of action potential only. Did you not say that when your stimulus is very high, your action potential, your voltage will be very high? No. Only have one only have one. Even though your stimulus is very high, your action potential, like for this one, is only reach 40 watt only, 40 milliwatt only. It will not say that if your stimulus is very high, it will reach to 100 milliwatt like that. No. You only have one kind of action potential only. And then about the speed of conduction, so normally there are two key difference that affect the speed of conduction. Either is the diameter of the axon and also pressure of myelin sheet. So diameter of axon, for the larger the diameter of the axon, your conduction speed of the electrical impulse is more faster more faster. So this is because the smaller the diameter, the greater the resistance of the exoplan, and lower the speed of the axon potential. And then also the pressure of myelin sheet. So this one I already mentioned. If there are the myelin sheet, the nerve impulse will be jump from node to node along the axon. So okay. So now I will explain about the transmission of action potential along axon. So you see this diagram. Okay, so if you see this diagram, you will find that the action potential actually is only occur at the certain region of the axon only. So meaning that if this region is triggered, the action potential is triggered at this region, other part of the region the action potential will not be triggered, and then the nerve impulse will be transmitted from left hand side to the right hand side, left hand side to right hand side. So an observed potential is generated as so that I also now, if like this diagram, the first region of the axon is action potential is triggered. Okay, so that how it will trigger the another region of action potential. So when the depolarization of axon, the depolarization of action potential was spread to the neighboring region of the membrane, reinitiating the action potential there. So to the left of this region, the membrane is repolarizing as potassium ion flow outward. And then the depolarization and repolarization is repeated in the next region of the membrane. So normally, if you see here, when the depolarization is occurred at here, actually it will spread this impulse to the neighboring region. So that this region was starting to trigger the action potential, and then at the same time your previous region was starting to repolarize, meaning that the action potential is already done, completed, and then the potassium ion now is pumped out from the cell. So this process, this depolarization, so that depolarization is triggered, and then this will trigger spread the impulse to the another region again, and then another region again, and then the region at here was starting to repolarize and repeat. This process is repeat again. So normally this is the explanation. So normally the difference in between the active and resting membrane cause the localized current. So localized current play the main role. So the action potential is generated, and then the localized current will cause the depolarization of the beginning location of axon membrane, spread the neighboring part of the membrane. So meaning that the neighboring part of the membrane or expansion potential is initiated by the depolarization of the membrane. So that if this part is starting to depolarize, so that it will transport the localized current will be spread into this area, so that this area was starting to depolarize, and then at the same time this area, the initial area, was starting to repolarize. Okay, so this is the transmission along the axon. So if you know that the transmission along axon is one by one, right? So if there are the myelin structure at here, so meaning that the transmission will directly jump to at this area. This part will not have the action potential occur. So when it jump directly go to here, so it will enable faster action potential. The nerve impulse is transmitted more faster compared to the non-myelinated neuron. Okay, now I will go to the synapse. Area was starting to repolarize. Okay, about the synapse, so I think structure of synapse I don't need to explain more lah, because you already learned in STPM BI. So we know that synapse is the junction between two neurons. So when there are the junction between the neuron normally is in the sensory neuron and interneuron or interneuron to the efferent neuron. So when there are the junction between the neuron, we call as synapse. So the normally structure of synapse, you know that there are the two main neurons, right? Presynaptic neuron and also postsynaptic neuron. So and then you will know that in presynaptic neuron you contain mitochondria that provide energy, and also the synaptic cleft that will contain many neurotransmitter. And then outside on the postsynaptic membrane, it will contain the neurotransmitter receptor that will be bind by the neurotransmitter, and then it will trigger the action potential to the postsynaptic membrane. Okay, about the impulse transmission in synapse. So when the impulse is generated until in the axon, and then when the electrical impulse is already reached the synapse, it will depolarize the presynaptic membrane. Okay, so the depolarization will cause that the calcium ion to be open, and then the calcium ion will be influxed into the presynaptic membrane. So this presynaptic, so the incoming of the calcium ion in the presynaptic membrane will cause the synaptic vesicle to fuse into the presynaptic membrane. So the synaptic vesicle that containing neurotransmitter was starting to penetrate the presynaptic membrane, and then to release the neurotransmitter. And then once they release the neurotransmitter, they will bind to the any ion channel or the receptor at the postsynaptic membrane. So when they are bind to it, it will trigger the opening of the ion channel, so that the sodium ion will be influxed into the cell, into the postsynaptic membrane, and then the potassium ion will be effluxed out of the cell, so that it will trigger the action potential. So this is another diagram. Okay. So normally activated receptor will send the impulse to the postsynaptic membrane. So once the neurotransmitter is already released into the synaptic cleft, it will bind to the postsynaptic membrane receptor. So when it is bind to it will trigger the action potential. So it will send the impulse to the postsynaptic membrane. So there are two types of impulse that will be produced. Either is EPSP, excitatory postsynaptic potential, or inhibitory postsynaptic potential. So excitatory meaning it will trigger action potential lah. So a change in membrane potential that bring to the neuron closer to firing, so will trigger the action potential. But for this one, inhibitory meaning it will prevent the action potential to be occur. You already say inhibit, right? So it will prevent the firing level. So about the EPSP, so the example of EPSP is normally is one of the example is acetylcholine. So this one I think I already mentioned last chapter, right? So when you know that when the acetylcholine is bind to the postsynaptic membrane, the ion channel will be permeable, both potassium ion and also sodium ion, so that the sodium ion will be go into the postsynaptic membrane, and then the potassium ion will be out from the cell, will be influxed out from the cell. So this one will require depolarize the membrane. Okay, and then another one is IPSP. So normally one of the example is GABA. GABA is one type of neurotransmitter. When it binds to the postsynaptic membrane, the ligand-gated channel now are permeable to both potassium ion and chloride ion. So if these channels are open, the postsynaptic membrane is hyperpolarized. It will not be depolarized, but it will be hyperpolarized. So this hyperpolarization will be known as IPSP. So hyperpolarization is the process that the membrane potential is lower than the resting state, right? So it actually inhibit the action potential to be occur lah, because the membrane potential is further from the threshold level. So in this situation, the IPSP it will trigger IPSP, and then no action potential will be occur. So the inside of the cell is negatively charged rather than positively charged. Okay, and then this one I just want to mention about the GABA mechanism only. So when you see that the most important is when the GABA neurotransmitter is released, it will bind to the receptor, and then the potassium ion and chloride ion will diffuse into it. And then about the termination of neurotransmitter signaling. So after the EPSP depolarize the postsynaptic membrane and already trigger the action potential, the neurotransmitter at the synaptic cleft will be clear. So if you know that termination of neurotransmitter, so the neurotransmitter will not be always appear at the synaptic cleft. So once it bind to the receptor, and then the already triggered action potential, it will directly be terminated. So either there are two types of termination. Either the neurotransmitter will be break down by an enzyme, or the neurotransmitter is reused again to the presynaptic membrane. Okay. And then among the EPSP and IPSP, there are the summation of postsynaptic potential. So normally, if you know that synapse in real situation, the junction between neuron and neuron not only contain one synapse only. You see this structure, you can see there are many synapses here. So normally if there are the summation process, so normally if E1, when the neuron at this neuron, the postsynaptic neuron accept the signal, EPSP signal from one part only, then there were no summation at all, because if only one signal one signal is received only, the signal is very vague, and then it cannot reach the threshold level, so no action potential at all. But if the signal is come from by one direction from single direction of the synapse, and the signal is come rapidly. So you see here this diagram, the signal is come rapidly, then it will reach the threshold level and generate action potential. But for this one, if no excitation, meaning that the signal it come from single direction, but the signal is very big, and then the signal between the another signal from the first signal, the period is quite long. So when they are long, so that the signal is not quite strong enough to reach the threshold level, and no action potential will be released. But for this one, if the signal with the two signals produced in the very short time, then temporal excitation will be produced, and then the action potential will be produced. And then another one is that the signal that come from two different synapses. So when there are two different synapses come from at the same time, so they will also have the action potential. This one we call as spatial simulation. So spatial is that the signal come from the two different sides. For temporal is the signal come from the same side. Another one, spatial simulation, is about the EPSP and also IPSP. So we know that IPSP is the inhibitory to the action potential. So when there are IPSP, although the signal is come from the different direction, but due to the IPSP, basal or IPSP, so the electrical impulse will not reach the threshold level, and no action potential will be produced. Okay, so I think this is the explanation. Okay. So normally input for an individual is insufficient to trigger a response. To see why, consider an EPSP arising at a single finance. So as the greater potential, EPSP becomes smaller as it spread from the synapse. Therefore, by a time particular EPSP reach the axon hillock, it is usually too small to trigger an action potential. So normally when the signal is come from the single synapse, actually the signal is very weak already compared to the signal that original come from the presynaptic membrane, because now it's already across the junction, so the signal is more weaker. So it requires temporal summation. So temporal summation is the signal that comes from the same synapse and then in very short time. And then spatial summation is that the signal comes from different synapses at the same time, it will trigger action potential too. But for if there are the involved IPSP inhibitory postsynaptic membrane, so normally it will not trigger the action potential, okay, because IPSP will counter the effect of the EPSP. And then this one is about the function of the synapse. So normally synapse function is act as the transmit information between the neuron, and then ensure that the transmission of nerve impulse is in one direction, and then it will be due to the release of neurotransmitter receptor site located on postsynaptic membrane. So it is act as a junction for summation. Additional of depolarization of several EPSP is able to produce adequate depolarization to reach threshold level to trigger action potential. And also it filter out the low level stimulus. So some stimulus like this or even like this will be filtered out lah. Okay, and then prevent overstimulation too. So when the stimulus is very high too, the action potential only occur once only, and the voltage or action potential also occur once only. It will not trigger the action potential in many times. Okay, if the rate of release of neurotransmitter more than rate of its synthesis, the release of neurotransmitter will be stopped as the junction for integration of impulse from many presynaptic membrane to produce coordinated response. Okay, another one is we now to go to the neurotransmitter. So in our syllabus, we will learn about two main type of neurotransmitter, which are acetylcholine and also norepinephrine, or another name is we call as noradrenaline lah. Okay, so acetylcholine, what are the function of acetylcholine? I think I don't need to mention much because I already mentioned already last chapter. So normally the main function is in the muscle stimulation, memory formation, and also learning process. So the receptor of this neurotransmitter is to the ligand gated ion channel. So normally it is found at the neuromuscular junction. So when the acetylcholine is released and then bind to this receptor, the ion channel will permeable to both potassium and sodium ion channel, producing APSP. Okay, and then G protein couple receptor. So normally it is in the vertebra, CNS and heart, and then both effect will reduce the rate of the heart pump, because the G protein in the pathway will inhibit this adenyl cyclase and open potassium ion in the muscle cell membrane. So for this one I just repeat again for the chapter 8 that you learn the regulatory. So we know that in the regulatory of heartbeat rate, when it release acetylcholine, meaning that it will reduce the heartbeat rate and the heart pump. Okay. Another one is about noradrenaline. So noradrenaline, if you remember that just now we learned, it will increase the heartbeat rate, right? So actually it is an autonomic accessory neurotransmitter, a branch of the PNS that make up of tyrosine. So normally the function is about the attention function, increased heartbeat, learning, memory, pain, mood, sympathetic response. Yeah. The acetylcholine is about parasympathetic response, right? So noradrenaline is about the sympathetic response. Okay. And another one neurotransmitter that you need to learn is dopamine. Although in syllabus that are not mentioned, but you need to learn too, because this one is very important. Later we learn about the drug mechanism. So actually it is also the one type of neurotransmitter in the CNS, the central nervous system, that make up from tyrosine also. Also normally it is help to balance between excitation and inhibition of the neuron. So it is very important in motor function, concentration in brain, affect mood, also involved in the motivation and reward process. So normally this one is more focused on the brain function, more into the mood, our mood and motivation process. And then this one is very important that we later learn about the drug mechanism. Due to the drug mechanism will involve this neurotransmitter. Another one is about gamma aminobutyric acid, GABA. So normally this one is the inhibitory neurotransmitter. So this is one type of example for IPSP lah. So normally if you exam want to explain about IPSP, you can include this example lah. Okay, next we go to the neuromuscular junction structure. Okay, so neuromuscular junction actually is similar to the synapse structure. It is also the junction between the neuron, but the junction is not between the neuron. It is between the neuron and the muscle tissue. So normally this one is only occur in the efferent neuron. So you see this diagram. This one is about the efferent neuron, and then this upper part is about our muscle tissue, muscle cell. So you see this diagram. So this is our efferent neuron, and then it would attach to our muscle tissue fiber, right? And then when they are receive the impulse, it will send the impulse into the muscle lah. Okay. So if you see this diagram, is more clear. So okay. So this is our effector neuron, motor neuron, and then this is the junction, and then this part is our muscle tissue. Starting this part will be our muscle tissue. This is our plasma membrane or plasma muscle tissue, and then this one is about our sarcoplasmic reticulum, one type of the SER in that unicule in the muscle only. Okay, so this is about the explanation. Okay, so it is the junction, and then muscle fiber region called motor end plane. and then it consists of pre-synaptic membrane, but the post-synaptic neuron now is called scleroma, sarcolemma. So this is a plasma membrane of the muscle tissue lah. So now it is not the post-synaptic membrane. You will say that it is the sarcolemma. Sarcolemma. Okay. At the surface of sarcolemma, they will also be a receptor for the neurotransmitter. Okay, and then the muscle fiber consists of transverse tubule system, T tubule, that deep down into the muscle fiber for transmission of action potential to myofiber. So you see here, the action potential is triggered. When the action potential is triggered, it will go to the T tubule, and then the action potential will be transferred into the T tubule into the muscle tissue, inside the muscle tissue, and then it will cause the contraction. Okay, next we go to the sarcoma structure. So I think sarcoma structure is the basic unit for the skeletal muscle that you already learned in semester 1 2. Okay, so this is about our skeletal. This is our skeletal sarcoma structure. So you know that this is our efferent neuron, neuromuscular junction, and then this is our T tubule. This is our sarcoplasmic reticulum. After that, after the sarcoplasmic reticulum, you will have the sarcoma there at here. So it is the basic unit of the muscle tissue that play the role in contraction and relaxation of the muscle. So this is the very important that once it receives the signal, it will directly cause the contraction of the muscle. So sarcoma is the very important unit to the skeletal muscle. So it's a basic conjugal unit of skeletal muscle. So you see here, this is our one unit of sarcoma. So you refer back in the sarcoma, there are two types of the element element we call as thick filament and also thin filament. Thick filament and also thin filament. So normally actin molecule and also myosin molecule. So this one I think you also already learned in semester 1, right? Right? Okay, so normally the muscle is made up of many muscle cells organized in bundle of muscle fiber wrapped by connective tissue lah. So within the muscle fiber, so you will see within our muscle fiber there are many bundle of muscle fiber at here, and then after that inside the muscle fiber we also have many myofiber at here, and then within the myofiber we have the sarcoma structure lah. And then each muscle fiber is surrounded by plasma membrane called sarcolemma, and then will have an invertation called T tubule. Within the single muscle fiber, there are many long longitudinal biofiber. The muscle fiber are made by the distinct unit called the sarcoma. Okay, so this is the structure of sarcoma that you need to know, that there are many lines at here, right? So the first one is I need to mention is about the I band. So if you see this I band is the place that only contain the thin filament only, meaning it's only contain actin filament only. And then there also have the Z line. You see here this Z line. So I band is this condition. So this condition is only contain the thin filament. And then you see the Z line. Z line is that the place that actin filament, the thin filament is attached together at the sarcoma end. If you can see here the actin filament is attached to each other. So this one will contribute to Z line. And then there are another line we call as M line. So M line actually is at the middle of the sarcoma. Okay, so even it is in the middle of sarcoma, so this one is about it only consists of thick filament only, or myosin filament. And then there are also the A band. So A band is the dark band that made up of overlapping of myosin overlapping of thin and thick filament. So this part is overlapping between the thin and thick filament. And then H joint is the narrow and light area in A band that made up of only thick filament only. Okay, so this one you need to know lah. Z line, like I band, A band, M line, and also H joint. Okay, next we go to the thick and thin filament structure. Okay, so we know that thick filament, the main component is about the myosin molecule, and then the thin filament, the main component is about the actin molecule. So we go to the thick filament first. So normally thick filaments will only contain the value of the protein myosin only protein myosin only, and then it consists of the long rod shaped region called the myosin rod and also myosin head. So you see this diagram. So this is the all the component, and then when it enlarge more, you will see that there are the myosin rod at here. This one is recalled as myosin rod, and then there are the myosin head at here. And then beside the myosin head, there are the actin binding site and also ATP binding site at here. So meaning that this part is there will bind to the actin filament, bind to the thin filament, and at here it will bind to the ATP. Okay, so this is very important in the muscular contraction. Later we will learn it. And then another one is about the thin filament. So you see that this is the thin filament structure. Actin will be the main component of as well, but actually there are also another component we call as troponin and also tropomyosin. So normally it is a two helical structure. They are coiled to each other, and then it also consists of tropomyosin, that is the regulatory protein which bound to the along the length of the actin filament. And then the tropomyosin is cover the myosin binding site along the actin filament to prevent the interaction of actin and myosin. So if you see this line, the line at here, it will cover the line here is the tropomyosin. It cover the binding site of myosin. So this part, lubang that have the hole, this one is the part that the myosin head will be bind. But in the relaxation process, normal situation, this hole will be covered by the tropomyosin because it want to prevent the myosin head bind to it. Only in certain situation the myosin head will bind to it only. Okay. And then troponin is the binding site of calcium ion. So you see here this more yellow color one is we call as troponin, and then it is the binding site for calcium ion. Okay, so this is the function for the neuromuscular junction. Okay, next we go to the muscular contraction. So we already learned about the structure of muscle, right? Actin filament, sarcoma, and thin filament, all these. Now we need to learn about how the muscle is contracted based on the hypothesis we know as sliding filament hypothesis. Okay, so this is about the summary process. So the summary process, actually the beginning process is similar to the synapse. What happened in synapse is similar to the neuromuscular junction. Okay, so once they already release the acetylcholine, so the neurotransmitter that will be released is acetylcholine. Once the acetylcholine is released, it will bind to the receptor on the sarcolemma. Okay, after that it will trigger the action potential. The action potential will be propagated along the plasma membrane and then will down to T tubule. And then once it reach the T tubule, and then when it reach the sarcoplasmic reticulum, it will trigger the calcium ion that inside the sarcoplasmic reticulum release out into the sarcoma. So when the calcium ion is inside the sarcoma, the calcium ion was starting to bind to the troponin, and then when the troponin is bind with the calcium ion, you see here, when the troponin is starting bind to the when the calcium ion is bind toward the troponin, meaning that meaning that myosin binding site will be exposed. Your tropomyosin will be changed their shape. Will they change their shape? It would change their shape and then expose the myosin binding site, so that the myosin head can be bind to it. If no calcium ion, it will like that. Your tropomyosin will directly block the myosin binding site as well. But if the calcium ion is already bind toward the myosin, bind toward the troponin, then the myosin binding site will be exposed. Okay, so after that they will trigger the 3-cycle reaction. So yeah, okay, so now this is the process that I think is similar that will happening inside the synapse, so I don't need to talk more. Okay, so this one I already mentioned lah. So normally when the axial potential is triggered, the axial potential will be propagated along the plasma membrane and then transferred down to the T tubule. And then once it release will causing the calcium ion channel to open, and then the calcium ion will be released out from the cytoplasmic reticulum. And then okay, now this is a very important step lah. Okay, so that when the calcium ion is already released into the cytosol of the muscle tissue, then it was bind toward the troponin, and then it will expose the myosin binding site. So now this is the sliding filament hypothesis that will happen to make the muscle to be contract. So the first step is that your myosin head will be bound to ATP, and it is in the low energy configuration. So the released calcium ion will bind to the binding site in the troponin complex. So the tropomyosin complex will be rearranged so that the tropomyosin will move and expose the myosin binding site at the globular actin. So at the other hand, the myosin head will be bound to ATP and it is in the low energy configuration. Okay, so when in the second step, when your myosin head is already hydrolyzed the ATP, since it already hydrolyzed the ATP, so your myosin head is now in the high energy configuration. So meaning that your myosin now is more active, is already activated. It will bind to the myosin. It will bind to the myosin binding site on the actin molecule at the globular actin, and then this one will form the cross bridge. So a bridge will be formed with the thin filament. So you will see here the atosine-myosin bridge or the cross bridge will be formed at here. Okay, so when it is formed, that this myosin head will be moved. Dia macam bergerak, will move the actin filament toward the center the sarcoma. Dia macam tangan awak bergerak, awak macam menyebabkan pergerakan of the actin filament toward the center of the sarcoma. So the myosin will release the ADP to the power shot that slide the thin filament toward the center of the sarcoma. So it will release the ADP, and then to slide the thin filament toward the sarcoma. And then on the other hand, your myosin head now will return to the low energy configuration state. Okay, so when there are the new ATP bind to the myosin head, the cross bridge is disrupted and release the myosin head from the actin filament. And then when the presynaptic nerve stop release acetylcholine, your cytoplasmic reticulum will begin pump calcium ion back into the cytoplasmic reticulum through carrier protein by active transport with the aid of ATP. So when your calcium ion now is already decreased in the cytosol, so that the filament will shift back to the starting position, and then the tropomyosin will block the myosin binding site again. Okay, so this is the final step lah. So you see here it is a repeat and repeat step, repeat and repeat step. So once they already sliding to the myosin head slide the thin filament toward the center of the sarcoma, it already it will release the ADP, so that the myosin head was starting to bind another ATP, become the low energy configuration, and then this process will be repeated again when the calcium ion is released again into the cytosol, and then it will bind to the troponin again. So the process is repeat and repeat again. Okay, so this is the structure when our muscle is contract, structure of sarcoma when our muscle is contract. So you see here when our muscle is contract, the actin filament, you see that the actin filament will be moved toward the center of the sarcoma. So you see here at the center, your M line should be not only contain the thick filament, right? But for now, if the contraction happen, your actin filament will be moved toward the center, move toward the M line. Okay. Okay, next is about the drug mechanism action. Okay, so in our syllabus, we only learn about two drug mechanism action. First one is about cocaine. Okay, so cocaine mechanism drug action is very important that I need to mention is about the dopamine. Okay, so you will see this diagram. So the left hand side is the normal situation, But this one is that the patient is already consume cocaine. Well, I think cocaine everybody know, right? Is one of the famous drug in the world. Okay. So in the normal situation, it look like the simple synapse only. When the signal is passed through, and then it will release the neurotransmitter. So the neurotransmitter that involved is dopamine. Okay. So when the dopamine it bind to the receptor, it will trigger the action potential, right? And then in the normal situation, your dopamine will be terminated, will be reuptake, right? Will be reuptake to the presynaptic neuron, so that the action potential will not be triggered again and again. But for the person that consume cocaine, the cocaine molecule is the inhibitor to the co to the is the inhibitor toward the uptake of the protein block of the dopamine. So it will block the reuptake protein channel of the dopamine, so that the dopamine will be released out and then bind to the receptor, but the dopamine cannot recycle, cannot be uptake again toward from be used back again for by presynaptic neuron, so that dopamine will maintain at the synaptic cleft, so it will trigger more and more action in the more and more action potential toward the postsynaptic membrane. So that is why that I already say dopamine is control our mood, right? So when you have the high amount of dopamine, awak masa gila punya lah, awak masa you are very shocked punya, macam very shocked, very high punya. So very high punya. So orang macam ni, why that? Because the dopamine is the dopamine is remain at here. The action potential is stimulated and triggered again and again. So when this kind of patient occur, the user now is addicted with the cocaine now. So when the drug is removed, normal reabsorption of the dopamine is occur. However, because you already have many dopamine at here, your receptor at here now will be decreased. Because our cell is also know, it will know that we also know that our cell cannot trigger the action potential in many times. One cannot trigger the potential again and again one. So when you want to decrease the times of action potential, it need to the receptor was starting to inactivated. So some of the receptor was starting to inactivated.
activated. So when the people that start to we start to not eating the cocaine, not consume cocaine, it will remain to normal reproduction. But due to the receptor now is very little for now, meaning that the dopamine release, the receptor that can accept the dopamine is very little. So in normal situation like 1, 2, 3, 4, 5, there are 5 receptor. But during when you have the consume cocaine, it will remain only 1 to 2 receptor only. So the only can maintain the normal functioning is to continue take cocaine only. So it will the decreased number of receptor already create a less sensitive nerve pathway. It is already less sensitive already. So if you want to create the normal function, you need to consume cocaine again. So that is why that the drug patient, the people that already consume cocaine or consume the drug, is not easily to be. If you want to stop it, it's not very easy. And then another one drug is what we call as QA. Okay, so normally QA is the poison drug. Poison drug. So normally it is generally used on the tips of hunting. So normally is hunting, used in hunting, because QA has the similar shape to the acetylcholine. So it acts as the inhibitor within the acetylcholine. So the QA, the person that already get the QA, it will bind toward the acetylcholine receptor or the sarcolemma neuromuscular junction. You know the acetylcholine play the role in the muscular junction, right? So you see refer to back here. Yeah, acetylcholine is play the role in the neuromuscular junction, right? So when the acetylcholine bind toward the receptor, it will trigger action potential toward here. But since the QA have the similar shape to the acetylcholine, so the QA will bind toward this receptor. So QA is inhibitor. So when it bind toward this kind of receptor, acetylcholine can bind it, so no action potential is triggered. So your muscle will not be contract. So no muscle contraction will be occur now. So your sodium ion channel will not be open, not activated, not open. So no sodium ion channel move into the sarcolemma, and then no EPSP, so no action potential. So no action potential, no calcium ion will be released into the cytosol. So no, the troponin is not activated. So everything is not activated. You see, refer to this, because no action potential, so the ion cannot pump into no action potential, so no action potential at all. So the calcium ion cannot release into the cytosol. So the calcium ion cannot bind to the troponin. So no muscle contraction as well. So no sliding filament hypothesis, it cannot be carried out. So no muscle contraction. So meaning that the people that get this kind of QA are able to contract the condition is called as flaccid disease. So or the people that the animal or the people that already hunting get the arrow that contain QA, the people know the will not die immediately, but they cannot move anymore because they cannot walk. Because our skeletal muscle play the role in walking, right? Muscle contraction, they cannot walk anymore. Okay. Okay, so that's all for nervous system. Okay, so I think I need to take a break first for a while. So we will continue back into 4 o'clock like that. Okay, we are back to the 4 o'clock. So see you later. Okay. And about the recording, it should be have record. I will not close the recording, but I don't know the YouTube have problem or not, because the live is very long. So I don't know, later you guys can, after the end live, you guys can see or not. But I hope can see refer back. And for the slide, I'm sorry to that, because this slide is my I cannot share to you guys. This bike is my effort I use many time to make it. So maybe you can take a picture or like that lah. So very sorry lah to you. Okay. So I think that's all for now. So we see you again 4 o'clock, and then I will proceed to the homework. Okay, thank you. And you can go to the washroom or drink a water like that. Okay. So now
Hello, hello. Okay, so now it's already 4 o'clock. Okay, so I will continue to another subtopic for this chapter, hormone. Okay. Okay, so what are the definition of hormone? So normally hormone is actually are the chemical that released by the endocrine gland in one part of the body that travel through the bloodstream, which affect the activity of the cell in other part of the body by binding specific chemical receptor on those cells. Okay, so the key term is about the endocrine gland, and then it will transport through the bloodstream, affect the activity of the cell, and then binding to the specific chemical receptor. Okay, so as we now know that there are many in human, there are many glands, right? Like thyroid gland produce thyroid hormone, pituitary gland, right? All the sexual hormone, LDH, secret here, hypothalamus, pancreas, insulin and glucagon, and also adrenal gland, all these lah. Okay, okay. Okay, so in human we will separate into two main type, either is steroid hormone and non-steroid hormone. Okay, so you need to know some of the example lah. So the example that we normally is we learn. So like the prolactin, prolactin is the non-steroid hormone, right? Non-steroid hormone, and then non-steroid hormone. So you go to ACTH, FSH, LH, ADH. All these actually is considered as non-steroid hormone. These are all the protein hormone. Only the normally is sex hormone like testosterone, estrogen, progesterone, and aldosterone, and cortisol hormone. All these will be considered as steroid hormone. So you need to know some of the example. Why? Because in examination it will not give you, maybe it will not give you the direct question, it will give you the hormones. Maybe you just want to describe the mechanism of acetylcholine. You want to describe the mechanism of hormone estrogen, progesterone. You need to mention, you need to know estrogen and progesterone is under steroid hormone. Or maybe it will ask you the mechanism reaction of ADH. Or ADH is non-steroid hormone. So you need to refer it. So if you don't remember it, although you know the mechanism, but because of you don't know the example, you will be rugi lah. Akan rugi lah. Okay, next I will consider about the steroid hormone mechanism. Okay, so for the steroid hormone mechanism. Okay, so for the steroid hormone mechanism is more simple compared to non-steroid, because the steroid mechanism steroid hormone is lipid hormone, right? One of the lipid. So the mechanism of the steroid hormone is known as gene activation. Gene activation, meaning that it involves the gene activate. It will involve the gene. It will involve the transcription and translation process. So meaning that when the hormone like aldosterone or progesterone or estrogen or any sex hormone or hormone, when they reach the target cell, this steroid hormone will bind to the protein receptor to form hormone receptor. They will reach the cell. It will directly pass through the plasma membrane into the cytoplasm, and then they will the receptor protein that fits to the this kind of hormone. So it will form the hormone receptor complex. Okay, so this hormone receptor complex later will enter into the nucleus, and then it binds to a specific section of the DNA that function as a gene. And then this bound protein will act as the transcription factor to stimulate the transcription of mRNA. So after the mRNA will move to the cytoplasm, the translation will be occur, and then after the translation, it will produce the protein, and then this protein will be diffused out from the cell to have another reaction. So this is the mechanism of steroid hormone we know as gene activation. So it will involve the transcription process and also translation process that you already learned in semester 3. So you should know what are their process. Okay, next is about the non-steroid hormone. Non-steroid hormone is more complicated compared to steroid hormone. So if you learn about non-steroid hormone, you need to learn about the signal transduction pathway. Okay, transduction pathway. So meaning that it is the transduction sometimes occur in a single step, but more often require a sequence of a change in a series of different molecules. So meaning that signal transduction pathway, meaning that it's realize the molecule. It does not involve only one molecule only. You need to one molecule activated first, and then this molecule will activate second molecule, and then this second molecule will activate third molecule, and then this third molecule will activate the cellular response. It cannot directly, the first molecule directly activate the cellular response, cannot. It need to realize to each other one. This one we call as the signal transduction pathway. Okay, so in non-steroid hormone, we will have the protein we call as G protein coupled receptor. So this is the receptor that the non-steroid hormone will be bound. So actually, it is a cell surface transmembrane receptor that works with the help of a G protein, a protein that can bind with the energy-rich molecule GTP. Okay. So if you will know that when the non-steroid hormone bind to this receptor, it will activate G protein. Okay, once it activates G protein, it will, the receptor is activated transaction, its cytoplasmic site will bind and inactivate G protein, causing a GTP to be disactivate G protein. Okay, so once the G protein is activated, it will dissociate from the receptor and then diffuse along the membrane and then bind to the enzyme. This is our activated enzyme. And then once activated, the enzyme can do a cellular response. Okay, so this is the general flow of the non-steroid hormone. Okay, so this is a full terms of the non-steroid hormone. So the mechanism of the non-steroid hormone is known as second messenger CNMP. So as I mentioned before, when the your hormone, any non-steroid hormone is bind to the G protein coupled receptor, it will activate it will become activate, and then this activated receptor will bind to the G protein, and then it bind to the G protein, it will activate the G protein. And then when the activated G protein will bind to the adenyl cyclase. When it binds to the adenyl cyclase, GTP is hydrolyzed and activating adenyl G cyclase, and then this adenyl G cyclase will convert ATP to CAMP. So that is why that the mechanism is known as second messenger CAMP, because this CAMP will act as the second messenger to activate the protein kinase. So this protein kinase later it will lead to the cellular response. So this is the overall process. So the hormone first will bind to the receptor first, and then the receptor is activated, it will bind to the G protein, and then G protein is activated, it will bind to the adenyl cyclase, and then when the adenyl cyclase is activated, it will convert the enzyme convert ATP to become CAMP, and then once it from the CAMP, it is act as the second messenger, it will activate the protein kinase, and then this protein kinase will lead to the any cellular response so any function like amphiphony, ADH, all that function okay. And then this is one of the example in Campbell mechanism by adrenaline. Okay. Okay, so that's all for the steroid and non-steroid hormone. Okay, next I will go to the plant hormone. When it is activated, it will bind to the adenine cyclase, and then when the adenine cyclase is activated, it will work at the peptide. Okay, about the plant hormone, we will have to learn 5 types of plant hormone. First one is auxin, gibberellin, cytokinin, abscisic acid, and also ethylene. So these are the 5 hormones that we need to learn in our syllabus. Okay, so the first hormone, that famous hormone, is about the auxin. Okay, about the auxin. So another name of auxin is about indole-acetic acid, IAA. So normally this auxin is produced in the shoot and apical meristematic and young leaf from tryptophan. So the key production is at the apical meristem of the plant, and then normally it is transported from cell to cell down the stem, and then it only move from tip to the base only, not in the reverse direction. And then the growth of the plant toward light, away from the light, or either the growth of the plant that toward the light or away from the light is called phototropism. In shoot, the cell on the darker side will elongate faster than the brighter side. Okay, so the positive phototropism will occur when the high auxin concentration accumulate at the dark side of the stem, stimulate the growth and bend toward the sunlight. And then geotropism is the growth of the root and shoot toward the gravity. Okay. And then positive geotropism occur when a high auxin concentration accumulate on the lower side of the root, inhibit the growth and bend downward. And then negative tropism is that when high concentration auxin accumulate on the lower side of the shoot, stimulate the growth and bend upward. Okay, so I think phototropism is the terms that you already learned, right? No right. So when the growth of the plant that either is away or toward the sunlight is called as phototropism. So in auxin molecule, so normally auxin molecule is accumulated at the tips of the plant. So you see, refer to it. When there are the sunlight, the auxin will be accumulated at the darker side part. As you see here, it is exposed to the sunlight, right? So no auxin molecule at that. All the auxin will be accumulated here. So meaning that the auxin will start to elongate at here. So it will start to elongate at here, and then it will bend toward the sunlight. So that is why that we look like the plant will be looked forward to the sunlight. This is the reason lah. And then about the geotropism, I already mentioned, is about the gravitation, right? So normally if you see here, this is the plant. So normally in root, the auxin will slow the growth in the root and curve downward. And then auxin will stimulate the growth in the shoot, and the stem will curve upward. So this is the very basic theory that you already learned. I think STPM you already learned already. And then for the phototropism, there are many experiments. Just a recap that you already learned in STPM 2. Okay, so one of the first experiment is by the Charles Darwin. So this scientist, he want to know why the plant is show positive phototropism. So there are 1, 2, 3, 4, 5. So it assumes 5 conditions. So the first one is control lah. So in normal situation, in normal situation when your sunlight is here, your plant will be bent toward the sunlight. Okay, how about when your tips is removed, meaning your collotype, the plant tips at here is removed. So meaning that there are no auxin at here. So no response, meaning no auxin at here. Okay, how about when you cover by the opaque cap? Opaque cap meaning it cap, meaning that this is totally black cap, it totally block the sunlight. So if you totally block the sunlight, yeah, no response too. But if you if you use transparent cap, yeah, because sunlight can penetrate it, yeah, so there are the response at here. So when if you cover the plant at the stem of the plant, not the coleotype, not the tips of the plant, you will show the response too. So in conclusion, this experiment shows that there are something inside the coleotype that make the plant bend toward the sunlight. Okay, next is about the experiment by Jensen. So Jensen experiment is using the barrier. So it using the two experiments. So meaning that the first one is it using the one of the barrier, mica barrier. Directly, one of them is it will not cover all the part. It only cover half of the part only. So when the barrier is covered to the left hand side, assume that the sunlight is on the right hand side, no response at all. But if your cover is at the right hand side, yeah, response will still occur. So in conclusion, you can conclude that auxin is stimulated at the darker part. But for this part, since you already block here, so no auxin is accumulated at here. Auxin cannot accumulate at here. And another one is this kind of experiment. If directly cut down, no response. But if you directly use a transparent cap at here, yeah, you still have the response. And then another experiment by veins. So normally this one it cut the tips of the plant is directly cut. So when it cut, allow time for hormone to diffuse into alga block. So you need to collect the hormone first. Okay, so when they collect the hormone in the alga block, and then it will transfer this alga block that already reached the hormone directly directly attach toward this cut tips plant. So when it attach to here, it will found that it still have bending, but the bending occur outside block, causing bending of should not exposed to light. So meaning that auxin move now the shaded region for response of the group. So meaning that the auxin will be diffused out to the plant and stimulate the growth. And another experiment by Bridge. Okay, so this one experiment is also cut the tips also. So cut the tips. So barrier block auxin development. It take the barrier to block the auxin development here, and then dark grown shoot, and then auxin induce the curvature at here. And then when the barrier in auxin is blocked, your auxin concentration will dependent of the curvature. So you will see this diagram for the part A1, because you put a barrier at here, so the auxin is concentrated at here. So normally your B agar will contain high concentration of auxin compared to A. So if you put toward the plant, so B will be more curvature compared to A lah. Okay, and then we go to the function of the auxin. So what are the function of the auxin? So the main function of the auxin is about the cell elongation. So it is responsible in the primary growth. Okay. So if you see this diagram, auxin increase the activity of proton pump, which pump the hydrogen ion from the cytoplasm. So the hydrogen ion will reduce the pH of the cell wall. So this will reduce the pH, activate weight shaped protein called asparagine, which separate the microfiber from the polysaccharide. So it need to loosening the cell wall first. Because cell elongation, right? Cell elongation meaning it need to elongate the cell, become more longer, right? So if you want to make the cell become more longer, you need to loosen the cell wall first. So the microfiber play the role inside the cell wall, right? So this reduced pH will activate the protein called the asparagine. So this asparagine protein will separate the microfiber from the polysaccharide. So this polysaccharide will cleave the cell wall loosening enzyme So this will loosen the microfiber and making the cell wall more flexible. So water uptake and increased sugar may cause the cell wall to elongate. At the same time, more microfiber are not formed. So so the main purpose is to loosen the microfiber. Because microfiber play the role in the interaction between the interaction of the cell wall, right? Cell wall, right? So you need to loosen the microfiber, make the cell wall become more flexible. So after that, the water molecule will be, when the water molecule is diffused into the cell, it will become more turgid, and then make the cell can be elongated. And then this is about the explanation. Okay, next is about the enhanced apical dominance. Okay, so you need to know what is the apical dominance. So apical dominance is the inhibition of the growth of axillary bud by the apical bud of a plant shoot. Okay, so if you refer to this diagram, apical bud is play the role prefer for the sugar zinc as a major site of auxin biosynthesis. So this is the place that the auxin will be produced. So when this apical bud is maintained at here, it will it will promote the apical dominance. So apical dominant meaning that no growth of the axillary bud. So we will see here, this is what we call as axillary bud. So if no auxin at here, you can see that the branch there will be many branch occur at here. So this is what we call as removal of apical bud. So if there are auxin at here, this phenomenon will not be happen. But if the auxin is scarred, this phenomenon will be happen. So this is what we call as apical dominance. And then this is the other function. Auxin also can alter gene expression, causing cell elongation to produce new protein. And then this protein can short large transcription factor to activate the expression of other gene. And also auxin can promote food growth and induce panetocarpy, food development without fertilization. And auxin also inhibit excitation by maintaining the structure of cell wall. And also auxin play the role establishing phyllotaxy, arrangement of leaves or stem, and also cell division in vascular cambium to secondary growth. Okay, next hormone is about cytokinin. So normally cytokinin is synthesis in the root. The primarily synthesized site location is in the root. And then the main function is to regulate cell division in shoot and root, and modify apical dominance, promote lateral bud growth, promote movement of nutrient into zinc tissue, and stimulate seed germination. Okay, so normally roles of cytokinin is normally collaborate with the auxin. So normally acting in concert with auxin, cytokinin is stimulate cell division and influence the pathway of differentiation. So compared to auxin is play the role in cell elongation. Dia hanya besarkan sel je, memanjangkan sel je. Tapi cytokinin it will produce many cells because cell division. So where a piece of parenchyma tissue from a stem is cultured in the absence of cytokinin, the cell grow very large but do not undergo mitosis. But when the cytokinin is added, the plant will plant cell will in very large amount lah. So normally the ratio of cytokinin and auxin is control the cell differentiation. So normally auxin, when your ratio of auxin and cytokinin is equal, concentration of auxin and cytokinin is equal, you will induce the catalyst. And then when your auxin is higher than cytokinin, you will induce rooting process. When your cytokinin is higher than auxin, it will induce shooting process. So this is very important in plant tissue culture lah, for the purpose, any purpose that you need to do. You need to do rooting or you need to do shooting. Yeah. And then another one is control or apical dominance. So same as the auxin, when your axillary part, when your apical part is already removed, your cytokinin coming from the root antagonize the action of auxin and limited amount of axillary part growth. Therefore, the axillary part will farthest from the apex and increasingly elongated. So normally, if you remove the apical part, your remaining part will receive more sugar for growth. So auxin also decline, particularly near the gut surface. This decline also allow the tropomose axillary part in the particular to go and take over as the new apical part. Another one is role of cytokinin is about the anti-aging effect. Okay, so cytokinin can slow down the aging of the certain plant organ by inhibiting the protein breakdown. So it can stimulate the RNA and protein synthesis and mobilizing the tissue for surrounding tissue. So it leaves removed from a plant are dived in a cytokinin solution. They stay green much longer than otherwise. So they are prevent the plant become older lah. Maintain the plant become green color and cannot become more older lah. Okay, next hormone is about gibberellin. So gibberellin actually play the role in seed germination, and also seed development. So normally the gibberellin is found in the meristem of apical part and also root, young leaf, and also developing seed. So normally the main function is about seed development and seed germination, and also pollen development, stem elongation. Yeah, so for the first role is about the stem elongation. Stem elongation. So best known for stimulating stem and leaf growth by enhancing cell elongation and cell division. So they activate the enzyme that loosen the cell wall, facilitating the entry of expansion protein. So this one is more focused on stem elongation. For auxin is more focused on cell elongation, primary growth one. But for this one is more about the stem. So the gibberellin act in concert with auxin to promote stem elongation. And then gibberellin also involved in promoting the rapid elongation of a floral stalk that occur naturally in many plants when they initiate flowering. And then gibberellin also promote the fruit development, fruit growth. And then this is one type of example of the grape bunch that be induced by the gibberellin lah. Okay. Yeah, another one is about seed germination. This one I will explain more detail in next chapter. Okay. Next hormone is about abscisic acid. Okay, so this hormone, the primary site that produce this hormone is almost all plant cell. All plant cell can produce this kind of the grape bunch, this of the abscisic acid. Okay, so normally the main function is to inhibit growth, and they promote stomata closure during stress, promote seed dormancy, inhibit early germination, promote leaf senescence, promote desiccation tolerance. So normally this main function is to inhibit growth. Okay. Okay, so the main role of the FCC acid is about seed dormancy. So seed dormancy can increase the likelihood that seed will germinate only there are sufficient amount of light. So seed dormancy is that it prevents seed to germinate. It will maintain the shape of the seed, inhibit the seed to be germinate. So high level of ABA in maturing seed inhibit germination. Okay, next one is about the drought tolerance. Okay, drought tolerance is normally it is the part that in very hot sunny day, in the place at very hot sunny day, FCC acid is the key role to play the role in drought tolerance. So when a plant begin to fail, ABA will accumulate in the leaf, cause stomata to cause rapidly to reduce the transpiration. Because in the uni hot sunny day, where the plant does not have the sufficient water, transpiration rate is very high. So very high. So when we want to reduce the transpiration rate, to prevent the water loss, ABA play the role to close the stomata so that the water will not flow, will not be evaporated. By affecting second messenger, ABA will also cause potassium ion in the plasma membrane of the gut cell to open, leading to the massive loss of potassium ion from the cell. So the accompanying osmotic loss of water will reduce the gut cell turgor and lead to causing osmata. Okay, so water shortage stress in the root system and ABA transported from the root into the leaf may function as early warning system. Okay, next, the last hormone is about ethylene. So normally it is a gaseous hormone that be produced by both part of the plant. However, it is produced in concentration during synthesis, addition, ripening, food ripening. And then normally the key function of this hormone is about the food ripening that everyone know. But actually, beside from the food ripening, it also play the role in leaf absorption, leaf lateral expansion, enhance rate of ascendance, and promote root and root hair formation. Okay, so the key function is about the food ripening. I think I don't need to talk much. Everyone should know already. Okay, so normally it will provide the enzyme break down the cell wall component to soften the food, and the conversion of starch and acid to sugar make the food more sweety. So another one is triple response to mechanical stress. Okay, so the hormone had then instigate a growth maneuver known as the triple response that enable the shoot to avoid the obstacle. So there are the three main response when there are the mechanical stress here. So you will see here this part when your ethylene concentration is very high, your shoot will be bent very your shoot will be bent very curve. So normally the three main response is slowing the stem elongation, thickening of stem, and also curvature that cause the stem to start growing horizontally. So normally this is what happens when there are the barrier at there. So this one is the experiment, but in the real situation, when there are the barrier at here, so that the plant, they found that they already cannot grow upward, they will stimulate higher amount of ethylene, and then it will give the tree response of this tree response. So it will slow the stem elongation. The stem thickening of the stem will be increased, and then it will cause the curvature. Okay. Another one is about the sensors. Okay, sensors normally is program depth. A definition of sensors is about the program depth of certain cell or organ or the entire plant. So normally, if you know that same like the animal, all the cell will be die right until the certain period, all the cell or the organ will be die. So normally this hormone play the role in modify the sensors, meaning that on a schedule do not simply shut down the cellular machinery and avoid that At the molecular level, onset of programmed cell death is a very busy time requiring new gene expression. So newly formed enzyme will break down many chemical components. So plant salvage many of plant breakdown process. So the birth of the Italy is almost always associated with the death of the cell during senescence. Another one is about the leaf excision, meaning it is the loss of the leaf from the tree that prevent desiccation during this seasonal period. So normally this one is that the leaf will be come out falling down from the tree lah. Okay. So this is the explanation. You can make yourself. Okay. And then there are also other plant hormone that you don't need to meet very focused for it. There are berry-sostenoid, desmonate, and also stevocanoid. So these are the hormone that we nowadays that the scientists discover, but in our syllabus you don't need to focus about it. Okay, now I will proceed to the last topic, which is about the cytochrome. Okay, cytochrome. So cytochrome actually is about the light response. So normally it is about the pigment that absorb mostly red of infrared light that regulate many plant response to the light, including seed germination and also shed avoidance. Okay, so you know that there are two main light, red light and also far red light. So cytochrome is the pigment that will absorb either red light and far red light based on the wavelength lah. So when the cytochrome PR, this pigment will only absorb red light at the maximum wavelength of 660 nanometer. And then cytochrome PFR, cytochrome far red light, will absorb the far red light only in the maximum wavelength of 730 nanometer. Okay, so normally during the day, your far red light is higher than red light because sunlight contain more red light, thus it will convert PR to PFR. So meaning that during the sunny day, PFR will be higher than PR. So because the sunlight contain more red light, and then same situation in night, PR will be more than PFR because the far red light because in the light night contain higher amount of far red light, so it will convert PFR to PR. Okay, so normally red light will increase the percentage of seed germination that PFR is the form cytochrome to trigger the development leading to seed germination. Oh, you know that PR absorb red light, right? So when it is absorb red light, it will convert into PFR, and then when PFR absorb far red light, it will convert into PR. And then PFR is very important. It will play the role in seed germination, inhibition of the growth and stimulation of branching, and setting internal clock and control of flowering. Okay, next is about the photoperiodism. So actually it is a physiological response to specific night or day length such as flowering. So in our you know that we have two type of plant, right? Either is long day plant or short day plant. So long day plant meaning that this kind of plant will only flower then when the plant is exposed sunny day more higher than the critical point, critical dark period. And for the short day plant, meaning that only the plant only can flower when the daylight period is shorter than the critical dark period line. So normally long day plant, the example is tomato, rice, and dandelion. And then the short day plant is example is soybean, poison teas, and chrysanthemums. Okay, so how the short day plant is flower? So normally Okay, so normally short-day plant will flower when daylight period is shorter than critical length lah. So when the dark period is longer than light period, PR level will increase and then PFR level will decrease, right? Decrease, right? Because PFR will absorb the PFR will absorb the far red light, convert to PR, so PFR will decrease. So more far red light is absorbed by PFR, convert to PR. So high level of PR will in short daytime will stimulate the inactivate florigen. So this is the flowering hormone become active florigen hormone. So the hormone will act as a flowering hormone, induce the flowering. Okay, so similar to long day plant. Long day plant only flower when the daylight period is longer than critical length, so that when the dark period shorter than light period, PR level decrease and PFR level increase. So this is due to the red light is absorbed by PR and convert to PFR. So high level of PR in long day plant will stimulate the inactive florigen, become active florigen. So the florigen hormone will act as the florigen hormone to induce the flowering. So this is the same process, just that the wavelength cytochrome activated is different. So short day plant the PR is increased, but the long day plant PFR is increased. And then this is the diagram. So normally this is very famous question that normally will ask in the objective question. Normally it will ask you if this is the this still is the normal situation. So you see here, when the with the daylight period is longer than critical length, so only the long day plant is flower, short day plant is not flower. But if the daylight period is lower than critical length, only the short day plant is flower. But if you refer to this, actually short day plant, the daylight is shorter than critical length, but short day plant is not flower. Why? Because there are the red light is exposed into the dark period. So red light when it exposed to the red light, it will increase the absorbed by red light, it will increase the conversion of the PFR. So the PFR is increasing, so that the long day plant will be flower. Long day plant will be flower. So similar to this, so if PR and PFR, if there are one more far red light is induced to it, so short day plant will be flower. And then if one more time red light is induced to it, long day plant will be flower. If one more time far red light is induced to it, short day plant will be flower. So this is very famous question in objective. So normally it's very tricky lah. So you need to alert for it. If there are red light here, so meaning that the plant will absorb red light and convert to PFR. So when the PFR increase, so that only long day plant is flower, so short day plant is not be flower. Okay, so that's all for this chapter. Okay, so that's all for this chapter. Now it's already 4:42. Okay, so we will take a break. We will continue another chapter. 5:30. We will continue another chapter in 5:30. Okay, so see you next and take a break. Okay. Now it's already 4:42. Okay, so we will take a break. We will continue another chapter. 5:30. We will continue another chapter in 5:30. Okay, so see you next and take a break. Testing, testing. Okay. Okay, now it's already 5:30. Okay, so today I will continue the last chapter for this today lah, for today class. So this is about the chapter 4, chapter 10, about the reproduction, growth, development and growth. Okay. Okay. Okay, now it's already 5:30. Okay. Okay, so this is our last class, last chapter today. So chapter 10, reproduction, development and growth. Okay. So firstly, we will learn about the sexual reproduction in human lah. Okay. So first topic, we need to learn about human sexual organ. So I think our male sexual organ, I think I don't need to explain much lah. So our male sexual organ, you will see that our main male sexual organ is about the testes, penis, and also the prostate gland. Okay. Then about the female sexual organ, so this is our female sexual organ. So this is vagina, uterus wall, ovary. Okay. Okay, so now I will go to the base of the sperm. Okay. Base of the sperm. So when the male and female do the sexual activity, male will produce a sperm, right? Will educate the sperm. Okay, so this one this theory I need to explain about how the sperm is being produced from the male and how it is transferred from the male sexual organ toward the female sexual organ. Okay, so normally sperm are produced in the semi-numerous tubule within the testis through the spermatogenesis. So we know that our sperm is produced inside the testis through the spermatogenesis process, right? Okay, so once the sperm is produced, it will pass through the choroid of epididymis, the place where the maturation of sperm occur about 3 weeks. So later it will go toward this epididymis for maturation process of the sperm. After that, it will go through the after when there are the sexual activity during ejaculation. So this is the female spatial activity. Activity, the sperm will transport through the vas deferens. So it will transport through this vas deferens. This is one type of the tube. One type of the tube. Okay, so the vas deferens will extend until behind of urinary bladder. So later it will form. There is a joint of the duct from seminal vesicle that will form the ejaculatory duct. So along the ejaculatory duct, the seminal vesicle, prostate gland, and the bulbourethral gland will secrete semen fluid that can be educated. So you see here. So normally when the sperm is already reach the seminal vesicle, and then it will go to the duct. So there are 3 ducts: ejaculatory duct, prostate gland, and also bulbourethral gland. So these 3 will starting to secrete the semen, the fluid that can be transport the sperm into the female sexual organ. Okay, so normally seminal vesicle about 60% semen, which thick, yellowish, and alkaline, that contain mucus, fructose, and ascorbic acid. And then the prostate gland will secrete the semen directly into urethra, which is milky and thin. Okay. And then the bulbourethral gland will secrete clear mucus to neutralize any acidic urine remaining in the urethra. So the sperm within the semen will enter to the urethra through the ejaculatory duct, then to the penis. So you will see this diagram. After the ejaculatory duct already secrete the semen. All these already secrete the semen, so that the sperm will pass through the penis inside the semen, and then it will go to the penis. So the penis is an arterial copulatory organ that delivers sperm into the female reproductive tract that contain the urethra and cylinder spongy erector tissue. Okay, and when this tissue is filled with blood during sexual intercourse, the high blood pressure will cause the vein and penis seal off, resulting penis to engulf with blood. So therefore, the penis erection will occur when penis increase in length, diameter, and thickness to cause the penis can be inserted to vagina. So this meaning that during the normal situation, this is our penis in our situation that does not have the sexual intercourse. But when there are the sexual intercourse, the blood will be filled up inside the penis, and then it will become penis erection, meaning dia akan berdiri lah. Ini penis untuk lelaki akan berdiri, and then increase in length, diameter, and firmness to cause the penis can be inserted to vagina. And then lastly, the sperm within semen will swim toward the cervix, then toward the oviduct for fertilization if the secondary oocyte is ovulated. So this is the overall process of the sperm passage from the testis into the uterus. Okay, next we go to the spermatogenesis. That does not have the sexual intercourse, but when there are the sexual intercourse, the blood will be filled up inside the penis. Next we go to the spermatogenesis. Okay, spermatogenesis, I think everyone should be know. You already learned in SPM, right? So it is a process to produce sperm. Okay, produce sperm. So this is the overall process of the produce sperm. So the spermatogenesis is occur in the seminiferous tubule. So in the at the beginning, the predominant germ cell will undergo amatotic diffusion to form the spermatogonal stem cell lah. So spermatogonal stem cell, after that it will undergo mitosis also to form spermatogonium, and then after that it will also another mitosis. It will form the primary spermatocyte. Okay, so beginning primary spermatocyte, it will undergo meiosis process. So we know that meiosis process is special for only the sexual organ only, right? So it will undergo meiosis one process to form secondary spermatocyte, and then after that it will undergo meiosis 2 to form the early spermatid, and then after that it will undergo differentiation to form the sperm that we will be produced. So it's more easy. So you just remember this diagram, then you will directly know lah. Okay. And then this is about the explanation. Okay, about the sperm structure. So sperm structure, actually you see here, sperm structure contain the head and our tail, right? And then at the middle, they contain the middle piece. It contain rich of mitochondria. So normally, there was or three part: head, middle piece, and flagellum. So normally, head it contain the nucleus with a membrane set called ecosome. So this is very important inside during the fertilization. That later we will learn lah. Okay, and then middle piece it contain the many mitochondria, and then the flagellum. Another name is about the tail. It act at the swimming lah. The sperm will be swim toward the oviduct, and then for the fertilization process. Okay, next is about oogenesis. So oogenesis is more complicated compared to spermatogenesis. Okay, so oogenesis actually is the development of major oocyte in the ovary. So oogenesis is very special compared to human, because compared to male, because in male spermatogenesis is occur when you are born. Even you are already in baby, when you are born, or you are in major in puberty stage. But for the oogenesis, it will already begin when you are in embryo stage. When the female is in the embryo stage, the oogenesis is already begin. So same as the spermatogenesis, they begin with the primordial germ cell, undergo mitosis, will form the oogonium. Okay, oogonium. Okay, after that it will undergo another mitosis process, form the primary oocyte. But at that time, primary oocyte will undergo meiosis to form secondary oocyte. But the key point is it will only stop in the prophase 1 in meiosis 1. It will not undergo the complete meiosis 1 process, and then it will stop at prophase 1. This is in embryo state. Okay, so after that it will born, the female will be born, right? From the female baby. After that, oogenesis will be stopped until the female reach the puberty stage. Okay, so once the female already reach the puberty stage, it will complete the meiosis 1, and then continue the meiosis 2, but it will stop at the metaphase 2 to form the secondary oocyte. So this secondary oocyte is stopped at the metaphase 2, and then at that point it will form the first polar body. So this is not a oocyte. It will be excreted out from the ovary lah. So only one ovary only will be produced. Okay, after that it will undergo ovulation. Okay, so after ovulation they will have two process. Either they will have a fertilization. So when they are the fertilization process, it will complete meiosis 2 to form the fertilized egg. But if there are no fertilization at all, it will the meiosis 2 process will not be completed, and then this oocyte will be excreted out from the female body through the menstruation. So that is why that every month the girl will need to undergo the menstruation every month, because every month this secondary oocyte will be released out from the female body. Okay. Okay. So that's all for the oogenesis. So it's, I think, very simple because you already learned in SPM, right? Okay. Okay, next is about menstrual cycle. So we already learned about that. After ovulation, if got fertilization, then it will form fertilized egg. The oogenesis is complete. But if no fertilization process, meaning oogenesis is not completed, it will undergo menstruation. So menstrual cycle. So that is why that every month a girl, the female, need to undergo this lah. So this is why that is called a cycle, because you need to female need to undergo this situation every month until you are reach menopause. Okay. So in menstrual cycle, there are very 5 important hormones that you need to learn. So the first 2 hormones is follicle stimulating hormone FSH and luteinizing hormone LH that are stimulated. They are secreted from the anterior pituitary gland. And then another hormone is gonadotropin releasing hormone GnRH that is secreted from hypothalamus. If no fertilization process, hypothalamus. And then there are another 2 hormone, progesterone and estrogen, that is secreted by corpus luteum. Okay, corpus luteum. Okay, so what are their role? So I think so this is the overall process of the menstrual cycle. So at the beginning of the menstrual cycle, you will see here, at the beginning of menstrual cycle, your hypothalamus will be stimulated, and then release the GnRH hormone, gonadotropin releasing hormone, to stimulate our anterior pituitary gland. Okay, so once the anterior pituitary gland is stimulated, it was starting to release FSH and also LH hormone. So FSH was stimulate the follicle growth of the follicle growth. So you see here, see this is the growing follicle. Our ovule will be enclosed inside the follicle one. So follicle stimulating hormone is to stimulate the follicle of the growth Okay, the growing follicle will secrete estrogen. Okay, since the concentration of the estrogen is low, so it will inhibit the production of the FSH and LH. But when the estrogen and the follicle is starting to stimulate higher amount of estrogen, the estrogen will stimulate the GnRH secretion, causing more CnRH is released. So when GnRH is more released, it will stimulate again the LH to touch the LH level. LH hormone will also increase. So GnRH will start to stimulate anterior pituitary gland again, and then trigger high secretion amount of LH. So when the LH is secreted in large amount, LH hormone will stimulate the ovulation, where the secondary oocyte will release out from the Garfield follicle. So at here point, this is the day 14. So you see this point, that LH hormone normally is very high in 12 day, day 12, and follicle stimulating hormone is normally high at day 8, day 9, like that. So LH will stimulate the ovulation, meaning that our secondary oocyte here will start to ovulate, will release out from the ovary, and then the LH will stimulate the rupture follicle to develop into the corpus luteum. So the corpus luteum now is formed. Okay. So once the corpus luteum is formed, the corpus luteum will start to secrete the progesterone and estrogen that inhibit the secretion of GnRH. So you will still remember that corpus luteum will start to secrete the progesterone and also estrogen in large amount. Okay, so when it is starting to secrete these, it will inhibit the hypothalamus to secrete secrete GnRH. So no GnRH, so anterior pituitary gland will not be stimulated, and no FSH and LH will be produced. So that this is to prevent the maturation of another egg. So at that time, you need to prevent that the another egg inside the ovary to be matured. So it need to mature one by one. Okay. So if there is no pregnancy, low level of AH and LH will cause the corpus to degenerate and also generating the level of progression and estrogen. So if no pregnancy will occur, no fertilization, no fertilization, our corpus will start to degenerate. So you see here, if the corpus no fertilization process, our progression and estrogen level start to decrease in large amount. So when they start to increase, this will cause the artery in the wall, endometrium wall will be constrict. So we know that the function of the progression and the estrogen is to thickening up the endometrium wall in the uterus, right? Because it need to prepare for implantation process. But now, because no implantation, no fertilization process, so that the progression and estrogen will be reduced. So when it reduces, meaning that this endometrium wall cannot be maintained, so it was starting to rupture down, so that the menstruation process is begin. So the menstrual flow process will stimulate the new set of ovarian follicle begin to go. So it will become recycle, and then it will stimulate another follicle to go. It will stimulate the hypothalamus, repeat again until the process is reach the fertilization. If got the fertilization, then the process is stopped until here. If no, the process is repeat again. Okay. And then this one is another diagram, so similar. Okay, next we go to the human embryo development. Okay, so in human embryo development, the first thing is we need to do a sexual intercourse. We need to sexual activity. After we do the sexual activity, when the sperm is already go to the vagina of the female, it will flow to the oviduct of the female, right? So that there will fertilization process. So once the fertilization is already completed, it will go to the implantation. After implantation, it will go to the gastrulation. After that, it will go to organogenesis, and then after that, it will go to the parturition. Parturition is the process that the baby is starting to release birth lah. Okay, so this is a summary process for implantation. So normally, when you inside within the ovulation, when the ovulation is starting, secondary oocyte release, and then got the sperm is already here, and then it will penetrate into the secondary oocyte, will pass through the fertilization. Okay, so once the fertilization is occur, cleavage will now to begin. So normally it will starting to begin the cleavage, become 4 cell, 8 cell, 16 cell, 32 cell, until it will become the ball site cell we call as spermatocytes. And then this spermatocyte was starting to implant into the endometrium wall. And then now pregnant. Next we will proceed to the gastrulation and organogenesis. Okay, so now we go to the fertilization. Okay, so in fertilization process, when the sperm is already come toward into the secondary oocyte, our sperm head will be contact into the secondary oocyte, so that our sperm will have the ecosome enzyme. So ecosomal reaction, so meaning that our sperm was starting to release the hydrolytic enzyme from the ecosome to make a hole from the secondary oocyte, so that growing actin filament from the ecosomal process, which protrude from the sperm head and through the jelly cord. So the protein on the surface of the ecosomal process bind to the specific partner protein to the egg of the plasma membrane. And then after they already penetrate into the process, they will contact and fusion of the sperm nucleus into the egg membrane. And then after that, it will go through the cortical reaction, meaning that our cortical granule that inside the secondary oocyte in the egg will fuse with the plasma into the plasma membrane, so that the secreted contents will click off sperm binding receptor and cause the fertilization envelope to form. So that this is the process to separate the cell from the plasma membrane to ensure that the fertilization only occur in once. Only one sperm can be only penetrate into the ovary only. So this will act as the slow block of the polyspermine. Okay, so this is the overall process. Okay, so normally in human, this one is more about in animal. So in human, normally in human there were additional process before the fertilization. The sperm undergoes capacitation. So capacitation actually is a maturation and secretion process in sperm in female reproductive tract. So the sperm will become more motile and able to sperm to undergo fertilization. So after that, they will flow through the corona vediata right here, through the venosa using the hyaluronidase enzyme. Okay, after that it will reach the gena pellucida. So corona vediata is the cell that surrounding from the ovary, surrounding from the secondary oocyte. So they need to penetrate this cell first using this enzyme. After they reach the zona pellucida, the pellucida, this is the extracellular matrix of A. The sperm then will bind to the ZP3 molecule, which act as the Z receptor in zona pellucida, will trigger the echosomal reaction. So when they bind to this ZP3 molecule, it will release the enzyme to digest the zona pellucida. So the sperm head will penetrate into the cytoplasm of the head. Okay, so when the head is already penetrated, the sodium ion channel in egg plasma membrane will be open. So this sodium ion will diffuse into the egg, repeatedly causing depolarization. So action potential will be occur. So this is to prevent the additional sperm diffuse again to the egg. So this depolarization will act as the fast block to polyspermine. At the same time, binding of the sperm to the egg activates signal transduction pathway, which trigger endoplasmic reticulum release more calcium ion into the cytosol. So it will also release more calcium ion into the cytosol again, and then increased level of calcium ion will trigger the cortical granule within the egg fused with the plasma membrane of the egg. So this one will trigger the cortical granule will fuse to the plasma membrane of the egg, and then it will separate the nucleus, the egg, from the gena pellucida. Okay, so this one is as the slow block to polyspermine. So the release enzyme to the space between plasma membrane and zona pellucida layer. So this will trigger cortical action, so will make the zona pellucida away from the head and harden the zona pellucida. And then finally the fertilization envelope is formed. So this is the overall process of fertilization. Okay, so after fertilization, you will go to the implantation. So implantation, the first step of the implantation is about the cleavage stage. So you see this diagram. So normally when they already fertilize egg before, they will go to the 2-cell stage, 4-cell stage, 4-cell stage, 8-cell stage, 16, 32, and even 66, until it form the blastocyst. Okay. Okay, so this is the explanation. Blastula or blastocyst is acceptable. Okay. Now the blastula is surrounding a fluid cavity called blastocele. So you see here, this is what we call as blastocyst, blastula. So inside the cavity, there are the fluid cavity we call as blastocele, and then the outer part of the cell wall is called as trophoblast, and then there are the many cells here we call as inner cell mass. When the blastula is already reached the uterus, it was starting to do the implantation. Okay, so the implantation is occur when the trophoblast, the outer layer of the blastocyst, is initiated into the endometrium wall, and then the trophoblast will release the enzyme to break down the molecule of endometrium. So it will implant into the endometrium wall. So this trophoblast will release the enzyme to break down, so that it can penetrate into the endometrium wall. Again, the trophoblast will extend the finger-like projection because the trophoblast tissue can capture the capillary of the endometrium, because it is also the cell. It requires the nutrient also, so it need to capture the capillary of the endometrium to get the nutrient gaseous entrance at there. So the inner cell mass will form a flat disc with inner layer epiblast and outer layer we call as hypoblast. So you see here, inner cell mass will have the two layer. The inner layer we call as epiblast, the outer layer we call as hypoblast. And then will come trophoblast will continue expanding into endometrium to form four membrane we call as extra embryonic membrane. So this extra embryonic membrane encloses the specialized structure that outside the embryo. Okay, so next, after the gastrulation, after the implantation, we will proceed to gastrulation. So what is gastrulation? Actually, it is a process by which the blastula become a three-layer embryo. So what are their three layer? Three layer we call as three layer is endoderm, mesoderm, and also ectoderm. It will form this three layer cell. So the in human, the embryo is developed almost entirely from epiblast cell. So some of the epiblast, so you know the epiblast is the inner part, right? Inner part of the cell. So all of the epiblast will be made as ectoderm, while outer will allow along the permittive strip to form the mesoderm and endoderm. So epiblast is the raw material that from the endoderm, ectoderm, and also mesoderm. Yeah. And then at the end of gastrulation, the three embryonic drainage layer is formed endoderm, mesoderm, and endoderm. Okay, so the epiblast, will develop into the extra embryonic mesoderm and also amnion. And then our hypoblast will develop into yolk sac. And then the trophoblast will develop into chorion. And then your extra embryonic mesoderm will and four extra embryonic membrane now will surround the embryo. So the eventing of the trophoblast, epiblast, and adjacent endometrial wall will contribute to placenta. So trophoblast is the key component to develop placenta. So how placenta is formed from trophoblast. So you need to know lah. Okay, so after the gastrulation, you will see this structure lah. So there are the three layer. The ectoderm, mesoderm, and also endoderm. You will see this diagram. Ectoderm is the blue color one, and then the mesoderm is the red color one, and the middle one, and then the endoderm is the yellow color one. Okay, after the gastrulation, we will proceed to organogenesis. Okay, so normally organogenesis, so the three germ layer, later they undergo organogenesis to form the organ. So this one is that you need to know that what ectoderm will form, what organ will be formed from ectoderm, what organ will be formed from mesoderm, and what organ will be formed from endoderm. So you need to know lah, all you need to memorize it. So normally it will come out in objective question, or even though it is a safe possibility to come out. Okay, so in organogenesis will consist of three phases, which is about the neurulation, neural plate formation, and then another one is about the neural tube formation, and finally is stomach formation. Okay, so what are they? Okay, so neurulation is the early step in the formation of the brain and spinal cord. And the spinal cord. So firstly, the dorsal mesoderm cell will develop into the notochord first. So this is our embryo. Already after gastrulation, you see that the notochord, ectoderm, and mesoderm, and then endoderm, right? So that the dorsal mesoderm cell was starting to develop into notochord. So notochord is the rod that extends along the dorsal side of the embryo. And then the mesoderm cell will start to secrete the signaling molecule to the notochord, causing the ectoderm to become the neutral plate. So the formation of neutral plate thus is an example of induction process which a group of cell or tissue influence the development of another group through range interaction. Okay, so after the notochord is formed, a neural plate, autococcus is formed, and the neural plate is already formed. Later, the neural plate will change their shape and curve inward, resulting the neural plate will roll itself into the neural tube. So dia akan macam roll itself and curve inward, and then to form the neural tube. So what are the neural tube functions? It will run along the anterior posterior axis of the embryo. So the neural tube later will become the brain in the head and the spinal cord along the rest of the body. And then in contrast, the notochord will disappear before birth, although part persists as the inner portion of the disc in the adult spine. Okay, so after the neural tube is formed, cell migration, the somatic cell starting to form. So cell migration is the long-range migration near the neural tube and including two sets of the cell. So first set of the cell called nucleus. So there are the two sets of the cell we call as nucleus. Cell that develop along the border where the neural tube is pitched off the endoderm. And then the nucleus cell will migrate to variety of tissue including peripheral nerve, teeth, and skull bone. And the second block is about the somae, which are formed when the mesoderm cell lateral to the notochord. So normally this one will dissociate into the mesenchymal cell, and then some mesenchymal cell will develop into vertebra. So yeah, so this is the overall process, the initial process of organogenesis. Okay, next we go to the placenta. Set of the cell called nucleus. So okay, so we know that placenta is the key component that during pregnancy, right? So it is a dish-shaped organ that contains embryonic and maternal blood vessel. So it is normally developed from the trophoblast when the implantation is completed in the second month of the pregnancy. So it is actually maternal portion of the placenta. The main function is to supply nutrient, provide immune protection to the fetus, and also provide oxygen for the gaseous exchange, and also remove carbon dioxide and metabolic waste from the fetus to mother. And then normally we see the structure of placenta. The blood flow from the fetus to placenta through the umbilical arteries, and then the blood flow from the placenta to fetus, from mother to fetus, is through the umbilical vein. And then the material are exchanged at the placenta by diffusion, active transport, and selective absorption. Okay. Okay, so what are the function of the placenta? So normally the main function is to make sure that the blood type of the blood capillary, blood vessel, and fetal capillary are separated, because that normally the maternal blood and the fetus blood group is differ, differ. And not only that, normally the blood pressure of the mother is very high compared to the fetus one. So if no placenta, when it directly pass through the blood is directly passed through from the maternal part into the fetus part, the blood vessel in the fetus will be burst. So it needs the placenta to like a barrier to reduce the blood pressure, and also the difference in maternal blood type to prevent the agglutination, and also protect the fetus, and then prevent the chemical reaction. Okay, and then the placenta will begin cover the role of composition to secrete estrogen and progesterone, and then it will also secrete the human placenta lactogen that promote the growth of mammary gland. And the placenta also protect the fetus against the infection, and then antibiotic from the mother can cross the placenta toward the fetus to provide the passive immunity. Okay, next is about the extra embryonic membrane. Okay, so there are 4 types of extra embryonic membrane: chorion, amnion, allantoid, and also yolk sac. So this one is already after the fetus is formed, the embryo is formed. These are the 4 tissue that protect that can be protect the fetus, the oral function, but they are the key function too. So chorion actually is a fetal portion of the placenta that encloses the entire embryo as well as the other membrane. So if you see here, this one is the placenta, the maternal portion of placenta. At this point, this is the fetal portion of the placenta we call as chorion. So chorion also, because it is one type of placenta, so it is also developed from trophoblast too. So chorion consists of chorionic release that like finger-like projection. So if you see here, this is diagram. So normally this diagram is not show that it is finger-like, but actually it is a finger-like projection to increase the surface area for exchange of substance. Then the main function is to exchange, get the new nutrient to the fetus, and also removing waste from the fetus. Okay. Another one is about the amnion. So normally amnion is the cavity that contains the fluid, amniotic fluid. So normally it's enclosed the entire embryo too. So it will secrete the amniotic fluid to fill the space between the amnion and embryo. So this it will secrete the fluid to fill the amniotic cavity we call as amniotic fluid. So what are the functions of amniotic fluid? Is to prevent the embryo from drying out and permit it a certain freedom of motion. To protect the shock. Serve as the protective cushion that absorbs shock and prevent amniotic membrane. So maybe there are sudden shock, like the mother suddenly get a shock, or suddenly fall down like that. This embryo will protect them. Amniotic fluid will protect the baby. Okay, another one is about the allantoid. So allantoid actually is the network between the embryo and also mother. So it is the outgrowth of developing digestive tract, and will accumulate the small amount of urine that produced by the fetal kidney. So it gives rise to produce the urinary bladder, and beside that it also will form the umbilical blood vessel. And then the last one is about the yolk sac. So normally this is by the least function. So normally it is just the first embryonic membrane to appear. So normally this is play the role in the blood cell formation. Okay. Next we go to the hormone that we will appear in during pregnancy. Okay. So in hormone during pregnancy, there are 3 key hormones that you need to know. The first hormone is we call as HCG, human chorionic gonadotropin, and then estrogen and also progesterone. So in menstrual cycle, if no fertilization or no implantation, your progesterone and estrogen will be decreased, right? But when there are the implantation process, the progesterone and estrogen will be maintained, and then will starting to increase in high level. Okay, so HCG normally will be secreted at the beginning of the pregnancy. So there are 6 hormones that will involve: progesterone, HCG, estrogen, oxytocin, ACTH, and also relaxin. Okay, so HCG, what are the roles of HCG? So normally HCG is secreted by trophoblast or chorion. Actually, it is to prevent the autolysis of corpus luteum. So we know that corpus luteum is the key structure after implantation. It will be maintained and then to secrete the progesterone or estrogen, right? So actually, it is to maintain the structure of corpus luteum, so that the corpus luteum will not be denatured. So, but however, after the first 2 months, where the placenta is already formed from the trophoblast, the corpus luteum function will be replaced by the placenta. So the corpus luteum will starting to degenerate. So when it degenerated, the HCG hormone was starting to decrease also. So the function of HCG hormone also The other function is about the stimulate the entry of amino acid into the cells for protein synthesis, and also development of mammary gland, or also stimulate the glucose and lipid metabolism in the mother. Okay, now is about the estrogen. So estrogen is the function. I think many of the function is to regulate the contraction of uterine muscle fiber lah. You need to maintain the uterine muscle endometrium wall, and also it acts as a regulate. This regulation will stimulate the secretion of oxytocin. Okay, in response to suckle by newborn. So normally by newborn. So normally this one is already after the birth. After the birth, that the mother need to give the milk to the baby. The estrogen level will cause the change of the hypothalamus signal, and the anterior pituitary gland will secrete prolactin hormone. So estrogen also can secrete prolactin hormone. So prolactin hormone actually is the secretion of the milk and then stimulate the production of enzyme to break down the collagen fiber in the cervix wall to make the cervix is more flexible. And then also it stimulates the uterine muscle fiber to produce the corneosin, the protein that form the gap junction between the cell to allow the uterine muscle is contract together. And also it stimulates the development of uterine wall and mammary duct, increase the ATP formation, stimulate the growth of mammary gland, and also inhibit the release of AH, LH, and prolactin. Okay, after that is about the progesterone. Okay, so the progesterone also has a similar function to estrogen, but the difference is that it increases the production of mucus in the cervix to form a plug that protect against infection. So normally in the estrogen, it actually just about the break down the collagen fiber to make the cervix is more flexible, but this one is to make the cervix to produce the mucus. Okay, and it encourage the growth of placenta, increase the size mother breast and uterus, and inhibit the ovulation and menstrual cycle. And also it will inhibit the release of AH and prevent ovulation, and stimulate the growth of mammary duct cell, and high level of progesterone will prevent the contraction of uterus. Okay, another hormone is oxytocin. So normally oxytocin is very high level when the estrogen is very high. When your estrogen level is very high, it will starting to secrete oxytocin. So normally oxytocin is happening in laboring or parturition. Already near the parturition, already starting the female need to birth already. Birthing process is starting to happen. So normally oxytocin is cause the contraction of smooth muscle, increase the power of contraction. So it need to make the mother, the uterus muscle need to have the high contraction power. So because you know that during parturition, during birth, mother is very difficult, right? It's very sangat sakit. So it need to increase the contraction power so that the baby can be pushed out from the vagina. Okay, so normally this is the positive feedback mechanism. Okay, mechanism. So if you see here, why it say that for positive feedback mechanism, when the oxytocin is released, it will stimulate the uterus to contract, and then this when it stimulate the uterus contract, it was starting to release more and more oxytocin, and also it will stimulate to placenta to produce prostaglandin. So this is another one type of an hormone, and then this will also stimulate the more contraction of uterus. So it will stimulate again the oxytocin to release more and more. So this is one of the example of the positive feedback mechanism. Okay, another hormone is we call as ACTH, fetal adrenocorticotropic hormone. Okay, so normally this one is high secretion by anterior pituitary gland before birth. So it is normally high secretion before birth. So actually this one is to stimulate the development of adrenal cortex, and the result in the increased cortisol secretion necessary for lung maturation, for fetal lung maturation. And then this will also stimulate the fetal adrenal gland to secrete corticosteroid, and then this corticosteroid will cross the placenta into the maternal circulation. And then corticosteroid also will decrease the level of progesterone, result in the increase of secretion of prostaglandin. So prostaglandin will contract the uterine wall. So normally this hormone, this is the function. Okay. Now I will go to the relaxin. So normally relaxin is secreted by placenta and ovary. So normally it is to relax the pelvic ligament and widen the cervix in preparation of parturition. Okay, now we go to the parturition process, the birthing process. So there are 3 stages of process: dilation, expulsion, and also placenta. Okay, so normally dilation stage is the first stage of the parturition. Normally is typically last for 8 to 10 hours. So at this time, all the hormone, especially your oxytocin and your progesterone, was starting to secrete in very higher amount. So at this time, the mother is feel very pain, very difficult. Your mother will feel that your lifespan is very difficult, sangat sakit punya. Dia kena sakit sampai 8 hingga 10 jam. So it's very long process. So because dilation, so it need to dilate your cervix. Dia kena dilate your, dia kena besarkan, dilate your cervix, so that your cervix ada lubang yang besar untuk menyebabkan baby awak boleh keluar lah. Okay. Cervix will become affected. It thins out so that fetal head can pass through. So normally at now M9 was usually ruptured, so that it will release the amniotic fluid. So when the big mother is they already got the fluid is released. So normally is the time that the mother need to birth a baby, need to born a baby already. And then the positive feedback mechanism will be operate during this process. So the baby head will push against the cervix. The reflex action cause the uterus to contract, and then the contract will cause against the cervix again, resulting another contraction. Okay. After that it will go to the expulsion stage, where the baby head is already come out. So when your baby head is already come out, it will last normally is between 20 minutes to 1 hour. So normally your contraction of uterus at this time is reach the maximum intensity. So the fetus will be passed through the cervix and vagina, and it is finally will be delivered at the final stage. And then uterine contraction, the woman will bear down so that the fetus is expelled by the combined force of uterine contraction and the contraction of abdominal wall muscle, and hence the baby is still connected to placenta by the umbilical cord. And then contraction of uterus will squeeze much of the fetal blood from the placenta into the infant. So this stage is very dangerous stage, where the blood will be rushed out, because now your placenta is starting to release out, starting to rupture, but the placenta still is connected with the baby, but your placenta is ruptured from the uterus wall already. So there are many blood will become out at this time. Okay. Now is about the placenta stage, which is we need to remove the placenta. So after the birth, after your baby is already come out from the uterus, so that your placenta and your fetal membrane are loosened from the lining of the uterus by another series of contraction and expel. So this stage, or collectively, or you can call as afterbirth, because this tissue are expelled from the vagina after the baby has been delivered. So the placenta and the fetal membrane, your umbilical cord, now we're starting to deliver out from the vagina, and then the laboring process is completed. Okay, so that's all for the sexual reproduction in human. Okay. So now it's 6:20, so we take a break first. We will continue at 6:30. Okay, so see you later. And then the next time is the last class. I will directly finish up the sexual reproduction in flowering plant, and also the seed germination, and also growth pattern. Okay, so thank you. So see you later. Hello. Okay. Okay, now is already 6:30, so I will begin our last class now. Okay. Okay. So we will continue about the last class, chapter 10, reproduction development goals. So now we will begin about the sexual reproduction in flowering plant. Okay. Okay, so first of all we need to learn about the flower structure lah. So I think flower structure in flowering plant, I think I don't need to mention many part. So we know that in each flower we contain stamen and also carpel, right? So stamen is the male organ, and also carpel is the female organ. And then under stamen we have anther and also filament. And then the carpel we have stigma, style, and ovary. Okay. Okay, so yeah, this one is the explanation. Okay, next we go to the life cycle of the plant. So the life cycle of the plant is special compared to human. So the plant have the life cycle we call as alternation of generation, meaning that the plant will spend their part of their life in multicellular haploid stage and also in part of a multicellular diploid stage. So if you see here, so normally you see here they are separated into two form, what? Sporocyte and gametophyte. Gametocyte. So sporocyte is the adult plant. So after they will have the sexual organ, right? The sexual organ, like the stamen and the carpel, they will undergo the meiosis to form the spore, right? To form the spore, so that they will have the gametophyte. So when they produce a gamete, they will go through the fertilization process to form the zygote and embryo that is already sporified. So it is the alternation of gut generation of the plant that it contains haploid stage and also diploid stage in their life. Like human does not have. Human only have diploid stage only. Only the human gamete is haploid. But in the plant, yeah, you can direct the whole plant can be in the gametophyte stage, which is haploid. Okay, so this one is normal general for the plant, but in flowering plant this is the summary process. Okay, so normally you see here, this is our mature plant, right? Mature plant. So we know that in our plant, we in the ovary and also anther contains the microsporocyte and also megasporocyte, right? Right. So this is the initial this is the spore that will undergo meiosis process to produce gamete. So when they already produce gamete, it will starting go to the haploid stage. Okay, so when the gamete is produced, this gamete is produced for enter microsporocyte, will undergo pollination microsporocy to produce the pollen grain, and then the megasporocyte will produce the megaspore, right? So once they already produce megaspore, this pollen grain was starting doing the pollination process, and then the developing pollen tube, and then will go through the pollen tube, and then they will undergo the double fertilization, right? And then once they undergo the fertilization, embryonic development is formed, and then after that seed germination is produced, and then it will produce another adult plant again. So this process is repeat again. So you will see here the stage of the haploid stage is the process that pollen grain is produced and the megaspore is produced. When they are produced, they will undergo a pollination and double fertilization. Once they finish the double fertilization, they will return back to the sporophyte generation, and then this generation they will repeat again. Okay, see, this is another diagram. Yeah. So normally I use this diagram explain more detail. So normally if you adult plant, adult plant. In the anther or stamina, each microspore contain microspore set, right? So they will divide, undergo meiosis to form microspore. And then microspore will develop into a pollen grain, and then we know that the major pollen grain contain two nucleus, right? Tube nucleus and also generative nucleus. And then at that same time, your female part, the ovary, was starting to undergo the mega sporagium was starting undergo that meiosis process to form the megaspore structure. So one form the megaspore structure, so megaspore, and then finally it will form the embryo sac structure. So we know that in embryo sac structure there are 1 2 3 4 5 6 7 8 nucleus, right? 8 nucleus there. So there are the 3 nucleus at the top we call as antipodal cell, and then there are the 2 central nucleus at the center, and then at the bottom at the middle one is we call as egg cell, and then the left one at right hand side is about the synergic cell, right? Okay. So when the embryo sac is formed, pollination was starting to form. The pollen grain was starting to pollinate to the stigma of the plant. So they will have the two nucleus, which is the tube nucleus and our sperm. So the tube nucleus will be penetrate throughout the stigma to create the pollen tube until it reach the integument. And then at the same time, your generative nucleus will be divided into 2 nucleus again. So this one we call as sperm cell lah. Okay. So when they already reach when the tube nucleus is already reached the integument of the plant, it was starting to penetrate it to let the 2 sperm cell can penetrate into the embryo sac. So the 8 nucleus, one of the 8 nucleus will be penetrate. One of the sperm cell will penetrate into the 8 nucleus, and then one of the egg cell will penetrate into the center part. We call as polar nucleus. So that is why that we have double fertilization. The name double fertilization because it consists of two fertilization process in one time. So first fertilization is in the egg nucleus, and another fertilization is in the polar nucleus at the center. So once the fertilization is formed, it will form the zygote 2N haploid, diploid zygote, and also form the triploid endosperm. So this one will form 3N. This one will develop into the endosperm that provide the nutrient, and then this one will develop into the zygote for embryonic development. So that after that they will form the embryonic development process. After that it was from the seed germination. So in our syllabus, this one is about the double fertilization process. We need to know about the double fertilization process. So I think double fertilization process is beginning from the pollen grain formation, that from the anther stamen. Okay. So at the finally they will form the pollen grain that contain the tube nucleus and also generative nucleus. And then also embryo sac formation, it will finally form the embryo sac that contain the 8 nucleus, antipodal cell, polar nucleus, and also synergic cell synergic cell. Okay, and also 8 nucleus there. Okay, and then this is about the embryo sac structure lah. I already mentioned it. After that, they will undergo for double fertilization. So double fertilization will be start when the pollen grain is starting to pollinate into the stigma of the plant, and then the tube nucleus will form the pollen tube until it reach the integument, and then at the same time, your generative nucleus will starting to divide into 2 sperm cell. So that these 2 sperm cell, after that, it will penetrate the integument and then reach the embryo sac. And then one of the sperm cell will penetrate into the egg cell to form haploid zygote, and then one of the nucleus will penetrate into the 2 polar nucleus to form the 3N endosperm. Okay, so this is a double fertilization. Okay, next is about the seed embryonic development. Okay, after we already formed the zygote, zygote is formed. The zygote was starting to divide mitotically to form the terminal cell and also basal cell. So the terminal cell is the cell that play the role in embryonic development. However, a basal cell will only just support the terminal cell only. So the terminal cell was starting to divide until it formed the pore embryo stage, heart stage, until it formed the cotyledon and the seed. The final development is from the seed. Okay, so the first stage is about the pore embryo stage. So after fertilization, the chopper nucleus will develop into form the multicellular endosperm. So the endosperm actually store the nutrient at all. Okay, so normally the zygote will undergo mitosis to form the multicellular embryo, and then they have the basal cell and terminal cell, right? So this is terminal cell, this is basal cell. So normally basal cell is at the super sensor. It just support the terminal cell only. So the super sensor cell will transfer the nutrient from the parent plant or endosperm to embryo. So the super sensor cell will also push the pore embryo into the embryo sac cavity. So the last point is that the terminal cell will divide by mitosis to become the pore embryo, which will attach to the super sensor cell. Okay, then when the pore embryo stage is still undergo mitosis again, it will form the globular embryo. Now it's called the globular stage, and then this globular embryo will develop through mitosis into two cotyledons for monocot, but one cotyledon for dicotyledon. So this one is very important. You need to learn for monocot plant and dicot plant, the cotyledon is differ. Okay, and then after that, when the globular embryo is further developed, it will form the heart stage, and then it will finally go to the torpedo stage. As the embryo, after the torpedo stage, it often curve back to itself and crush the super sensor. So the embryo cell between the cotyledon will differentiate into apical meristem of the shoot. So the embryo cell that near the super sensor will develop into the apical meristem of the shoot. Okay, so now is the final stage. So final stage, it was starting to develop the seed lah. So if you still remember the outer integument, so the outer integument will develop into testa, and then the inner integument will develop into the tegment, and then the nuclear central part of the ovule is broken down to be used by the embryo, and then the ovary wall will develop into pericarp that will further develop into the fruit, and then finally your seed tissue will undergo hydration and seed dormancy is induced. So this is the final process that your seed is fully enveloped. Okay, next is about the fruit formation. Okay, so the fruit normally is formed from the ovaries. So there are the three main parts of the fruit we call as epicarp, mesocarp, and also endocarp. So this part of the fruit, or in general we call as pericarp lah. So normally this pericarp, if you already know, ovary wall. Okay, and then when ovary wall, pericarp is developed from the ovary wall. So normally pericarp may become the fleshy or outer layer or covering. So the ovary will develop into the seed. So this one is ovule is the embryonic development that we discussed just now, and then the fruit development will continue until the seed mature. Okay, next is about the seed germination. Okay, so if you still remember, the hormone that play the role in seed germination is gibberellin, right? Okay, so this is the process of the seed germination. So it's very simple process, I think. So germination is the process of seed sprouting out, so that the seed was starting to have sprouting out the root and shoot. Okay, So germination is initiated by water imbibition. So it is a water imbibition that that high amount of water is absorbed by the seed, so that the seed will be expanded and the seed cord will be ruptured, and then this will trigger the change of embryo that is releasing gibberelli. So the seed will contain the gibberelli, and then when it already absorb water, the seed cord is ruptured, and then gibberelli will be released. Okay, so what are the functions of gibberelli? So gibberelli will diffuse into the alluvial layer, which is made from protein, and then the gibberelli will switch on the gene to synthesize specific protein in the alluvial layer, and then the alluvial layer will secrete the hydrolytic enzymes such as alpha amylase, maltase, and protease. So this enzyme is required to hydrolyze the nutrient inside the endosperm. We know the endosperm is rich in the nutrient, right? So this hydrolytic enzyme will hydrolyze the nutrient inside the endosperm. Okay, and then this digested nutrient will be transported to the embryo, so the nutrient will be used for respiration and growth embryo. And then finally is that the germination is completed when the seed produces the vesicle and plume. So if C refer to this, and there are the vesicle and plume is already released, is already formed, so that the seed germination is completed lah. And then after that, the rooting and shooting will be produced. And then this is the factor will affect the seed germination. So you see that there are 8 factors that can affect the seed germination. So water, the main water lah, oxygen, temperature, and also light intensity, and also pH value. Okay, next we go to the last topic for today, which is about the growth of the growth. Okay, so firstly we need to learn about the growth curve first. Okay, so there are 3 types of growth curve you need to learn: absolute growth curve, absolute growth rate curve, and also relative growth rate curve. So what are their difference? The difference is that the parameter used is different. So absolute growth curve is normally the growth parameter is plotted against the time. So meaning that your y-axis is the parameter that you use. Normally in plant is dry mass. You use dry mass, and then against the time. So you just record the growth of the plant according to the time. So when the time is increasing, your growth also increase. So this is called as absolute growth curve. Okay, and then this one is about the absolute growth rate curve. So when you see the growth rate, when they are the rate at the graph, meaning it involves the differences. It involves the change in parameter, the differences between the parameter result. So normally you need to require to final parameter result minus initial parameter result divide by initial parameter result. This is the key point that you need to know lah. So this is because of change in parameter, but the axis is still same. It will produce the time, so it will produce a bell-shaped curve like that. Another one is about the relative growth rate curve. So you see the relative, normally it will begin from 100% first. So meaning that this is a change growth rate time, show the efficiency of growth. That is the rate growth relative to the size of the organism. Okay. Another one is about the growth pattern. Okay. So normally there are limited growth and unlimited growth lah. So limited growth normally is happening in human. So in limited growth in human, there are many phases: lag phase, exponential phase, stationary phase, maturation phase, sensor phase, and dead phase. So I think lag phase is normally is the human like us is normally is age from 0 to 12 years old lah. 12 years old, meaning that our age is very baby stage until you enter the primary school. This is what we call as lag phase, because our growth rate at that time is very low, and then your cell divide mitotically and differentiation is very slow too. However, when we reach the puberty age, which become about between 12 to 18, exponential phase will be occur, meaning that the growth rate at this time is very high, is the highest, and then cells are actively divided and elongated. Okay, and organism will grow rapidly and increase their size rapidly. And then when we reach to the stationary phase, the growth rate is starting to slow down and reach constant now. and then the organism are already reached the maximum size, meaning that the organism like human already cannot become more taller again. It will cannot become more, cannot increase their height already, maximum size already. Okay, so normally this one growth rate will be limited by internal factor or external factor lah. So normally this one is already you are in adult lah. Okay. So normally when you reach the maturation stage, yeah, your organism already reach maturity. No, the growth rate now is constant, no growth at all, and then your cell division now is equal to the rate of cell mortality, meaning that your cell division is only take place to replace or repair the dead cell only. It will not increase the number of cells again. And then when you reach the sensor phase is that your growth rate starting to negative or starting to decrease. So normally is starting to aging lah, akan menjadi bertua lah, jadi orang tua lah. Until that, finally we are human, we will undergo that phase, meaning we will die. Okay, this one is for human and also for plant. For plant, there are also limited growth in annual plant. Annual plant normally is non-woody plant lah. So if you see here, the curve shape so similar to human. It is also sigmoid curve, sigmoid curve shape. So you compare to human is also show the sigmoid growth curve. But in plant, at the initial lag phase, they will decrease. The growth rate is decreased. Why? Because due to the seed germination. Because during seed germination, they need to use up the energy. They need to use that energy to break down the nutrient for seed germination. Seed germination. That is why your either initial lag phase, the growth phase of the plant is decreased. But for the unlimited growth in plant, okay, it's only meaning that it is unlimited, meaning that the plant will be survived at any space, at any throughout the lifespan, but unless the human terbang pokok tu, if not the plant will not be die. Okay. So normally it consists of a series of smaller sigmoid curves. So actually it is the combination of the sigmoid curve lah. Combination of the sigmoid curve in the annual plant. So dia akan menjadi macam ni and then decrease and then dia jadi macam ni again. Yeah, so this is the unlimited growth in plant. And then we also have the allometric growth in human. So allometric growth is the growth that our organ of an organism occur at a rate that is different from the rest of the body, meaning that your organ development is different from time to time. So if you see this diagram, this graph more important. You will see here, the development is starting first when your head is the first organ that start to develop. After that, they will lymphoid tissue, all the lymphoid organ, lymphatic vessel will start to develop, and then this red line is the overall growth curve, and then the reproductive organ is the least part that the organ to be developed. Okay. And then another one is isometric growth in fish. So normally this one is referred to the organ that organism develop at the same rate as the rest of body, meaning that the organism increase its size with the same rate. Same rate one. So there when it is increase its size, the organ is increased same, and also the body size is also same. So the size of the body organism will change along the time. So however, in this situation, change in size of the organism is not accomplished by a change in the shape of the organism. And another one is intermittent growth in arthropoda. So all the arthropoda, I think you are already finished cemetery, you know what is arthropoda lah. So all the insect, all the arthropoda organism will exhibit to the intermittent growth. So normally intermittent growth is a discontinuous growth. The growth is not continuous like the human or plant like that. Is indiscontinuous, meaning that the growth is suddenly increased one. Because that the hard skeleton does not grow but must be shared for the organism to grow. So normally the growth it needs to occur, mounting process and adhesive process is required first. Only this process form, then the organism can grow. If not, the hard skeleton cannot can already inhibit the growth of the organism. Okay, now is about the ADC. So if you know that every point that the organism, that the insect starting to go at here, ADC was starting to occur, right? So ADC actually is a molting process where your old exoskeleton of the arthropod are replaced by a new soft layer and leg become harder again. So during ADCs, arthropod need to absorb more water to expand its body. So normally breaking of old skeleton will form the new soft exoskeleton. So before the treating exoskeleton become harder, the arthropod will increase its size of body until the exoskeleton is hard. Okay, and then there are another process we call as metamorphosis. Mainly is special for insect, that there is change in shape occur very suddenly, and the process of changing from one stage to the following stage in insects. So there are two types: complete metamorphosis and also incomplete metamorphosis. So incomplete metamorphosis is that the insect will remain similar structure, but for the complete metamorphosis, the structure is totally different. Every stage the totally is different. So you will see the butterfly, the children of butterfly is caterpillar, right? After that they will form the moth, they will form the pupa, and then directly form the butterfly. So it's totally different at all. But for incomplete, the structure is similar, just that it increase its size only. Okay, so the role of hormone in metamorphosis is the last topic for today. So normally in the brain of the insect, it will contain the neural secretory cell, and then the corpus caudicum and also corpus allotum. Okay, so the corpus caudicum is the site to secrete PTTH. So you see PTTH, the full name is porthoracicotropic hormone PTTH. So in exam, in structure question, you need to write in full name, cannot write in short form. Even though in essay question, you also need to write in full name at the first sentence. And then PTTH will be transported to the corpus caudicum and stored at there. PTTH was stored at there first, and then the porthoracic gland will secrete the addison when the PTTH is released. So when the PTTH is starting release, porthoracic gland is released. It will stimulate the porthoracic gland. It will secrete the addison. So addison actually is the growth hormone, responsible to triggering disease and development of adult characteristic only. Okay, so corpora allotum, the endocrine gland, were found in the immature insect will secrete juvenile hormone we call JH. So normally JH is inhibition for adult characteristic. It maintains the insect in the children state. So if you see here, the high concentration of juvenile hormone will ensure that the insect is in the children state too. So only the ADC is formed, and then metamorphosis is no occur. However, when the JH hormone is starting decrease, and then your additional hormone is increasing, so your metamorphosis will occur, and then the insect will transform into the pupa, and then it will replace the larva structure in the adult form within the pupa. And then when the JH hormone is absent, the pupa will mount and become an adult. So this is what we call as butterfly. So at here, low JH hormone, it will already now become pupa already. When the JH hormone is totally disappear, it will form the adult. Okay, so that's all for today's topic. Okay, so that's all for today. Okay, now it's already 7 o'clock. Okay, so first, so hopefully today's revision for this 4 chapter, you guys can get the information, can get the recap from us from that. Okay, so I think that's all for today. So thank you guys for attending class today. So tomorrow we will have other 3 chapters that we will learn. So we will see you again tomorrow 8 a.m. lah. Okay, so thank you and have a nice day. Have a good rest too.